please let me know when I should start. Hi everyone, good evening. Apologies for this little hiccup. There was some technical issue with my iPad only. Anyways, uh, a warm welcome to all of you to this session. In this session, as you have been told already, I'll cover the important topics of the FMGE exam. I'm Dr. Mamta Jawa, microbiology faculty, and I've been teaching medical students, medical graduates, second MBA students since 10 years. And it has been an absolutely uh, wonderful teaching, budding medicos. And teaching is my passion. It's not more than more than being passionate about microbiology. I think teaching is extremely therapeutic and spiritual for me also. Okay, I hope you guys have one second, guys. I hope you guys have downloaded this PDF. If you have some tab, you can make the notes, with me on the tab. Or if you have hard copy, that's also wonderful. Whatever I'm going to write in today's session, please jot down all of that in your workbook. And I've selected the most high yield topics for your upcoming FMG exam. And my aim as a teacher in this session would be to make sure that you all are able to remember just by one revision at least 50 to 60% of whatever I'm speaking today. Of course, after this session I expect you all to revise also. Okay, just a question to all of you. How many of you would want me to use mixed language, that is both English and Hindi, and how many would be comfortable with only English? Because I'm getting requests of using English, but I suppose not everyone knows Hindi, so we can continue the session in complete English also, because I would want each and every student to understand what I'm teaching today. Okay, no issues, I'll stick to pure English. Okay, understood. All right, guys. Okay, microbiology is completely, incomplete without talking about gram stain. As the name itself says, gram stain. It holds a lot of weightage for your exams. No lab is there in the world which does not do gram stain. It is the most important differential stain done in any microbiology lab the world over. Okay, so it was devised by Sir Hans Christian Gram. To honor him, this technique was given the name gram stain. And what is the basis of gram stain? Guys, whatever is there in this workbook, all of them are MCQs. Each and every line is an MCQ. So what is the basis of gram stain? The basis is differences in the cell wall of bacteria. Now, who will tell me what is the bacterial cell wall made up of? The cell wall of bacteria has got a very fancy name. The cell wall has got a very fancy name. What is the name? Who will tell me? All right, guys, don't worry. In case I happen to mention something in Hindi, I will translate it to English also. So I'll make sure that whoever even does not understand Hindi, they are not. Don't worry, you're not going to miss anything. Fantastic. Peptidoglycan. ठीक है? Peptidoglycan. ठीक है? अब देखो, पेप्टिडो मीन्स प्रोटीन, ग्लाइकन मीन्स शुगर. सो सेल वॉल इज अ कॉम्बिनेशन ऑफ प्रोटीन and sugar. Now, glycan part, see, peptido is the prefix and glycan is the suffix. Always the suffix is going to be, more in amount. So glycan part adds to what? The weight or the bulk of the cell wall, and peptido part confers strength to the cell wall. ठीक है? Rahul, it is not teichoic acid. Please note. I'll write it over here. Teichoic acid. What is that? Teichoic acid is an example of an adhesin which is present in G plus bacteria, gram positive bacteria. ठीक है? It is not part of, peptidoglycan is what constitutes the cell wall. Teichoic acid arises from the cell wall. It does not make the cell wall. Please remember this. Now, gram stain classifies bacteria into gram positive, which appear what? Blue in color, blue or violet in color, and G minus, that is gram negative, and such bacteria appear pink or red in color. Always remember, blue is the color of positivity. So remember, gram positive bacteria will appear beautifully blue under the microscope, whereas red reminds us usually of what? War, bloodshed, something very negative. Now, people who have red as their favorite color, please don't get offended by this statement of mine. This is just a little memorizing tip for all of you, that gram negative bacteria, they are going to appear reddish or pinkish under the microscope. Is this clear? Tofika, yes, purple is mentioned in certain books. Let's write that as well. So all these three, adjectives are used synonymously for gram positive. They appear blue or violet or purplish. I hope this makes sense. Now let's understand the steps of gram stain quickly. As you can see on the screens, there are 4 basic steps. Now, gram stain, obviously, as I said, it's an example of what? It's an example of a differential stain. Differential stain automatically implies that it is going to help us differentiate between at least 2 types of bacteria. Based on what? Based on what? Based on the differences in their cell wall. Yes, even I can notice that medical guideline. And guys, just give me a moment. I'll just quickly text the tech team regarding the echo problem. Just give me a moment, please. No, no, mobile audio is off only. All right. I think there's no echo now. Okay. I texted the team. All right, guys. Now, step number 1 is what? Step number 1 is the addition of a para-rose aniline dye, that is, for example, crystal violet or methyl violet or gentian violet. As the name itself implies, these are violet color stains. These are violet color stains. All right. So when you add any of these dyes, be it crystal violet or gentian violet or methyl violet, all the bacteria which are there in the smear, they will take up this color very well. All will appear bluish or purplish or violet in color. Okay, after that you have to wash the slide. The duration is 1 minute. 1 minute के लिए you will keep the stain on the smear. The smear is there on the slide. ठीक है? So at this moment, if you see the smear under the microscope, everything will look beautifully blue. ठीक है? Now, after 1 minute, you have to wash the slide under tap water, and then you have to add what? Iodine, grams iodine, which serves as mordant. Mordant simply just understand it as fevicol or fevistick. So itself, it is not going to impart any color to the bacteria. It is just going to make sure that the primary stain which you have added, that is crystal violet, it sticks to the bacteria. So here also, as you can see, there is no change in the color. Okay, this again will be kept for 1 minute. After that, you will wash the slide under tap water. And now comes the most important step in gram stain, that is decolorization. Very, very crucial step. Now, in this diagram, what has been mentioned? Alcohol wash. Now, alcohol is an organic solvent. You can use what? You can use even acetone or alcohol acetone mixture. Now, girls in the comment section, please let me know what is there in nail polish remover, by the way. Wait, let me message team again. Fantastic. Nail polish remover has got what? Acetone. And the MCQ which is asked regarding this is that acetone is the fastest decolorizer. Acetone is the fastest decolorizer. Okay. So if you use acetone, you don't have to wait at all. You just have to wash the slide. And if you're using alcohol or a combination of alcohol and acetone, then you will be told by the lab people regarding the duration, whether it is 10 seconds or 15 or 30 seconds, depending on the composition, depending on the concentration of the alcohol being used. Okay. So after that, again, you have to wash the slide. You have to wash the slide after every step. And then final step is going to be what? Final step is going to be the addition of counter stain. What is the meaning of the word counter? The meaning of the word counter is opposite. Opposite. Meaning we have to use a stain which is going to give us a very good contrast with the violet color. Agar aap koi, if you use any blue color stain again in the fourth step, then we are not going to get a good contrast, है ना? What is going to give us a good contrast with the violet color? Some reddish or pinkish color. So here the counter stain is saffronin. Okay. Or another example you can write down, you can use dilute carbol fusion. ठीक है, dilute carbol fusion. Please remember, sin ये जो word है ना fusion, इसके end में क्या आ रहा है? The end has got sin. Fusion. Now, please remember, in Zn stain, the primary stain becomes concentrated carbol fusion. Please remember this as well. All right. So at the end of these four steps, we will get some bacteria which have retained their, blue color. They are always, they always have a positive mindset. So gram-positive bacteria are, are teeming with positivity. They will not lose their blue color right till the end. Whereas those bacteria which are a little bit negative, they will give into the step 3, that is organic solvent. They will lose color. And who comes to their rescue? मतलब उनकी colorless life में color कौन add करता है? Who adds color to their colorless lives? The counter stain. The counter stain. So obviously it's a differential stain. So we have to use two stains which give us contrasting colors. If you see this picture, which is frequently asked in exams, if I ask you to interpret this, what do you see? How will you comment? How will you make a conclusion कि भाई patient को किस चीज से infection हुआ है? By the way, when somebody asks you to comment on gram stain, you have to comment on two things: gram reaction, that is gram positive or negative, and the shape of the bacteria. So please let me know in the comment section. In this picture, you can see some round things which are blue in color, and some, this cylindrical structures like this pen is, which are pinkish in color. So what is the interpretation of this gram stain picture? Quickly tell me. क्या लिखूं? So I can see gram positive cocci. So these round things, by the way, these bacteria which are round, they are referred to as cocci. But please remember, all cocci are not round. But when we generally use the word coccus is singular, cocci plural, we are referring to berry-like bacteria. Berry-like matlab round or spherical bacteria. So I can write over here that I can see gram positive cocci, and if I can notice some specific arrangement, I should comment on the arrangement as well on gram stain. Always arrangement is not going to be there, it is not going to be visible, but if it is visible, if it is clearly visible to you, then you have to make a comment on the arrangement also. For example, in this picture I can safely write ki gram positive cocci are present as grape like clusters. Okay, grape like clusters. Plus I can see gram negative rods or gram negative bacilli. Bacilli is plural, bacillus is singular. Rod shaped bacteria are like this pen, that is, they are cylindrical. Another adjective for rod shaped bacteria is bacillus. Bacillus singular, bacilli plural. I hope this makes sense. Any doubts so far in gram stain interpretation? Okay, so this picture is showing us what? Gram positive bacteria, gram positive cocci, which are spherical and they're arranged as grape like clusters, and gram negative rods. Please remember, we are not supposed to write the name of the bacterium, because we just have to describe what morphology we are seeing. We have to comment on the shape, the color, that is gram reaction. A battery of tests is then conducted. It is carried out to find out the specific identity of these bacteria. So we microbiologists are like CID agents who are just after the life of these bacteria which we see under the microscope or whatever has grown on the cultured media, so that we can help our patient by giving them correct diagnosis, so that correct treatment can be commenced. Is this clear? No, no, I've used green color. I don't know why it is not visible. Wait, I'll rewrite it. Guys, I've used green color. I don't know why it is not visible. Wait, let me change the pen. Gram positive cocci arranged in grape like clusters and gram negative rods or bacilli. Okay, now I got it, because in chroma here we are not supposed to use green color. Okay. Now I hope everyone can read this interpretation. Very, very important. Okay. Now, okay, now. Okay, my bad. I forgot that when we are using chroma, we are not supposed to use green color. Okay, चलो आगे बढ़ते हैं. Next important differential stain is what? Zeal-Nielsen stain or acid-fast stain. It was devised by not Zeal or Nielsen. Zeal and Nielsen, they are the names of two people, two German bacteriologists who had modified Zn stain. ठीक है? Zn is the short form for Zeal-Nielsen, also known as acid-fast stain. This stain was devised by Sir Paul Ehrlich, who is regarded as the father of chemotherapy. Again, MCQ. Whatever sentences are there in this workbook, as I said before, sab kuch MCQs, and everything is important. Okay? It was modified by Zeal and Nielsen. Okay? Please remember, it is ZIEHL. Okay? ZIEHL. Now, what does acid fast means? Acid fast means acid non decolorizable. Now, let me know in the comment section what will be the outcome when acid falls on clothes accidentally, or it is spilled on furniture. What is the outcome? So basically, the furniture or the clothes will lose color. That is going to be the outcome. Okay, that is going to be the outcome. But when we say acid fast, it means when we treat something with acid, that object, that structure will not be decolorized. So it is very, very resilient to acid. So remember, acid is like an adversity, some problem, but something which is acid fast is not giving in to that adversity, to that problem. Somebody is not losing their charm, in other words their color, when that organism is being treated with acid. So there are certain organisms which are said to be acid fast. That is, let's say if you stain them with some stain and afterwards you treat them with acid, what do we usually expect? The color should be lost. Whatever colored stain we had used initially to stain those organisms, the color should be lost on subjecting them to acid. But ये वाले organisms के साथ ऐसा नहीं होता है. These organisms on the other hand, they will retain their color. So such organisms are referred to as acid fast organisms. And when we are suspecting infection with acid fast organisms in some patients, then we are not supposed to use the most commonly used differential stain, that is gram stain. We are supposed to use Zn stain, Zeal Nielsen stain. Is this clear? Is this clear, guys? Again, it's a differential stain, just like gram stain is. So two different stains are going to be used, which give us different colors. So the first step over here also is application of the primary stain, that is carbol fusion. Okay, carbol fusion. Now, when we apply carbol fusion, we also have to do heat application. We have to apply heat to the bottom of the slide. You know why? Because when we say acid fast, we mean these acid fast organisms have got what? They have got mycolic acid. Mycolic acid, which is a lipid present in their cell wall. And now I'll give you a good analogy for mycolic acid. How many of you really like it when some relatives see? It's not that all we don't we dislike all the relatives, but I'm sure all of us have some relatives who we really don't like that much. So mycolic acid is is like a relative of these bacteria, that is acid fast bacteria. These acid fast bacteria actually does not have too much of liking for the mycolic acid. You know why? Because mycolic acid being a lipid, it's a very resilient substance. It is not going to allow the entry of the stain into the bacteria, so the bacteria would remain colorless. So if we are suspecting acid fast bacteria infection in some patient, we are obviously dealing with these bacteria which are having mycolic acid, and mycolic acid, as I said, will not allow the entry of stain into these bacteria. So that is why it is very important to apply heat to the bottom of the slide, because heat will make sure what? Heat will ensure liquefaction of this lipid so that the stain can enter. Okay, so if you want to impart some color or stain to these acid fast organisms which have got mycolic acid, which hate stain, please make sure that you have to devise some way to allow the stain to penetrate these acid fast organisms. And that penetration of a stain inside the body of these bacteria, that is done by heat. So application of heat is very important, and over here it is going to serve as the mordant. So when you have made a smear from a patient in whom you are suspecting infection with some acid fast organism, make sure you apply heat, which is going to serve as the mordant. So whatever organisms are there in the specimen, be it acid fast or non-acid fast, they will take up the stain. See, there is no problem with non-acid fast organisms. They will take up the stain even if you don't apply the heat, but your objective is to not miss out on the acid fast organisms. If you don't apply heat, acid fast organisms will not take up the stain. So heating is very, very important. Okay, and obviously after every step you have to wash the slide. That is sort of a given, you have to remember that. And now comes the third step, that is application of what? A decolorizer. Most commonly, mineral acids like H2SO4 are used most commonly. So over here, as you can see, some bacteria, the gold round bacteria, they have lost color. Okay, they have lost color on use of decolorizer. Now, most commonly in Zn staining, we use H2SO4 only, but yes, combination of acid alcohol also, is used in a few places. So irrespective of whatever you are using, some bacteria would lose the color. Now, these are those bacteria, obviously, which are non-acid fast. Now, the final step is what? Application of the counter stain, that is methylene blue. As the name itself implies, methylene blue will be now taken up by the colorless bacteria. I mean, those bacteria which had lost the color after decolorization. So they will appear blue. So the acid fast organisms are going to appear pinkish, beautifully pink. They will appear, and the non-acid fast organisms will appear what? Blue, because the color of the counter stain is blue. Is this clear, everyone? Are the steps clear of this stain, differential stain, Zn stain? And look at this. This is a beautiful picture. This organism was later on found out, was later on confirmed to be Mycobacterium tuberculosis. But जैसे मैंने आपको बोला, you are not supposed to write the name of the organism. ठीक है, when you are interpreting some smear. ये तो मैंने इसलिए बताया because this organism actually later on culture and other methods was confirmed to be MTB. So this is how MTB looks like when it is subjected to Zn staining. So what is the interpretation of this smear? Long, slender, pink, that is acid fast. B for bacteria, seen morphologically, and then we write in the report morphologically resembling Mycobacterium tuberculosis. Okay. Okay, this has been typed in green color. That is why, Anandu. Don't worry, I'll overwrite on it. So genus Mycobacterium tuberculosis. But in your soft copy of the workbook which you have downloaded, and in the print copy, it will be okay. All right. So Mycobacterium tuberculosis के लिए what percentage of H2SO4 is used? 25%. Previously this was 20%. Now it has been changed to 25%. And for M. leprae 5%. All of these are MCQs. For bacterial spores कितना percentage use करते हैं? 0.25 to 0.5%. नो डॉक्टर केवल ऑल एसिड फास्ट के लिए नहीं करते हैं. डिपेंडिंग ऑन व्हाट एसिड फास्ट ऑर्गेनिज्म यू आर सस्पेक्टिंग, based on your suspicion you have to decide the concentration of H2SO4. If you are suspecting nocardia, you have to use 1%. Just a little memorizing tip from me to all of you. Let me write the name nocardia like this. Okay, now let me underline N and O. Let me add a reverse arrow like this, and let me prefix the name nocardia with what? Small e. And let me encircle it. Read it backwards. क्या मिल रहा है आपको? 1. So nocardia के लिए 1% H2SO4 is used. Rhodococcus के लिए भी 1%. Head of sperm के लिए 0.5 to 1%. Legionella migdadi also 0.5 to 1%. Legionella migdadi is the name of a bacterium. Please remember all these percentages. इतना मुश्किल नहीं है. MTB, let's revise. Let's revise together. MTB 25%, M. leprae 5%, nocardia 1%, स्पोर्स के लिए सबसे कम है 0.25 to 0.5%, एंड बच का गया हमारे पास हेड ऑफ स्पर्म एंड लीजियनेला मिगडाडी 0.5 to 1%, एंड हू सिस्ट ऑफ कॉक्सीडियन पैरासाइट सबसे आसान 1%. 1% याद हो रहा है साथ साथ? Okay, coccidian parasites बहुत important है. The 3 important coccidian parasites are these. Who will tell me why they are so important? Why they are so important? Cryptosporidium parvum, Cystisospora belli, which by the way is the new name of this parasite. Previously it was known as Isospora belli. और तीसरा क्या है? Cyclospora catenensis. Why are these fellows important? Fantastic, sort of wonderful. Refractory diarrhea in HIV positive patients. अगर आपको या मुझे infection हो गया, by God's grace our immunity is good, so we will develop self-resolving, self-limiting illness. ठीक है, diarrhea will resolve on its own. But unfortunately, in HIV infected individuals, these three fellows, these three villains, they cause what? Refractory diarrhea. Okay, fantastic. Now, so all of these are important MCQs. Please revise all of this very, very well. Now we come to another very important PYT previous year topic, that is the biological functions of antibodies. Firstly, we'll start with, by the way, there are 5 classes of antibodies and their serum concentration. Their serum concentration can be simply remembered as by remembering GAMD. Let me write GAMD and E, or बस ऐसे बना दो, हो गया काम. GAMD. IgG maximum concentration followed by IG, AMDE, GAMD. Now let's start with IgM. It's a very royal antibody. I personally call it extremely royal or rich. You know why? It will be clear in just a few minutes. It is an antibody of, what should I write? Acute infection or past infection? What should I write? Wait, this again is typed, is printed in green color. It's an antibody of, let me write it. It's an antibody of, very good, acute or recent infection. IgM से याद करो, M से me first, me first. It's a very narcissistic antibody. सबसे पहले यही आएगी. Irrespective of whatever infection is happening in a particular individual, this is the first class of antibody which will be generated. Later on, other antibodies will be produced based on whatever cytokines have been generated as a result of that infection, but सबसे पहले यही आएगी. Okay. Okay. Then it is also referred to as what? It is also referred to as millionaire molecule. You know why it is called as millionaire molecule? It is very, very heavy. It measure, I mean, इसका molecular weight होता है 10^5 daltons. 10^5 daltons. मैं bracket में लिख देती हूँ millionaire molecule, because the green text, I will notify team about it कि avoid using green color. So 10^5 daltons हो गया. Now what is the structure? What is the structure? Look at this. It is a pentameric molecule. This is one antibody molecule, meaning one monomer. IgM has got 5 such monomers. So total 5 are there. Let me draw it like this. Obviously, Y wala part will be outside Y, because it is the one which is going to grab the antigen. Now, these 5 monomers are not hanging in the air. There must be something which is going to bind them together. And what is that something? That is the J chain, or the joining chain. Okay. Now, who will tell me how many antigens bind to one antibody molecule, that is one monomer? Let's say if this is one monomer, ठीक है, इसको क्या बोल रहे हो? One monomer. It binds to how many antigens? Yeah, it's still echoing. I had messaged the team. Fantastic. 2 antigen binding sites. Okay. 2 antigen binding sites. For example, this monomer binds to these 2 antigens. Then similarly, third, fourth, fifth, sixth, seventh, eighth, ninth, 10th. And what is meant by valency of an antibody molecule? Valency of an antibody molecule means the number of antigens. Valency of IgM is therefore 10, but the effective valency drops down to 5. You know why? Because there is so much of traffic encountered amongst the 10 antigens, so all 10 antigens actually are not able to bind. Is this clear, everyone? Yes, steric hydrins. Yes, Dr. Keval, I had tried it. Actually, I had given the workbook to the team. I think they also did not know about this green color. They probably missed it. Apologies for the same. See, if I even if I highlight, no. All right. Such a heavy molecule, what do you think? Where should it be present predominantly? In other words, 80% of such a heavy molecule, that is no wonder it is known as millionaire molecule. It's a very royal, very rich antibody because it can engage 10 identical antigens, but effectively it is able to bind only 5. But 5 also is not a small number. Okay? 80% of IgM, therefore, is, remember, it is intravascular. It is intravascular because it is very difficult for such a heavy molecule to leave the intravascular compartment and enter the extravascular spaces. So again, ye bhi aapko ratta nahi marna hai. This is very, very logical. Okay, very good. Now this is the only antibody class which is made by the fetus, that is before birth. One second, guys, just give me a minute. This is the only antibody class which is made by the fetus before birth. So let's say some pregnant lady is there and she unfortunately gets infected with some microorganism, and that is such a microorganism which can cross the placenta and enter the fetus. In that case, how will the fetus respond? By making IgM antibody. So this is the only antibody class. Yes, Ashwin, I just informed one team member about it. Okay. The original workbook was not having any of the green text. I think the team probably had missed it. And this is the only antibody, as I said, which is manufactured by the fetus before birth, and it is also present on the B cells as B cell receptor. Let me overwrite on this only antibody class made by the fetus before birth. It is also known as what? The antibody is also known as agglutinating antibody. Don't worry, against that the green text I am writing everything again. So when you are given the soft copy anyways, it will have the proper text. But for the purpose of this live session, I'll write again, I'll rewrite again everything against that green text and not overwrite it. Otherwise, in the soft copy notes, you will not be able to follow. Okay. And IgM, remember, it is the most potent activator. It is the most potent activator of what? The classical pathway of complement activation. All of these are MCQs. तो चलो फटाफट से हम रिवाइज कर लेते हैं. We have learned about IgM, the royal rich millionaire molecule. It's a pentamer. Effective valency is 5, ideal valency is 10. But ideal चीजें हमेशा लाइफ में नहीं होती हैं. ठीक है. दैट इज द लाइफ लेसन वी कैन लर्न फ्रॉम IgM. इट इज टीचिंग अस कि कई बार आइडियल चीजें नहीं भी होती, तब भी ठीक है, सर्वाइव करना है. 80% of this heavy molecule is intravascular. Only antibody class made by the fetus. यहाँ पे भी देखो me first का फंडा लगाओ. Me first. This is the antibody class made after any infection. Fetus भी कैसे respond करेगा? By manufacturing IgM. It is also present on what? It is also present on B cells as BCR. That is B cell receptor. That is B cell receptor. Okay, Preeti, noted. Okay, don't worry, you have not missed anything, Preeti, by the way. Okay, whatever I happen to say in Hindi, I already had said in English, but okay, point noted. I will avoid Hindi completely, no issues. Now, it is also known as agglutinating antibody. What is meant by agglutinating antibody? Look at this. This is the pentamer, right? So this pentamer is, grabbing onto five antigens. So we say that these antigens get clumped or agglutinated. So agglutination is an antigen antibody reaction, and in that antigen antibody reaction, which is the most important antibody, it is IgM antibody. Okay, and then IgM, being a royal antibody, is the most potent activator of classical pathway. Okay, hope you guys, hope you guys will remember all of this. Okay. Now, moving on to the next antibody class, IgG. Let me highlight G of IgG. G for IgG, G for going across placenta. So this is the only antibody class which goes across placenta and provides what? It provides, this antibody goes across placenta and it provides natural passive immunity. It provides natural passive immunity to the newborn. Very good. This is the most abundant, this is the most abundant antibody in the serum. As I mentioned at the outset, only remember gumdi IgG What is the half life? Now I will say, a phrase in Hindi which I will translate to English, because that's important to remember the next point. Now when your elders give you blessings, what they usually say? Jeete raho. Jeete raho in English means may you have a long life. Okay, so IgG, G से जीते रहो, which helps us to remember that IgG has got the longest half life, longest T half, that is around 23 days. That is around 23 days. Yes, Saurav, you're absolutely right. Okay. Now, IgG signifies what? IgG signifies past or chronic infection. Again, remember G for gone by, जो चला गया, बीत गया, something which has already, gone by, over, in contrast to IgM, which means recent infection. Now, there are four subclasses. These are the four subclasses of IgG and the order of their serum concentration: IgG1, G2, G3, G4. Remember, G1 is number one in concentration. It wins the race. वो first आ जाता है. IgG1 maximum concentration, followed by IgG2, G3, G4. Please remember. And most potent activator of classical pathway, what did I say? IgM. And IgG is the second most potent activator of the classical pathway of complement. Okay, of the classical pathway of the complement. Is this clear? One second, guys. I have requested team for some other colored PDF, so that just bear with me for for a few minutes until I get another PDF which does not have the green color. Okay, till then we'll continue with this. Okay. Then now four subclasses I have told you: IG, G1, G2, G3, and G4. Amongst all of these four, subclasses, remember IgG4, it activates the alternate pathway. It activates the alternate pathway, not the classical pathway. So guys, please remember, in this initial part of today's session, you will see double content, in the sense that for the sake of the video, I'm rewriting stuff, all these lines together. So when you are receive the soft copy, you will understand that because of this green thing issue, I had to rewrite it, but it's the same thing. Okay, all right. Now, IgG is also known as precipitating antibody. IgG is also known as precipitating antibody. It participates in precipitation reaction. So remember, agglutination में कौन participate करता है? IgM. IgM participates in agglutination. It is the agglutinating antibody. IgG is known as precipitating antibody. And what is the valency? And what is the valency of IgG? It is 2, because it is a monomer. And also remember, IgG is a very good word. It is a very good opsonin. What is an opsonin? Anyone can tell me in the comment section. What is an what is wrong, Ramesh? Can you please mention your doubt? Opsonin is a substance which facilitates phagocytosis. It facilitates phagocytosis. Okay. So IgG, remember, it's a very good opsonin. And amongst all the classes, all the subclasses, IgG1 and IgG3, IgG1 and IgG3, they are said to be better opsonins than IgG2 and IgG4. Okay, noted, Preeti. I'll go a little slow. See, guys, this was not my original workbook. I had submitted it to team. I think they also did not know about the green thing. I have requested them for a fresh copy. They'll just change the color and share with me in a few minutes. Okay, they were also not aware of this. So let's quickly revise IgG antibody. So how did we start about IgG? Ig for going across placenta. So it goes across placenta, providing natural passive immunity to the newborn. It is the most abundant antibody class in the serum. And G say, remember, jeete raho. It has got the longest half life of approximately 23 days. G for gone by. It signifies past or chronic infection. There are four subclasses whose serum concentration is as follows: G1 maximum concentration, followed by G2, followed by G3, followed by G4. Okay. IgG is the second most potent activator of complement. Okay. Then IgG is also known as precipitating antibody because it participates in precipitation reaction. IgG is a monomer, so valency is 2, meaning being a monomer, it can bind to two antigens. Then I said IgG is a very good opsonin, and amongst all the four subclasses, IgG1 and IgG3, they are much better opsonins as compared to IgG2 and IgG4. Okay. So FMGE Jan 23 question: which immunoglobulin has got the highest concentration in the serum? Just a moment, guys. Let me just talk to the team once. Just give me a moment. hope my screen is visible. Team has shared with me other. Okay. Okay. Now, without further ado, let's just continue focus on the session. Now, which immunoglobulin has got the highest concentration in the serum? Asked in FMGE Jan 23, highest concentration kya hai? IgG antibody. Okay. So IgM, IgG we are through with. Next is IgA antibody. Now, A for IgA. What should I write? Which pathway of complement also begins with A? Alternate pathway. Very good. Alternate pathway. And IgA can exist in two forms. IgA can exist in two forms, monomer and a dimer. And two subclasses are there, IgA1 and IgA2. This is the serum concentration. This is the serum concentration. मेरे साथ साथ याद करते जाओ. Just like in IgG, I told you IgG1 comes first. IgG1 maximum concentration followed by IgG2, G3, G4. Here also IgA1 and IgA2, these are the two antibody subclasses of IgA. A1 comes first. This is the, it has got maximum serum concentration followed by IgA2. And it exists in two forms, monomer form like this and dimer form, which is also known as secretory IgA. Secretory IgA. So these are the two monomers, and obviously they are going to have therefore J chain. But in addition to J chain, it has got one more protein chain, which is known as what? Which is known as the secretory piece. The secretory piece. This also is an MCQ key. J chain is present in. J chain is present in which classes of antibodies? IgM and IgA, meaning those antibody classes which are not monomers, polymeric antibodies. Okay. Now look at this. I have put up a picture of secretory IgA. Look at this. So there are two monomers, a secretory component and a J chain. Another MCQ which is asked over here is Now, both the monomers and the J chain. Monomers and the J chain, they are produced by what? They are produced by the plasma cells. But the secretory component, it is produced by what? It is produced by the mucosal epithelial cells. This diagram is self-explanatory. Let me just explain, and you need not write anything. So let's say this is a lumen. By the way, IgA antibody is needed where? IgA antibody is needed in the lumen, like GIT lumen, respiratory lumen, because we are eating microorganisms, we are inhaling them also. So IgA's, IgA dimer is present on the mucosal surfaces so that it can grab the microorganisms and prevent them from binding to our receptors. Now, let's say if this is the GIT lumen, and this is the submucosal region where you have got this plasma cell. Remember, P for plasma, P for kya? Protein. Antibodies are protein. So plasma cells, I personally call them the manufacturing factories of proteins called as antibodies. So look at this. Plasma cell has produced this dimeric IgA. Good evening, Sanjana. Oh, 1 second, guys. I have informed technical team to please resolve the echo thing. Okay, so in the submucosal area, you have got the plasma cell, and as you can see over here, plasma cell has manufactured the dimeric IgA, which is still not complete because it doesn't have the secretory component. So, who produces both the monomers and the J chain? It is the plasma cell. This is an MCQ in itself. Okay. Now, once this dimeric IgA has been produced in this way, it binds to this receptor, poly Ig receptor, poly Ig receptor. So it binds to this receptor and gets internalized into this epithelial cell, mucosal epithelial cell, in a vesicle like this. Then it undergoes what? Enzymatic cleavage. And you see one remnant of this poly Ig receptor. This poly Ig receptor, by the way, look at this. This is this, maroonish, beaded thing, just like, chanting beads are there. So this chanting bead, like, like thing, that is nothing but the poly Ig receptor. So it binds to this dimer, gets internalized, and then some enzymes act on this whole complex. And in the lumen, what is secreted? The dimer, the J chain, and some remaining part of the poly IgG receptor, which we call as what? Which is now referred to as secretory component or the secretory piece. Secretory component or the secretory piece. Is this clear? So if somebody asks you, this by the way is an MCQ, secretory component is produced by, it is produced by what? It is produced by the mucosal epithelial cell. Any doubts in this? Can I continue? Mucosal epithelial cell. Okay. So what are the functions of the secretory component? It protects the secretory IgA molecule against attack by the bacterial proteases in the gut. Please remember, IgA also is a protein. Antibodies are proteins. So in the gut, the bacterial proteases should not degrade or damage IgA molecule. If IgA itself is damaged, how will it, protect us? It should survive in the gut so as to bind or grab the microorganisms. Then it helps in transporting IgA to the lumen. And remember, IgA is the most abundant antibody class in breast milk and colostrum. Remember, A, B, C. A for IgA, B for breast milk, and C for colostrum. Is this clear? Acha. One more thing, guys. Let me just go back to the IgG wala part. Don't worry, I will rewrite all of this and share with the team. So you will not get that green colored PDF. Okay, I will rewrite everything in the beginning few pages and then share with team. Okay, so don't worry about that. Okay, so I said IgG, goes across placenta. There is one class of Ig, one subclass of IgG2 which does not cross placenta. It is IgG2 which does not, it does not cross placenta. Remember, In Hindi, illegal work is known as what? There's a phrase for illegal work, a Hindi phrase for illegal work. क्या बोलते हैं? दो नंबर का काम. So दो नंबर का काम. Remember number two. IgG2 is not good for the baby. It should not cross placenta. Just a memorizing tip. Okay. All right. So we are through with IgM, IgA, IgG. Okay. Now IgD. Okay. Good news is that you don't have to remember much about IgD because it is still, it is still being investigated. Whatever little we know about IgD is that it is present on the B cell surface as B cell receptor, that is BCR. Now, interestingly, the free IgD which is there in the serum, it does not bind the antigen, and therefore, interestingly, IgD is not known as an antibody. It is simply called as an immunoglobulin. Guys, this is a very conceptual thing. What is, even though immunoglobulin and antibody, they are synonymously used, but there is a technical difference. Antibody, if an immunoglobulin is able to bind to an antigen, then that immunoglobulin is known as an antibody. Technically, antibody is a substance which should bind to its antigen, but immunoglobulin D does not bind to its corresponding antigen. So it is not referred to it. Ideally, it is not called as antibody. Please remember this. And then what else is asked on IgD? It has got the second highest carbohydrate content amongst all the classes. Okay. And it activates what? It activates the alternate pathway of complement. It activates the alternate pathway of complement. Is this clear, everyone? Now coming to IgE, even more interesting antibody. Remember, from E, it is associated with extreme of things. It is associated with extreme of things. What do I mean by this? It has got the least serum concentration. It has got the shortest half-life. Maximum serum concentration, what did I say? IgG. And maximum half-life. जीते रहो क्या बताया था? IgG. It is the most heat labile of all antibody classes. See, guys, antibodies are what? They are proteins. No, proteins are heat labile or heat stable. Proteins are heat labile. So, amongst all the five classes of antibodies, the most heat labile class is what? It is IgE. And it is also known as homocytotropic antibody. Okay, homocytotropic antibody. Now, what does this mean? Homo means same, cyto means cell, tropic means to have affinity for. So, this antibody, it binds to what? It binds to the mast cells and basophils, okay, in humans, because these cells have got receptors to attach to this antibody, and this antibody plays an important role in type 1 hypersensitivity reaction. It is also known as reagenic antibody and has got the highest carbohydrate content. So many extreme of things are associated with IgE antibody. Okay. Any doubts so far in the biological functions of antibodies? All of these are important questions. Shall we proceed? All right. Now, next, Staphylococcus aureus. Bacteriology cannot be complete without this particular bacterium. So, this is the gram stain of Staphylococcus aureus from blood agar. Let me zoom it. So, what are these? Gram positive cocci. These cocci are spherical. Okay? These are spherical cocci and they are arranged in grape like clusters. So, GPCs, gram positive cocci in what? In grape like clusters. Okay, grape like clusters. And the word aureus itself means what? It means golden yellow. It means golden yellow. It means golden yellow. So this fellow produces what? A golden yellow pigment. Golden yellow pigment. Now colonies on nutrient agar and blood agar. Colonies on nutrient agar, they are 2 to 3 millimeter sized colonies described as pinhead sized colonies, and these are golden yellow because of the golden yellow pigment. And if there is confluent growth, it is also referred to as oil paint appearance. Very good, sort of oil paint appearance. Okay, so guys, let me know, nutrient agar is a simple medium or an enriched medium? Nutrient agar is an example of a simple, that is basal medium. See, the ideal way to learn culture media is with the organisms themselves. There is no point in ratifying the list of culture media. Okay, so in incorporation of the culture media with the relevant bacteria will give you a good grasp or hold on these media. Okay, now let me ask you a question. Answer very honestly in the comment section. How many of you, without any tantrums, eat whatever your mom makes at home? No tantrums. If mom makes simple dal chawal, you just eat it happily, or you're not very finicky about food. Let me know. Okay, very good. So in that case, Sanjana, Niranjan, Saurav. So all of you are non-fussy, meaning non-fastidious. So those bacteria which grow on simple or basal media like nutrient agar, they are referred to as non-fastidious, because simple or basal media are those which have got basal, basic minimum nutrients. Okay, so these kind of organisms are said to be non-fastidious. So Staph aureus is an example of what? It's an example of a non-fastidious. B for bacterium. B is my own short form for bacterium. Now, if somebody is given dal chawal and you offer that person pizza or some exotic food, biryani and all, they will happily eat that also. Hai na? So, blood agar is an example of what? It's an example of enriched medium. Enriched media are those media which have got incorporation of some additional nutrients, like for example blood, serum, ascitic fluid. So non-fastidious bacteria will grow on enriched media also. All right, but the reverse is not true. There are certain bacteria, certain organisms which have got very, which have the habit of throwing tantrums. They will not grow on simple media, so they are going to be referred to as fastidious bacteria. They need enriched media also, always. So the colonies on blood agar, same as above, plus an additional feature, beta hemolysis, meaning complete hemolysis. Now let's have a look at this picture first. Can you see golden yellow beta hemolytic colonies on sheep blood agar? So beta hemolysis means complete hemolysis. See, I've zoomed it enough, I think. So you are not looking at the colonies are looking at you. Look at the area, adjacent to the colonies. There is complete breakdown of RBC, so that you can see the medium R par. You can see the medium through and through. The complete transparency of the medium is there around the colonies. This is known as beta hemolysis, meaning complete hemolysis. Is this clear? All right. Then we have got salt milk agar also available for Staphylococcus, not just aureus but other species also. So this salt milk agar has got an incorporation of 7.5% NaCl. So remember, Staph aureus can tolerate 7.5% 7.5% NaCl. And what is the selective medium? The selective medium is mannitol salt agar. Now what I have told you, simple media, I've told you enriched media, blood agar, chocolate agar, these are all enriched media. What is selective media? I'm sure all of us have faced at some point or of time or other in our life partiality. Okay, somebody's selective behavior towards us or against us. So it's not a good thing to do to anybody else, and not a good thing to be subjected to also. But when it comes to diagnostic microbiology, please remember there are media called as selective culture media, which show partiality or favoritism or selectivity to the growth of only certain microorganisms, not all microorganisms. All right. So if you're suspecting, let's say, Staph aureus, and you have got the availability of, selective media in your lab, then you can go ahead with mannitol salt agar. Look at this. Mannitol salt agar over here. Now, Staph aureus ferments mannitol, so produces yellow colonies. So Staph aureus ferments mannitol, so it produces yellow colonies, whereas Staph epidermidis does not ferment. It does not ferment mannitol, so it produces the colonies which are of the same color as that of the background medium. And this is Staphylococcus epidermidis. It is an example of cons. Okay, coagulase negative Staphylococcus, and it is notorious for showing biofilm formation and causes infection in the presence of prosthetic devices. So prosthetic infection in the presence of prosthetic devices. All right. And Staph aureus is the MCC, most common cause of so many important infections. For example, pyoderma, okay, folliculitis, okay, superficial infection of the hair follicle. Then furuncle is deep infection of the hair follicle. Carbuncle means what? Multiple furuncles. Then most common cause of breast abscess, more common in lactating females. Most common cause of acute endocarditis in intravenous drug abusers. Most common cause of acute osteomyelitis. Most common cause of acute osteomyelitis. Is this clear? Clear, everyone? Then it also causes toxic shock syndrome and food poisoning, which is because of the exotoxins. Then staphylococcal scalded skin syndrome. These are toxin mediated illnesses. And these exotoxins which cause food poisoning, they are what? They act on the gut, so they are known as enterotoxins. Please do not confuse the word enterotoxin with endotoxin. Endotoxin is found in gram negative bacteria, but Staph aureus kya hai? It is gram positive. It is gram positive. Please remember. Then we come to other important S in bacteriology, Streptococcus pyogenes, which is an example of what? This fellow is an example of group A, beta hemolytic. Group A, beta hemolytic Streptococcus. Rebecca Lansfield was a very eminent microbiologist. She had classified, beta hemolytic streptococci based on the cell wall carbohydrate antigen. So this is known as Lansfield grouping. Lansfield grouping, and this is the gram stain of Streptococcus pyogenes. Dekho, the word strepto means strepto is a reminder of for what? Strips. Strips means chains. So these are the GPCs in chains. GPCs in chains, as you can see in this picture. And Streptococcus pyogenes, by the way, is the most pathogenic species in the genus Streptococcus. Now, staph, I told you it is non-fastidious, whereas in general, all streptococci, they are what? Fussy. All streptococci are fastidious. All streptococci are fastidious. So you cannot expect them to grow on nutrient agar or nutrient broth. Now the colonies on blood agar. Now we are talking about strep pyogenes, the most pathogenic species. Colonies on blood agar are what? They are 1 to 2 millimeter sized, which are described as pinpoint colonies. Pinpoint colonies, these are translucent, meaning there is no pigmentation, unlike staph aureus, and surrounded by beta hemolysis. So beta hemolytic. And what is the selective medium, as rightly told by Saurav? Very good. It is crystal violet blood agar. Crystal violet blood agar. Today at the outset of the class only I had talked about crystal violet, where in gram stain. It is the primary stain. Crystal violet or gentian violet or methyl violet you can use as a primary stain. So साथ साथ मेरे साथ ही revise करो. What is the transport medium? Very good. Pike's medium. Transport medium is Pike's medium. Now what is meant by transport media? Let's say you are an intern in a hospital, and the hospital lab is on strike for some reason. The people have gone on strike, and your SR tells you to carry the sample of the patient to some other lab which is far away. Now you are expected to, and the organism which he's suspecting in the your SR is suspecting in the patient sample, that's a very fragile organism. And fragile meaning the organism will die if it is not transported on time to the lab. So in that case, your SR gives you one transport medium. Ki, okay, I am suspecting streptococcus pyogenes in this patient, so let's use Pike's medium. Okay, so based on the suspicion of the type of organism, and if some delay is anticipated in transporting the sample to the lab, we will use a transport medium. So transport medium, it maintains what? It ensures that the organism stays alive. It does not die. So it maintains what? It maintains the viability of the microbes. It maintains the viability, उनको जिंदा रखता है. ठीक है? Is this clear, everyone? Are these media clear? What is enriched medium? What is nutrient medium? Oh, sorry, what is enriched medium? What is simple, or basal medium? What is transport medium and what is selective medium? Are these terminologies clear? All right. Now, the disease is caused by Streptococcus pyogenes. What is meant by pus? Pus is nothing but dead bacteria plus dead neutrophils. Dead bacteria plus dead neutrophils. Guys, am I audible? Okay, okay. So pyo means pus, genic means to produce. So pyogenic infections or suppurative infections are characterized by production of pus. Okay. So pus means a dead bacterium plus dead neutrophils. Pus is nothing but immune response of the body produced, against bacterial infections. So remember, strep pyogenes is the most common cause of what? It is the most common cause of cellulitis and impetigo. Impetigo. Please make a note of the fact that impetigo can also be caused by Staph aureus. Can also be caused by Staph aureus, but more commonly strep pyogenes is responsible. And also note that bullous impetigo. Bullous impetigo is caused only by Staph aureus, not by strep pyogenes. Not by strep pyogenes. Okay. No, I was increasing the volume only. Okay, Ramesh, I was increasing the volume only. I think, some hiccup had come, but now it's okay. It's an important cause of what? It's an important cause of also, pyoderma. And it is the most common cause of what? It is the most common cause of necrotizing fasciitis. Necrotizing fasciitis, and that is why these bacteria are also known as flesh-eating bacteria. Very good, flesh-eating bacteria. And this is the most common bacterial cause of what? Most common bacterial cause of sore throat. Most common bacterial cause of sore throat. Otherwise, most common cause of sore throat is what? Viruses. Now, the most common bacterial cause of sore throat is strep pyogenes, and this sore throat, if not treated with antibacterials, it can give to complications. What complications? For example, quinsy, that is peritonsillar abscess, and Ludwig's angina. Okay, Ludwig's angina. Okay, these entities you study in detail in ENT. Okay, then some rash associated illnesses. Please remember, rash is set in these conditions. So just a memorizing tip, SET set. Rash is set in set. First is scarlet fever. Scarlet fever is, by the way, uncommon these days. Scarlet fever is characterized by what? It is characterized by sore throat, of course, rash, and what? Strawberry tongue. Strawberry tongue, by the way, is also seen in Kawasaki's disease. Okay, that you read in medicine. Then what is erysipelas? Erysipelas is nothing but a superficial lymphangitis. It is nothing but a superficial lymphangitis. Okay, and it is characterized by a well demarcated rash. It is characterized by a well demarcated rash. Then TSS, that is toxic shock syndrome, characterized by fever, shock, DIC, multi organ system failure, and obviously rash. Okay, so remember, rash is set in set conditions. Rash set matlab introduced. Rash is set in means rash is introduced in or present in SET conditions: scarlet fever, erysipelas, and toxic shock syndrome. Okay, then non-suppurative immunological sequelae, this you read in medicine also and pediatrics also: acute rheumatic fever and acute PSG and acute post streptococcal glomerulonephritis. Acute rheumatic fever is what type of hypersensitivity reaction? What should I write? Let me know in the comment section. And acute PSGN is what kind of hypersensitivity reaction? Kya likhu? Saurav, very good. Type 2 hypersensitivity reaction, and acute PSGN is what? Very good. Type 3. Royal Khan. Okay. Now the treatment of group A streptococcus infections. The drug of choice is penicillin, and in case of allergy, give erythromycin. Give erythromycin. Is this clear, everyone? Okay. Now also remember these important points about Streptococcus pyogenes. It has got M protein, which is anti-phagocytic. Then some strains have got a hyaluronic acid capsule, which also is anti-phagocytic. And it has got another virulence factor. All of these are virulence factors, by the way. Teichoic acid, which is an adhesin, meaning it will help attachment of this bacterium to the host cells. When we say virulence factors, we are referring to the arms and ammunitions of a microorganism with the help of which it harms us, that is human beings. Okay. All right. Yes, Wabhav Konde, absolutely right. Molecular mimicry is the basis behind acute rheumatic fever and all its manifestations. All right. Now, the two important species of Neisseria which you need to know for your exams are Neisseria meningitidis and Neisseria gonorrhoeae. So Neisseria is the only medically significant gram-negative coccus for us. There's one more also, but that's not important for your exams. For as far as Neisseria is concerned, you should remember both of these species. So Neisseria meningitidis, it is also known as meningococcus. Meningococcus, and Neisseria gonorrhoeae, it is also known as gonococcus. It is also known as gonococcus. Okay. Now gram stain, now let me use this color only, reddish color, because they are gram negative. Meningococcus, if I write M like this, okay, let me just highlight this part and let me draw up this curve. What do I get? Plano convex. So these fellows, meningococci, they are what? Plano convex, one side plane and the other side convex, and the plane sides are facing each other. Okay. And meningococcus has got a beautiful capsule, whereas in gonorrhoeae, remember, which is also known as gonococcus, remember, capsule gone away, no capsule. It has got no capsule. Isko kaise yaad karoge? Capsule gone away, there is no capsule. And what is the shape of gonococcus? Let me just highlight G of gonococcus. So the shape is something like this, looking like G. Basically it is kidney or bean shaped like this, and the concavities are facing each other, and there is no capsule. नो कैप्सूल. गोनोरिया में गॉन, मैं लिख देती हूँ, गॉन अवे कैप्सूल. कैप्सूल गॉन अवे. Gone away. Okay. So meningococcus does have a capsule. You can also think it this way. Meningococcus, as the name implies, it causes meningitis. Okay. What are meninges? Meninges are covering around the brain. Let me label meninges as a capsule. Okay. It's not called as capsule, but just for our for the sake of remembering this, Just consider this fact कि meningococcus, which causes meningitis, meninges are a kind of capsule around the brain, so meningococcus has got a capsule. Gonorrhea, the name itself helps you remember that capsule gone away, no capsule. Okay, no capsule. Now, sugar utilization. Sugar utilization. In case of gonococcus, only glucose is utilized. Only glucose is utilized because glucose is a simple sugar. Meningococcus will utilize, of course, glucose because it's a simple sugar in addition to maltose. Remember, M for meningococcus, M for maltose, M for maltose. Okay, all of these are questions. Hello, Ajay Kumar Sharma. If you're reading micro for the first time, this will help, but this is the most high yield session. Okay. I would advise you to read micro in detail also for your exams. This session is meant for those students who already have done micro from some source or the other. This is basically a revision session. Okay. But of course, if you are attending this session, this will help you, but you have to do few other topics also in detail. Okay. So attending this session is definitely going to help. So you are more than welcome to attend the session. Okay. Bagisha, let me just finish this part, and I'll come back to Streptococcus viridans. By the way, it is not called Streptococcus viridans; it is viridans streptococci. It's a group of streptococci. Let me finish this part, and then I'll comment on viridans also. Okay, Bagisha. Okay. Now, virulence factors. Those microorganisms which have got capsule itself is a very, very important virulence factor. The virulence factor, यहाँ पे we will write capsule, and in case of gonococci, it is pili. Pili plural, pilus singular. Pili again are a kind of adhesins which are found in gram-negative bacteria. Then beta lactamase production, less common in case of meningococcus, therefore drug resistance also is less common, whereas beta lactamase production is more commonly encountered in gonococcus, so drug resistance is way more common. Now, Neisseria meningitis or meningococcus, what is the site of carriage? The site of carriage is nasopharynx. And what are the disease causing serotypes? By the way, remember, this is a strict human pathogen. We are the residents of this bacterium. We are the residents of this bacterium. So हमारी human body में where does it stay? In the nasopharynx. And so mode of infection is respiratory droplets. And what are there are actually 13 serotypes, but the ones which cause disease in humans, they are A, B, C, X, Y, and W. Previously, W was known as W135. Okay, W135, but now it is simply called as W. Good for us. Okay. Now, the most common serotype in India, this is, by the way, an old question. The most common serotype, A. But now, this is an old question, but nowadays, the exact data is lacking as far as Indian statistics are concerned. Still, A has been reported sporadically from certain parts of India, and the most common serotype in the West today is B. Remember, Western people are bold. Western people are bold. All right. And overall, if somebody asks you overall what is the most common serotype causing invasive disease, it is A. A is A1 or number one. Okay. So overall, the MCC, the most common serotype, the most common. Let me make a box over here. Overall, the most common serotype is A. इसको ऐसे याद कर लो, A is the A1 or number one serotype disturbing, you know, overall. Okay. Now coming to the diseases. So number one disease obviously is what? Meningitis. And this meningitis is special. You know why? It is associated with a rash. Okay. A petechial or a purpuric rash. Then the second disease is what? The second disease is meningococcemia. If from the CNS the bacteria spill over into the bloodstream, that entity is going to be known as meningococcemia. Meningococcemia is characterized by, remember, PSM. P stands for purpura, which is actually known as purpura fulminans. So very florid rash is there. S stands for what? Shock. The patient goes in shock. And M stands for obviously meningitis, meningococcemia and meningitis. meningitis plus presence of bacteria in the bloodstream, that is meningococcemia. So now the infection is not confined to the CNS, it has spilled over into the bloodstream. Okay. And the third one, the most dangerous manifestation is what? Waterhouse Friderichsen syndrome, and we know in which the patient just dies unfortunately in 3 to 5 hours, hours, not days, hours. So the third entity is Waterhouse Friderichsen syndrome. What is the hallmark of this entity, Waterhouse Friderichsen syndrome? Sort of, you're absolutely right. Right? Okay, you have already answered before I even asked. On autopsy, see, this condition is invariably fatal. So it's an autopsy finding. What is an autopsy finding? Bilateral adrenal hemorrhage. Bilateral adrenal hemorrhage. Okay. Now, diagnosis. Now, if you collect the samples, for example, CSF or bloodstream, okay, or sometimes material from the rash may also be collected. So what will you see? Neisseria meningitis in CSF as seen on gram stain. Look at this. Now, these beautiful big cells with multi-lobe nuclei, these are all neutrophils, and the small tiny dots which you can see, which are there both inside the neutrophils and outside also, they are nothing but meningococci. Okay. So gram negative diplococci. Remember both gonococci and meningococci. यहाँ देखो, they are diplococci. They are हम साथ साथ हैं. They follow the philosophy of we are together. These are diplococci. So gram negative diplococci, both inside and outside the neutrophils, that is polymorphonuclear, that is PMN cells. Polymorphonuclear cells, that is PMNs. Okay. Then culture. Now please remember, let me make a box over here. Neisseria, socho, these are nakchade. Nakchade means they will throw tantrums. So Neisseria is fastidious. Do not expect it to grow on simple or basal media like nutrient agar. Okay. So what enriched media may be used? Blood agar. Okay. Chocolate agar. Chocolate agar, by the way, obviously is better than blood agar, and most commonly chocolate agar is used if we are suspecting, Neisseria meningitis, because it is more nutritious than blood agar. Then selective media. That means a medium which selectively allows the growth of this bacterium only if at all it is there in the specimen of the patient and no other bacterium. So what is the very good? Saurabh, as you rightly said, modified Thayer Martin medium. Modified Thayer Martin medium. The treatment, drug of choice for treatment as well as prophylaxis is what? Ceftriaxone. Ceftriaxone. Very good. Third generation cephalosporins like ceftriaxone. All right. The second important species in genus Neisseria is what? It is Neisseria gonorrhoeae, also known as gonococcus. And what disease it causes? It causes the disease gonorrhea, which is an STI, a sexually transmitted infection. The incubation period is 2 to 7 days. 2 to 7 days. Now, this is a very good question. Capsule or motile or non-motile? Remember, mostly it is non-motile only. If you see any textbook, genus Neisseria is labeled as, it is mentioned as non-motile. But a few years back, AIMS people had asked a question even before INICD exam came into existence. When AIMS used to conduct its own exam, there was a question regarding gonococcus, and the correct option in that question was motility. So mostly it is non-motile, but some strains, please note down, some strains show twitching motility. Some strains show twitching motility. And meningococcus is uniformly non-motile. So please remember. Okay? Now, virulence factor, as I said, pili. All right. The pili will help this bacterium attach to the host cells, that is the urethral epithelial cells and the endocervical cells. And typing. Typing means what in microbiology? Typing means classification. So you are analyzing some structure of the microorganism and you use it as a basis to classify them. So when we say serotyping, what we are trying to imply? Typing means classification. Zero means Zero means surface. So that is the meaning of serotyping. All right. Just like I mentioned about the different serotypes of Neisseria meningitis. So serotype, sero group more or less is used synonymously. Now, in case of gonorrhea, Neisseria gonorrhea, the typing is based on the amount or the distribution of pili. It is known as what? It is known as Kellogg's typing. It is known as Kellogg's typing. Okay. Now, what are the clinical features? In males, this fellow is going to cause urethritis. The patient will have urethral discharge, burning micturition, and males, by the way, are usually symptomatic. Males are usually symptomatic. Okay. Yes, females are going to have endocervicitis if at all they develop symptoms. Endocervicitis, but females, remember, are usually kya? They are usually asymptomatic and they serve as the reservoir or the carriers of infection. Remember, let me make a box and write down. No vaginitis. You know why gonococcus does not cause vaginitis? Because vagina is lined by stratified squamous epithelium, thick stratified squamous epithelium, which is resistant to gonococcal infection. This is the reason. Okay. Then famous MCQ, DGI, disseminated gonococcal infection, meaning the bacteria have entered the bloodstream and they are going here and there. This is seen in people. This is seen in people with deficiency of C5 to C9. That is the late complement proteins. This is the famous MCQ. Yes, absolutely right. Water can perineum also. It is a complication of gonococcal urethritis, multiple discharging sinuses in the perianal region. Absolutely right, Dr. Keval. Very good. So DGI is characterized by what? Tenosynovitis, tenosynovitis, rash, and septic arthritis. And septic arthritis. So remember, this organism, gonococcus, is the most common cause of septic arthritis in sexually active adults. Now, coming to diagnosis, what sample would you collect? In males, urethral discharge is the ideal sample. If frank discharge is not available, you have to milk the urethra and express the discharge. If that also doesn't give any discharge, then the next best sample is the periurethral swab. But the ideal or the best specimen in males is urethral discharge. Okay. Bagisha, meningococcus, yes, it is in nasopharynx, but it invades the bloodstream. That is via hematogenous route, it enters the CNS. Okay. It has got a predilection for the central nervous system. Now, in females, the sample collected is endocervical swab. Endocervical swab. Now look at this gram stain. Now I want you all to compare the gram stain picture of gonococcus and meningococcus. पहले इसको देख लो. Meningococcus, anyways, you have seen. Now, can you notice something special in this picture? Of course, you are seeing beautiful neutrophils and those small dots. Look at this. They're concentrated mostly inside the polymorphs. Few dots are there outside also. So that is why in books the description is given about gonococcus that it is present predominantly intracellularly. It's not that it is not found extracellularly, it is definitely found extracellularly also, like you can see few dots here also in this picture. But remember, it is present predominantly predominantly intracellularly. Intracellularly. Now let's revisit the picture of meningococcus ka gram stain. Look at this. यहाँ पे it is present, almost in equal proportions both inside the neutrophils and outside the neutrophils. Okay. Now this is also important. See, micro is a lot about concepts. If you understand something, you will remember it, retain it for a much better, a much longer period of time. So what is the implication of remembering this? Again, there was an AIMS question same year when they had asked about motility. I'm talking about the same question. ठीक है, तो उसमें there was a option given. Gonococcus is present only intracellularly, which is obviously wrong. If your basics, your concepts are right, you will do these questions correctly. So, if we look at the statement, gonococcus is present only intracellularly. In that case, if this is true, can gonococcus grow in media like chocolate agar or modified thyr martin medium? It will not grow, because these media do not have live cells. So the correct statement is it is present predominantly intracellularly. The statement that says ki it is present only intracellularly, that is a wrong statement, because that would imply that we cannot grow gonococcus on cell-free culture media. Cell-free culture media like blood agar, chocolate agar, basically any medium which does not have live cells. What culture media are there to grow viruses? They are the media which contain cells like HeLa, Hep2. All right. So, the viruses have to be grown on some culture, some medium which has got live cells. All right, because viruses are obligate intracellular. But please remember, gonococcus is not obligate intracellular. It is predominantly intracellular. So this option in that AIMS question was wrong. Okay. Now coming to culture. Now for culture, again, enriched medium, blood agar, chocolate agar, and modified, modified higher Martin medium. This is the selective medium. This is the selective medium. All right. Now, what is the transport medium for both gonococcus and meningococcus? Anyone? Amy's medium. Jay Kamlesh, is my voice low? Amy's medium and Stuart's medium. Is the voice okay, everyone? Jay Kamlesh and everybody else? Now, for treatment, drug of choice for gonococcal infections again is ceftriaxone. So, for both the species of Neisseria, that is meningococcus, Neisseria meningitis, and Neisseria gonorrhoeae, drug is the same, ceftriaxone. All right. Okay. Now, coming to a very important parivar or family of bacteria, Enterobacteriaceae. Entero means what? Gut. Bacteriaceae meaning bacteria. So the bacteria which are there in this family have got something to do with the gut. Either they are gut commensals, or they cause problems in the gut, meaning they will cause diarrhea or dysentery, or via the gut. From the gut they will enter the bloodstream. They will invade the bloodstream, and then they will create problems elsewhere. For example, they can cause meningitis, and while present in the blood they can cause septicemia, like that. So they've got something to do with the gut. Now, the very first organism which we need to know about in Enterobacteriaceae family is Escherichia coli. Okay. So coli itself tells you what? Colon. So what is this fellow? This fellow is a gut commensal. Gut commensal. And Escherichia is the name of the person who discovered this organism. So after his name, this bacterium was named as Escherichia coli. So what is this? What is the interpretation of this gram stain? Please let me know in the comment section. What is the interpretation of this gram stain? First, tell me gram reaction, meaning are you seeing gram positive things or gram negative things? And the second comment you have to make about is the shape. Quick, guys, what is the interpretation of this gram stain? You are definitely not seeing anything spherical. You are seeing, let me just zoom it. You are seeing again, rods or bacilli which are pink in color. So obviously, what should I call them? These are gram negative rods or gram negative bacilli, hai na? Chalo, let us write the name. Gram negative rod. So Escherichia coli is nothing but gram negative rod, gram negative, in other words, bacilli. This entire family Enterobacteriaceae contains what? GNRs, that is gram negative rods, which are catalase positive and oxidase negative. Catalase positive, oxidase negative. They are non-fastidious. Okay? They are non-fastidious, and they are, all the members are facultative anaerobes. They are facultative anaerobes. Very good. So what is the residence of Escherichia coli? Gut. And what is its response to lactose? It ferments it. It ferments it, therefore it produces pink colonies. Pink colonies on McConkey agar. Now, in FMG Jan 23, this question was asked regarding McConkey agar. Now, for the next few minutes, pens down, hands up. Listen to me very, very carefully. I have told you that there is a medium called as selective culture medium, which shows selectivity or partiality or favoritism towards the growth of only certain microorganisms and not all microorganisms. All right. So we have got McConkey agar with us, which shows selectivity or partiality or favoritism towards the growth of only gram negative organisms. So it is selective for what? Gram negative fellows. Okay, gram negative fellows. Now, McConkey agar, in addition to being a selective culture medium, it is also labeled as differential culture medium. Okay. Now, differential stain, say we started the class today, gram stain, Zn stain. What is a differential medium? A differential medium helps us to differentiate between the types of bacteria which are growing on it. So McConkey agar kisko allow kar raha hai grow karne ke liye khud pe? Gram negative. Now, amongst the gram negative which are growing on McConkey agar, some will ferment lactose, some will not ferment lactose. Those which ferment lactose, they will produce pink colonies. They are known as lactose fermenters. And those which do not ferment lactose, they are known as NLF, non-lactose fermenters. And as you can see over here, so on this McConkey agar, two organisms were streaked. Both of them grew on McConkey agar, so obviously both are gram negative. The ones on the left, they fermented lactose, so you can see the pink colonies, and on the right, NLF bacteria are there, and their colonies are described as colorless colonies. Now please remember, colorless colonies does not mean that you can't see the colonies. Obviously you are seeing the colonies, है ना? When we use the adjective colorless for some colonies, we are implying that the color of the colonies is same as the color of the background medium. So if a gram negative organism is not fermenting lactose on McConkey agar, this is how the colonies will appear. You can compare the color of the colonies with the color of the background medium, that straw colored medium. Is this clear, guys? So McConkey agar is helping us to differentiate amongst the lactose fermenting gram negative bacteria and non lactose fermenting gram negative bacteria. Is this clear? Let me know. I'm reading comments, Amit. Amit, till this workbook gets finished, it will go on beyond 10 only. Guys, any doubt in this? McConkey agar? Chalo. Then what else is asked about McConkey agar? Its constituents. Okay. The constituents are PLANT, plant. P stands for what? Peptone. L for lactose. A for agar. N is neutral red, which is the indicator used in this medium, and taurocolate bile salt. Okay. Bile salt, this bile salt inhibits the growth of gram positive bacteria. It inhibits the growth of gram positive bacteria. ठीक है? Gram positive bacteria. All right, this is very, very important. So with bacteria only, we have covered these important concepts about the culture media. Simple media grows non-fastidious. Enriched media will grow both fastidious as well as non-fastidious. है ना? Then transport media we understood. they maintain the viability of the microorganisms. They don't allow microorganisms to grow in them. They just ensure that they are alive. That's the deal with transport media. Then selective media, I told you. And this is an example of a selective medium, which in addition to being selective is a differential medium also, and that is why it is also known as indicator medium. Okay, so it has got this indicator neutral red. All right, and so once lactose is fermented now, acid is going to be produced, so the color of this indicator will change. Okay, all right. Now E coli ferments glucose. It ferments glucose, producing acid plus gas. So it is referred to as aerogenic. Please don't confuse it with aerobic. Aero means air and genic means to produce. Okay. So all Enterobacteriaceae, family members do not produce gas on fermentation of glucose. When fermentation of glucose occurs, acid to definitely is going to be produced, because acid is a definite product of fermentation. Some members of Enterobacteriaceae produce gas also along with acid. Some do not produce gas. If gas is being produced, we label that bacterium as aerogenic. Okay. Yes, Dr. Cable McConkey is a very frequently asked and a very basic thing, okay, in bacteriology. Good. Now, you don't have to know the details of these biochemical reactions, that is INVIC. INVIC, by the way, is an acronym, and I stands for indole test, M for methyl red test, V for VP, that is Vokes-Prosker reaction, and C for citrate test. Okay, just know their results in case of E. coli. So E. coli is indole positive, MR test positive, VP negative, and citrate negative. Okay. Now let me just give you a quick memorizing tip. E. coli, we said it's a commensal, so it is present in our gut. It is present in our body. So imbic reactions, when you recall for E. coli, recall as plus plus. Okay. So 2 plus followed by 2 minus. Okay. Now capsule. Capsule, as far as the presence of capsule is concerned, remember capsule is present in the pathogenic strains. For example, for example, those which cause those which cause neonatal meningitis and neonatal septicemia. Neonatal meningitis or neonatal septicemia. Is this clear? Just a second, guys. Now, as rightly pointed out by Saurav Kumar, the capsule has got what? The capsule has got K antigen. Okay. So in German language, the capsule is written like this. So that K comes from that German word. Okay. Now, next, very, very important aspect about E. coli, diarrheagenic E. coli. Remember, diarrheagenic E. coli are not gut commensals. They are the ones which disturb us. Matlab, as the name implies, they cause diarrhea. And there are 6 important diarrheagenic E. coli which you need to know. Okay. So let's quickly revise them. So firstly, we have got the enteropathogenic E. coli, abbreviated as EPEC. Okay. Now P for kya? P for, remember, pediatric. P for, we will recall, pediatric. Pediatric. So EPEC cause diarrhea in infants. They cause diarrhea in infants. They are an important cause of diarrhea in infants. But if I ask you, what is the most common cause of D for diarrhea in infants? What should be your answer? Most common cause of diarrhea in infants. बच्चे तो रोते रहते हैं. I have already given you the hint. Most common cause of diarrhea in infants. वेरी गुड, रोटा वायरस. बच्चे रोते हैं, रोते से याद करो रोटा वायरस. ठीक है. व्हाट इज द मैकेनिज्म ऑफ डायरिया? लुक एट दिस. इमेजिन माय palm is an enterocyte. ठीक है. And my fingers are what? The microvilli or the brush border. And this pen is an EPEC, enteropathogenic E. coli. So it attaches to these microvilli and it will flatten them, it will efface them. Why God has blessed us with these microvilli? To increase the absorptive surface area. So when these microvilli are flattened out, there will be a drastic reduction in the absorptive surface area and the end result is going to be diarrhea. Okay. So nutrients are not going to be absorbed. In this case there is no physical damage happening, so we do not expect the presence of blood or pus in the feces sample. Okay. So what is the mechanism of diarrhea? Attachment, effacement, lesions. Attachment, effacement, lesions, that is AE lesions. Okay, effacement का मतलब होता है flattening, flattening of these microvilli. All right. Then enterotoxigenic E. coli, that is ETEC. देखो, ETEC में T आता है ना, T for travelers. This is the most common cause of travelers diarrhea. ठीक है. Yes, absolutely right, Saurav Kumar. The gene involved is Lee gene, which stands for what? Locus for what? Locus for enterocyte effacement. Locus for enterocyte effacement. Is this clear? Then it attaches to the enterocytes via what? Via colonization. Via colonization fimbrial antigen, that is CFA. Colonization fimbrial antigen, that is CFA. ठीक है. See, पायलाय का एक और नाम क्या होता है? Fimbriae. So when we say colonization fimbrial antigen, this is an antigen which is present in on fimbriae. Fimbriae is another name for pyli, and as I said before, pyli are the adhesins which are present in gram-negative organisms. ठीक है. And this organism produces, after it attaches to the enterocytes, then it produces these two types of toxins: labile toxin and stable toxin. ठीक है. Labile मतलब labile to heat, stable मतलब stable to heat. Labile toxin does what? Labile toxin has got two components. Two components it has got, A and B. B attaches to the, ये भी MCQ आया है, it attaches to the GM1 ganglioside receptor on the enterocyte. उसके बाद A enter करेगा, A will enter the enterocyte. And A component, it brings about the ADP ribosylation. ADP ribosylation of the GS protein, G stimulatory protein. It just means कि ADP ribose will attach to a molecule of GS. So for example, let's say if this is ADP ribose, it gets attached to GS. This is, by the way, happening inside the cytoplasm of the enterocyte. So together, both of these, they will stimulate what? They will stimulate the enzyme adenylate cyclase, which is there in the host cell in the enterocyte. And when this gets activated, then ATP is going to be converted into cyclic AMP. So what are we getting actually as a result of this toxins activity? We are ending up with an elevated concentration of cyclic AMP inside the enterocyte. And this elevated cyclic AMP in the enterocyte will lead to what? It will lead to, secretion of, sodium and inhibition of absorption of bicarbonate from the gut, and water will passively follow. So it will lead to what? you know uh, Secretory diarrhea. So only biochemical changes are there, so here also no pus or blood will be there in the stool specimen. Stable toxin has got a simple mechanism of action. It directly stimulates what? It stimulates the guanylate cyclase enzyme, and that in turn is going to lead to increased concentration of cyclic GMP. So cyclic AMP, cyclic GMP, you have read in biochemistry, they have got the same action. Is this clear, everyone? So no damage to the enterocytes, no blood or pus cells in the feces. So next we have got entero-invasive E. coli. So as the name implies, it is going to invade the gut cells. This invasion occurs because of what? Because of the virulence marker antigen, abbreviated as VMA. And VMA is plasmid encoded. So VMA is a protein, and the gene of this protein is present on a plasmid. Who will tell me what a plasmid is? What is a plasmid? Yes, exactly. The mechanism of action cable is the mechanism of action of the labile toxin is same as cholera toxin. But why are we so afraid of cholera? Because the cholera toxin is 100 to 1000 times more potent than the labile toxin of ETEC. That is the reason. Who will tell me what a plasmid is? देखो मैं ही बता देती हूँ. P से plasmid, P से क्या? Prokaryotes. What prokaryotes are medically significant for us? The bacteria, the medically significant ones. So plasmid is present in prokaryotes, that is bacteria. It's an extra chromosomal genetic material. Okay. So just like the chromosome has got genes, plasmid also is nothing but DNA only, but it is smaller in size than the bacterial genome. It is not there in all bacteria, by the way. Okay. Now, EIEC is also known as atypical E. coli. Why? There are three reasons. EIEC is NM, non-motile. वैसे E. coli, if somebody asks you, E. coli, in general, it is said to be motile by Peritrichus flagella. The members of Enterobacteriaceae family, they may be motile, they may be non-motile. If they are motile, what is the arrangement of flagella present in the motile ones? Peritrichus. Peri means perimeter. So flagella will arise from all over the surface or from the entire body of the E. coli. Okay. But this fellow, EIC is non-motile, NM is non-motile. Number two, it is NLF, non-lactose fermenter. In the beginning, I told you that E. coli is a lactose fermenter, but this fellow is different. This is not going to ferment lactose. The third atypical feature is that it ferments glucose. It ferments glucose with the production of acid only, no gas. So that is why we are going to label it as an aerogenic. Please don't confuse it with anaerobic. Remember, Enterobacteriaceae family मैंने क्या बताया? They are facultative anaerobes. So an means without, aero means air, and genic means to produce. Next important point. It penetrates or invades the tissue culture cells, for example, HeLa cells. Guys, the name itself tells you entero-invasive E. coli. So ये invasion करेगा. So the invasive property can be tested by treating this organism with HeLa cells, and it is found to invade these cells, HeLa cells, which usually are used for cultivation of what? Viruses. Then we have got a test which is not done nowadays, but it's an important test for exams. Serenis test of invasiveness. So what is done in this test? You take a culture filtrate of EIEC and you inject it into the lower conjunctival sac of a guinea pig or rabbit, and the poor animal goes on to develop keratoconjunctivitis. Okay, so the very occurrence of keratoconjunctivitis is a proof that this organism has got invasive property. Is this clear, everyone? So we are done with enteropathogenic E. coli, enterotoxigenic E. coli, enteroinvasive E. coli. Next is enterohemorrhagic E. coli. Okay. Now EHEC, that is enterohemorrhagic E. coli. Okay. It produces a toxin known as virocytotoxin. It is also known as Shiga-like toxin, also known as SLT. The accepted short form is SLT. Okay, SLT. And what is the MOA, mechanism of action of this toxin? It damages the 60S ribosomes. Matlab eukaryotic ribosomes. That is why it troubles us. And because it produces this toxin, this fellow EHEC is also known as VTEC, matlab virocytotoxin producing E. coli, or STEC means Shiga like toxin producing E. coli. Okay. Then EHEC causes what? Hemorrhagic colitis and hemolytic uremic syndrome. देखो H से ही याद कर लेंगे. The short form has got H. So एक H is hemorrhagic colitis, and the second H, let me draw an arrow like this. The second H is hemolytic uremic syndrome. Okay. And also one more thing. See, H से also remember high five. meaning this fellow is found in industrialized nations. It is not that common in India, but questions are common in Indian question papers. That's a different thing. So you have to know this. Industrialized countries. Now, hemorrhagic colitis, characterized by abdominal cramps, bloody diarrhea. After all, the name itself says hemorrhagic colitis. Remember, there is no fever. Please remember, in case of shigellosis, which also causes bloody diarrhea, uh, fever is an important, symptom. But here there is no fever. Okay. There was a question on, dysentery, that is bloody diarrhea, a few years back, where the stem of the question mentioned that the patient did not have fever, and in the option they mentioned both Shigella also and EHEC. Unfortunately, many students had marked Shigella because it's the most common cause of bacillary dysentery. But the answer was EHEC because the patient did not have fever. That was clearly mentioned. So please be very mindful of these details. Okay. Now, what are the foods implicated most commonly? Beef hamburger. And what is the most common serotype of EHEC? O157H7. Okay. O stands for what? Somatic antigen. It is present in the LPS, lipopolysaccharide, which is also known as what? Endotoxin, which you all know is present in the outer membrane of the gram-negative bacteria. And H is nothing but the flagellar antigen. So today I have told you three antigens. Previously we learned about the K antigen or the capsular antigen. Okay. Now capsule can be present in both gram positive and gram negative organisms. So capsular antigen or K antigen can be there in both gram positive and gram negative. Likewise, H antigen, that is flagellar antigen, can be there in both gram positive and gram negative, because gram positive भी motile हो सकते हैं, gram negative भी हो सकते हैं. O antigen, on the other hand, which is the somatic antigen, it is there only in gram negative, because O antigen or somatic antigen is a part of endotoxin, which is there only in gram negative organisms. Okay. This, by the way, what is H antigen? Such a simple thing was asked in NEET PG 2024 August exam, when there was when the paper was in two shifts. I remember it was asked in paper two. Okay. And as I always say in my classes, the as far as microbiology is concerned, there's quite a lot of overlap amongst FMGE topics, NEET PG and INICT. So the topics are more or less similar only. So this fact, that is why I'm telling you ki this was asked in NEET PG. It may be asked in FMGE as well. Then we have got hemolytic uremic syndrome, which is characterized by the triad of, remember, MTR. M stands for micro angiopathic hemolytic anemia. T stands for thrombocytopenia, and R stands for renal failure. R stands for renal failure. So remember MTR. Okay. Now, a culture medium pe question was asked in FMG December 24. Now, sorbitol, McConkey agar. On this agar, EHEC produces pale colonies because it does not ferment. Because it does not ferment sorbitol. It does not ferment sorbitol. All right. Then enteroaggregative E. coli. It is called so because it aggregates in a very specific fashion on the enterocytes, which is referred to as what? It is referred to as stacked brick appearance, causes diarrhea in developing countries, and produces a toxin known as EAST1, which is referred to as enteroaggregative heat stable toxin 1. ठीक है, EAST1 entero aggregative heat stable toxin 1. Exact mechanism of action of this toxin is not known. Then lastly, we have got the diffusely adherent E. coli, which, as the name implies, it is going to diffusely adhere to the enterocytes, and it causes D for diarrhea in kids 2 to 6 years of age. Any doubts in Diarrheagenic E. coli so far? Let me know. Any doubts? All right. Just a moment, guys. Somebody had asked about viridans, no? Let me write it. Uske baad we will continue with UTI. All right. Please remember there is nothing called as Streptococcus viridans, because when we say Streptococcus viridans, we are implying that viridans is a species of genus Streptococcus, which is absolutely wrong. There is what is known as viridans, streptococci. This is a group of alpha hemolytic streptococci. ठीक है, alpha hemolytic means partial hemolysis of RBCs. In case of partial hemolysis, biliverdin accumulates around the colonies, which imparts a greenish discoloration around the, greenish discoloration to the colonies. Okay, from that biliverdin came the name viridans. So viridans is a group of streptococci which are alpha hemolytic. Okay, so these fellows are oral commensals. All right, they are the MCC, most common cause of subacute bacterial endocarditis, and for something else, which for which all of us, most of us would have visited a dentist, that is dental caries. Dental caries. And what are the examples of the species? Please remember, streptococcus mutans, okay, streptococcus salivarius, streptococcus sanguis, and streptococcus mitis, to name a few. All of these can cause these two bimariyan, SABE and dental caries. And mutants, by the way, is the most common cause. Mutants is the MCC, most common cause of dental caries, and now a vaccine also is available for mutants. But don't worry about the details of this vaccine. It is more relevant for dental exams. ठीक है, तो आपको इसकी डिटेल में नहीं जानना, इतना काफी है. ठीक है, is this clear? Who asked this, by the way? I forgot. Anyways, I have told you this. Now let's continue. Now, if we talk about E. coli and we forget UTI, how can that happen? E. coli and UTI are, by the way, rhyming words, है ना? E. coli, UTI. सुबह 4 बजे भी कोई उठा के पूछे कि what is the most common cause of UTI? What you have to answer? E. coli. Okay. So the most common cause of UTI overall is हमारा E. coli. ठीक है? E. coli. Most common cause of community acquired UTI also is E. coli. Most common cause of hospital acquired, that is nosocomial UTI, also is E. coli. Please remember, these days the word nosocomial is not used. Healthcare associated infection is the phrase which is used. Okay? Then the most common nosocomial infection. Tell me in the comment section, what is the most common nosocomial infection? फटाफट से बताओ. Most common nosocomial infection is what? Most common nosocomial infection is UTI, and therefore the most common nosocomial pathogen. This again was, a previous year question. Most common nosocomial pathogen was asked, and in the option they had mentioned Pseudomonas, Acinetobacter, and many people marked Pseudomonas. Pseudomonas definitely is a nosocomial pathogen, but it is not the most common one. The most common nosocomial pathogen again is E. coli, because the most common nosocomial infection is UTI. Really, Mama. What exactly was asked in Streptococcus sanguis? If you can just tell me the exact question, I will definitely elaborate on it. Is this clear, everyone? Then diseases caused by Salmonella. Again, it belongs to the family Enterobacteriaceae. Enterobacteriaceae. Okay, it is a non-lactose fermenter. Unlike E. coli, it is a strict pathogen. It is never a commensal. It is never a commensal. And remember, it is motile, just like E. coli. And I said before that if at all an Enterobacteriaceae family member is motile, it will be so by virtue of peritrichous flagella. Peritrichous flagella and the NM. The non-motile species, remember, they are Salmonella, pullorum, and Salmonella gallinarum. Gallinarum. Okay, let me just highlight P and G, give you a little memorizing tip. PG मतलब post graduation, तो PG करने के लिए एक जगह बैठ के पढ़ना पड़ता है. तो remember PG, Pullorum gallinarum, non motile. ठीक है, हिलेंगे नहीं. Types of virulent streptococci, these only you have to remember. As I mentioned, look at this. There are many, but you don't have to remember all of them. This is enough. I think they must have mentioned this only, which of the following belongs to virulent streptococci, and they would have mentioned, Streptococcus sanguis. Please check the question, and you can get back to me if any other doubt is there. Yes, Visca, of course. I just commented few minutes back that there's a lot of overlap of topics in microbiology amongst NEET PG, INICET, and FMG exams. Okay, so if you are a NEET PG aspirant, this session will be fruitful for you also. मामा, आई हैव क्लियरली स्टेटेड हेयर. देखो ये इन्फेक्शंस बताए तो सही. ये देखिए, सब acute bacterial endocarditis and dental caries. These are the two infections caused by all of these. Sub acute bacterial endocarditis and dental caries. Chalo, let's continue now. Now, typhoidal salmonellosis, enteric fever. Enteric fever should not mislead you into thinking that it's a gut infection. Enteric fever is a systemic infection. Okay, it's a systemic infection, but the mode of infection is actually feco-oral route. Feco-oral route, that is why it is known as enteric fever. What is the most common cause? Salmonella typhi. Second most common cause, Salmonella Paratyphi A. Third most common cause, Salmonella paratyphi B. Rarest is Paratyphi C. Remember, C is not seen in India. C is not seen in India. And all of these are human pathogens. There is no animal reservoir. These species, these all these fellows, they reside in humans. They don't have any animal reservoir. So what is the source of infection? The source of infection is going to be a human case or a carrier. All right. Then we have got second type of disease entity, that is NTS, non-typhoidal salmonellosis, caused by certain species. It's another species of genus Salmonella. So two important bimariyan you have to remember over here. One is gastroenteritis. The incubation period is 6 to 48 hours. So it is, quite broad, I would say, and it is caused by Salmonella enteritidis and Salmonella typhimurium. Salmonella typhimurium. Just remember GET, get. G for gastroenteritis, E for enteritidis, and T for kya? Typhimurium. And whatever agents cause non-typhoidal salmonellosis, they have got animal reservoirs. So therefore, for example, in case of gastroenteritis, what is the source of infection? What kind of food items? Okay, eggs, poultry and their products, meat. Okay, basically animal products. Okay. Then the second type of NTS is septicemia, notably caused by Salmonella cholera suis. What is meant by suis pertains to what? Actually, pigs. So remember, for this organism, the reservoir are pigs. So NTS में दो important things you have to दो important diseases you have to remember: gastroenteritis and septicemia. A basic difference between typhoidal and non-typhoidal salmonellosis is that Typhoidal salmonellosis means the disease enteric fever, and the causative agents are strict human pathogens, no animal reservoir. Lekin in case of NTS, the causative organisms, be it gastroenteritis or those causing septicemia, these fellows, these organisms have got animal reservoir. Is this clear? Yes, Nagan, PDF, annotated PDF will be shared with you after the class. Okay. Now coming to enteric fever. Enteric fever. Yes, we medicine. There is a little bit of a echo problem. Guys, just let me know if I say if my voice is this much loud. Is it audible to all of you? Has the echo reduced a little if I maintain this, this much voice? Okay, chalo. So this voice is fine, no? Chalo, theek hai. Now coming to enteric fever, which as I said before, enteric se yaad karo kya? It is transmitted, the mode of infection is fecal-oral route. The incubation period is 1 to 2 weeks. The incubation period is 1 to 2 weeks. The mode of infection, fecal-oral route. ठीक है, मेरा खुद का शॉर्ट फॉर्म है FOR, फीको ओरल रूट. एक्चुअली शॉर्ट फॉर्म इसका नहीं है. ठीक है. What are clinical features? Quickly मुझे बताओ. What is the pattern of fever encountered in enteric fever? Come on, in the chat box. What should I write? Muskan, nosocomial infection can be caused by multiple types of organisms. Yes, Pseudomonas is a nosocomial pathogen, but it is not the most common cause. Okay, Acinetobacter, even Staph aureus can also cause nosocomial infection. Okay, thank you, Nilanjan. Fantastic. Motabar, step ladder pyrexia. Okay, step ladder pyrexia or remittent fever, rash, a salmon colored, maculopapular rash may be seen in 30% of the cases by the end of first week. By the end of first week. And non-specific features may be there like sar mein dard, okay, headache, cough, chills, myalgia, arthralgia. So let's say you get a clinical vignette on enteric fever. They will give you all of this. A patient came with headache, cough, and then a rash appeared by the let's say 7 to 8 days of fever like that. Okay. Then GIT features. Remember, abdominal pain, anorexia, nausea, vomiting. Diarrhea is not common. Okay. Nausea, vomiting. Diarrhea is uncommon. Then hepatosplenomegaly will be there. Complications nowadays, thankfully, are very rare. And previously they were common. And what were the complications? GI bleeding and intestinal perforation. Okay. Then rarely neurological features may be seen, like muttering, neuropsychiatric illness, that is muttering delirium, meningitis, cerebellar ataxia were very very rare. Yes, constipation is also common. Okay, let me write it over here. Then relative bradycardia. Can anyone tell me why relative bradycardia is seen over here? In general infections में क्या होता है? Tachycardia होता है. Why bradycardia? It occurs because the B4 bacteria have an inhibitory effect on the SA node. That is the reason for relative bradycardia in this condition. Recent MCQ. Relative bradycardia is also seen in, remember, yellow fever. This is also a question. Okay. Now, some organisms seed the gall bladder and are shed in feces, and some organisms are shed in urine. Is this clear, everyone? आगे बढ़े. Now, diagnosis of enteric fever, very famously asked. Isolation या culture of the organism. Isolation and culture are one and the same thing. What are the samples you can collect? You can collect blood. There is bacteremia in this condition. You can collect feces. I said organisms seed the gallbladder and are shed in the feces, and organisms are also shed in the urine. But feces culture and urine culture is hardly ever done. Blood culture is said to be the gold standard method. Blood culture is said to be the gold standard method for diagnosis. ठीक है ना? Now this was asked in FMGE July 2024. Now why it is blood culture, by the way, is the gold standard method because it has got 100% specificity. That is the reason. ठीक है? A diagnostic test is said to be the gold standard method of diagnosis when it has got highest specificity. और यहाँ तो 100% शत प्रतिशत specificity है of blood culture की, तो it is the gold standard method, but the sensitivity varies. The sensitivity of blood culture first, second, third, fourth of fever respectively is 90%. Distinction याद करो, 75%, 75%, फिर 60%, फिर 25%, 25%. ठीक है? So let's say if a person comes with the features suggestive of enteric fever, the person presents in first week, go ahead with blood culture. Blood culture अगर positive आ गया, start treatment. ठीक है? Then feces culture. Now I come to a very important concept in microbiology about enrichment broth or media. Firstly, not to be confused with enriched medium. Enriched medium, today I have told you examples are blood agar, chocolate agar. Enriched media, as the name implies, they are incorporated with some additional nutrients. They are meant to grow fastidious microorganisms which will not grow on simple or basal media. Enriched and enrichment सुनने में similar एक जैसे लगते हैं, but they are different. पहली बात तो enriched media are solid. I will write it over here. Enriched media are solid, but enrichment media भाई ये broth है, meaning they are liquid media. ठीक है? And enriched media जो होते हैं ना, they are used for processing the feces when we get a fecal sample, a stool sample in the lab. अब मेरी बात please बड़े ध्यान से सुनना. When we get a fecal sample, stool sample, it has got millions of commensal organisms, and even if some pathogen is present, the pathogen is far outnumbered by the plethora, by the millions of commensal organisms which for us are traffic. Are we interested in growing commensals on the culture medium? Of course not. We are interested in detecting the pathogen. Here comes the role of enrichment broth. So we add stool sample to an enrichment broth and we incubate it for a few hours. During that time, what happens? The commensals ka traffic will be suppressed. It will be toned down and the pathogens will multiply. Depending on the type of enrichment broth, the pathogen may multiply. In some others, the pathogen will not multiply, but at least the commensals will be suppressed. So basically, enrichment broth ka kaam kya hota hai? To tone down the commensals. The pathogen may or may not multiply depending on what kind of enrichment broth you are using, but all types of enrichment broth are going to make sure that commensals are suppressed. Is this clear or not? Let me know. Commensals का मतलब है हमारे दोस्त, good friends. Commensals are the microorganisms who are living on our skin and inside our body. We are never alone, guys. कभी अकेला फील होना तो अपने आप को ये बोलो कि भगवान ने ब्लेसिंग दी है कि millions of bacteria are there in our body, inside our body, like for example upper respiratory tract, skin पे commensals होते हैं, gut में bacteria होते हैं. These don't cause disease in us, they are good for us. These are known as commensals, the good bacteria, the good microorganisms, हमारे दोस्त. Clear? Okay, Nirisha is asking me to repeat it. I'll happily repeat. अब मेरी बात ध्यान से सुनना. देखो, let's say I got a stool sample in the lab. Now stool sample, I know it has got millions of commensal bacteria like E. coli and other bacteria are also there. Okay, but I'm suspecting the presence of salmonella in some patient. So what will I do? I will have to first cut down or decrease the number of the commensal bacteria in the stool sample. So what will I do? I will add the stool sample to enrichment broth, that is enrichment medium, and keep it like that for certain number of hours. During this time, what will happen? The commensals ka number will come down. Okay, the commensals are a kind of traffic for me. Why should I grow them on culture media? commensal तो stool sample में वैसे ही होंगे ना, हमें तो पैथोजन को मिस नहीं करना है. ठीक है. So the role of enrichment broth, which are liquid media, their role is to suppress or decrease the number of commensals which are there in the stool sample, but the pathogen को वो touch भी नहीं करेगा. Is this clear? Yes, incubation period का range है 6 to 8 hours. In certain kind of enrichment broth, sometimes up to 12 hours also incubation is done. Rough estimate 6 to 8 hours. No, motabar midstream urine is collected early morning midstream urine, because midstream urine is the cleanest, and it is early morning it is taken because it is concentrated urine sample. Okay. So in urine sample we are referring to the urethral flora. Okay, I think that's what you meant. Motabar, am I right or not? You were referring to commensals wala part, no? Then you are right. Midstream cleanest होगा, और early morning midstream का क्या मतलब होगा? Early morning will be highly concentrated. चलो. So what are the examples of these enrichment broth or media? Selenite, F broth, tetrathionate broth, and gram negative broth, and incubate for 6 to 8 hours. कुछ कुछ जगह पे 4 to 6 hours भी रेंज लिखी है, कुछ पे 6 to 12 भी लिखा है, कुछ कुछ में अप टू 24 hours भी लिख रखा है, बट इट इज सेफ टू यूज, 4 to 6 रेंज भी सही है, 6 to 8 भी सही है. ठीक है. Now, once after this incubation period, next what you have to do, you want colonies, but colonies you will not get on liquid media. So you will take out the stool sample from the enrichment broth, and you will subculture it on selective media. Matlab you will add it onto selective media. Again, selective media meaning if you are suspecting salmonella, you should use those media which show selectivity or partiality or favoritism towards the growth of salmonella and not no other bacteria. Meconic agar. What should I write? LF or NLF? Fantastic sort of NLF colonies. NLF colonies. DCA, that is deoxycholate citrate agar. Pale colonies with black center. The black center is because of production of H2S. Okay? H2S. H2S is produced by S. typhi. But remember, H2S is not produced by. It is not produced by Salmonella Typhi A. Okay. Then XLD, xylose lysine deoxycholate agar PB, red colonies with black center. With black center. Wilson and Blair. B से याद करो, jet black colonies. Jet black colonies with a metallic sheen. With a metallic sheen. ठीक है. एंड अगर यूरिन कल्चर करना पड़े तो हम क्या यूज कर सकते हैं? वी कैन यूज क्लेड मीडियम. We can use clad medium or a combination of blood agar plus McConkey agar. If clad is available, that is better. Okay. If clad is not available, make sure you streak urine sample on both blood agar also and McConkey agar also. So blood culture, remember, is the gold standard method. And what else can be done? Antigen detection in the serum, which is best done in the first week of fever. Best done in the first week of fever because of maximum antigenemia. Then antibody detection in the serum by Vidal test. It is not recommended by WHO because it has got a lot of problems, and the interpersonal variation is there in the interpretation of results. So what is this, by the way? Vidal test is an example of tube agglutination test. You know why it is called as Vidal test? It has been named after a person who called as Vidal. He was from US. So after the name of a US fellow, this test got its name. So Vidal test detects antibodies to the O and H antigens of Salmonella. Okay. Random vlogs, there is no direct or indirect here. It is simply tube agglutination test. Okay? It's a quantitative test. Now, Vidal test के case में false positive reactions can be encountered in case of, vaccination, and false negative in case of early antibiotic therapy, or something known as anamnestic response. ठीक है, anamnestic response. Anamnestic response becomes negative in 7 to 10 days. रैंडम फ्लॉग्स, टेल मी द होल क्वेश्चन. ऐसे नहीं पूछा होगा ट्यूब एग्लूटिनेशन या स्लाइड एग्लूटिनेशन, पूछा होगा कि वेदर इट इज अ ट्यूब एग्लूटिनेशन या स्लाइड एग्लूटिनेशन. ठीक है. एंड जो ट्यूब एग्लूटिनेशन वाला होता है ना, दैट इज क्वांटिटेटिव. इसमें सीरम सैंपल इज सब्जेक्टेड टू सीरियल डाइल्यूशंस. ठीक है. चेक द एग्जैक्ट क्वेश्चन एंड देन यू कैन गेट बैक टू मी. Then carriers in enteric fever. Carrier कौन होते हैं? Carrier is somebody who is harboring the pathogen in the body, but no disease, no signs and symptoms are there. And carrier state develops in 10% of the untreated cases. This is the recent update. Previously, this stats used to mention 3% previously. Okay. Now, convalescent carriers are the ones which shed the bacteria less than 3 months duration. Temporary carriers shed the bacteria from the body for a duration of 3 months to 1 year, and chronic carriers shed the bacteria from the body for more than 1 year. Can you name a chronic carrier? Mary Mallon, a New York chef, notoriously named as Typhoid Mary. Okay. Now, sometimes in fecal carriers, a gallbladder is removed. Okay. Gallbladder is removed. Now, coming to the next important member of Enterobacteriaceae family, Klebsiella pneumoniae. Now, what is the morphology? Because it's Enterobacteriaceae family member, it is a gram negative capsulated rod, and remember it is non-motile. It is non-motile. Yes, it is short and stout as compared to E. coli. E. coli is a little longer and slender. Okay, it is short and stout. छोटा और थोड़ा सा bulky. Imbic reaction. In E. coli, I told you that E. coli are gut commensals. They are present in us. So plus plus minus minus. यहाँ पे लिख लो it is opposite to E. coli. मतलब कि E. coli में क्या था? Plus plus minus minus. तो यहाँ पे हम लिख देंगे minus minus plus plus. It is indole negative, MR negative, VP positive, citrate test positive. Okay. Now look at this reaction to lactose. It is a lactose fermenter. All right. And you can see these, pink beautiful colonies of Klebsiella pneumoniae on McConkey agar. And why these colonies are like this? They are referred to as mucoid colonies. So pink mucoid colonies, the colonies are mucoid because of the hygroscopic, capsule. Hygroscopic means the capsule absorbs a lot of water, and that is why these, giri see mucoid colonies are seen. All right. And this fellow ferments glucose producing acid plus abundant amount of gas. Remember, E. coli also is aerogenic. Which E. coli is anaerogenic? It is EIEC, enteroinvasive E. coli. Urease test is positive. So this is an important example of a urease positive organism. Okay. Now, what are the diseases which this fellow causes? I have written spunk. Let me start from the right hand, from the right to the left. K for Klebsiella, N for what? N for neonatal meningitis and neonatal septicemia. U for UTI, especially in catheterized people. But it doesn't mean that in the absence of catheter, it cannot cause UTI. कर सकता है, तब भी कर सकता है. P for pneumonia. Pneumonia. S for septicemia in geriatric population. Septicemia in geriatric population. Which lobe is affected in case of Klebsiella pneumoniae का pneumonia? It's a lobar pneumonia. Most commonly lower lobe is affected. ठीक है, most commonly lower lobe is affected. ठीक है. Can you think of any important radiological sign in this pneumonia? Anyone, you read that in radiology and medicine also. Bulging fissure sign. पढ़ते हो ना इसके बारे में, bulging fissure sign. Then we have got Klebsiella granulomatis. इसका पुराना नाम क्या है? The old name is Calimato. काली माता से याद कर लो, Calimatobacterium. Calimatobacterium granulomatis. And what it causes? It causes a sexually transmitted infection, STI, namely donovanosis or granuloma inguinale. All right. So this disease is characterized by a पेनलेस जेनाइटल अल्सर. बट पेनलेस जेनाइटल अल्सर तो उसमें भी दिखता है सिफिलिस में. बट व्हाट इज द डिफरेंस? इन सिफिलिस यू गेट पेनलेस inguinal lymphadenopathy also, but यहाँ पे there is no inguinal lymphadenopathy. Lymphadenopathy is very very uncommon. So painless genital ulcer and no LN-pathy, that is lymphadenopathy. Now from this ulcer, if you collect the sample and subject it to GMSA staining, so what will you see? Look at this. This picture is famously asked. So light microscopy of impression smear of granulation tissue from genital lesion of a patient infected with Klebsiella pneumoniae. Note the numerous bacteria in the cytoplasmic vacuole of this mononuclear cell. So look at this. Now listen to me very, very carefully. So guys, this cell, this is a monocyte or this could be a macrophage. Okay. Now inside, so this is the nucleus of the monocyte or the macrophage. You can see inside the cell all of these tiny tiny things. What are these? These are nothing but the bacteria. B for bacteria. And this entire thing, meaning the monocyte or the macrophage filled with the bacteria, this is referred to as what? It is referred to as Donovan body. Please don't confuse it with LD body. LD body is seen where? It is seen in leishmaniasis. Okay? It is seen in leishmaniasis. No, Nirisha, school of fish appearance is not seen in this condition. It gets seen in Haemophilus ducreae infection. All right. And also remember, these bacteria also show safety pin appearance. Safety pin appearance means they will take up the stain more, darkly in at the ends as compared to the middle part of the bacteria. Yes, also known as bipolar appearance. All right, so these are the important Enterobacteriaceae family members you need to know about. चलो आगे बढ़ते हैं. The spore bearing. Okay, sure, Keval. The gram positive spore bearing rods. Spores are present in which bacterial genera? Remember, B and C. Bacillus and Clostridium. Bacillus and Clostridium. ठीक है, तो Bacillus and Clostridium, these are gram positive rods which have got spores, and genus Bacillus, remember, genus Bacillus, these are GPBs, gram positive bacilli, or GPRs, gram positive rods, and these are aerobic or facultative anaerobes, and they are motile by what? They are motile by peritrichous flagella. They are motile by peritrichous flagella. So यहाँ पे you have to learn about two pathogens, anthracis and cereus. So Bacillus anthracis जो है, that is the causative agent of anthrax, but remember it is non-motile. So this is an exceptional species in genus Bacillus. Otherwise genus Bacillus we are saying it is motile by peritrichous flagella. लेकिन Bacillus anthracis is non-motile, and remember this is the biggest medically important bacterium. The biggest pathogenic bacterium is Bacillus anthracis. इसका साइज क्या होता है? 4 से 8 बाय 1 to 1.5 माइक्रॉन्स. ठीक है. Then what is the selective medium? What is the selective medium for Bacillus anthracis? Remember, plet medium. Polymyxin B, plet medium. P से polymyxin B, L से lysozyme, E से EDTA, and T stands for thallus acetate. Thallus acetate. ठीक है. Now what are the cultural appearances? कल्चरल अपीयरेंसेस में आता है हमारा FIM से याद करो, फ्रॉस्टेड ग्लास अपीयरेंस, inverted fir tree appearance, and medusa head appearance of the colonies. Medusa head appearance is seen under the microscope. And what are the microscopic appearances? BBS, boxcar appearance. Boxcar appearance, by the way, is also seen in, it is also present in Clostridium perfringens. Okay. Then bamboo stick appearance. And S stands for string of pearls appearance. This is seen on a penicillin containing medium. Okay. So these are important MCQs which are asked on the microscopy and the cultural appearances. Coming to the virulence factors. So we have got a capsule over here, and that capsule is a protein capsule. It is a polymer of D-glutamic acid. So poly-D-glutamate capsule, poly-D-glutamate capsule, and this capsule is antiphagocytic. All capsules are antiphagocytic, so they are valence factors. matlab ki they will resist phagocytosis. Okay. Then it has got a toxin which is made up of three fractions. Three fractions, 1, 2, and 3. Fraction or factors दोनों एक ही चीज है. Factor 1 is known as the edema factor. Factor 2 is known as protective antigen, and factor 3 is known as lethal factor. It is known as lethal factor. Thank you so much, Amit. खालिद, वायरोलॉजी आएगी, उसका भी नंबर आएगा. बैक्टीरियोलॉजी थोड़ी लंबी है क्योंकि अब क्या करें, हैं ही इतने सारे बैक्टीरिया, लेकिन ये सब इंपॉर्टेंट है भाई एग्जाम के लिए. ठीक है. What is the mode of infection? The mode of infection, remember, are the three eyes. Are the three eyes. देखो एंथ्रैक्स अपने आप में इट्स अ डिजीज ऑफ द हर्बीवोरस एनिमल्स, जैसे कि काउ, शीप, गोट. ऑल राइट. एंड when we acquire this organism from these animals or their products, we can get infected. So we are getting it from animals. So for us, anthrax is what? It's a zoonotic disease. For humans, it is a zoonotic disease. It's a zoonotic disease. Now, how can we get the infection? How can we get the bacterium? First eye is inoculation. So people who carry animal skin on their shoulders or back, the animal skin might be having spores of this organism, and the spores, when they are rubbed against the skin, that is how cutaneous anthrax occurs. So the mode of infection over here is inoculation of the spores. It leads to what? It leads to cutaneous anthrax. It is also known as Hyde Potter. It is not Harry Potter. It is Hyde Potter's disease. Hyde Potter's disease. And this is the lesion over here, anthrax lesion on the skin. This lesion is also known as malignant pustule. It is also known as it is known as malignant pustule, but it is neither a malignancy nor it is filled with pus. It's a misnomer. You can see the central necrotic area surrounded by edema. The central necrotic part is because of the lethal factor, and the surrounding edema is because of the edema factor. Luckily, cutaneous anthrax is, least dangerous anthrax, and it is most common. MC is most common. Then inhalation of the spores can also lead to infection, and it is seen in people who are in close vicinity with sheep. Sheep से वो लोग wool बनाते हैं. So inhalation of the spores will lead to inhalational anthrax, also known as wool sorter's disease. It is also known as pulmonary anthrax or inhalational anthrax. So it is characterized by what? It is characterized by hemorrhagic mediastinitis. And आप इसको रेडियो में इसके बारे में क्या पढ़ते हो? There is going to be, widening of the mediastinum, and wool sorter's disease जो है ना, that is the most dangerous type of anthrax, and it is second most common. Yes, Dr. Keval, anthrax, bacillus anthracis can really cause meningitis also. Yes, definitely it is documented. Okay. Then ingestion of the meat of an animal who died of anthrax who died of anthrax. It is characterized by black necrotic lesions in the intestine, and this kind of anthrax is the rarest anthrax, and second most dangerous. In fact, पता है क्या, oropharyngeal anthrax also has been reported. ठीक है, oropharyngeal anthrax also has been reported. ठीक है. As far as treatment is concerned, drug of choice क्या है? Drug of choice is ciprofloxacin. And remember, A से anthrax, A से category A bioterrorism agent, category A bioterrorism agent. बायोटेररिज्म एजेंट का ना एक निमोनिक मैंने काफी पहले अपने इंस्टा हैंडल पे डाला था. टुनाइट आफ्टर द क्लास आई डू वन थिंग गाइस, on my insta handle, that is mj micro, I will share some stories. ठीक है, late night I will share some stories. You can आराम से see tomorrow. ठीक है, please save those posts. They are going to be really useful for all of you. For example, ये वाला, तो बायोटेररिज्म एजेंट्स देख लेना, आपको याद हो जाएगा. ठीक है. ऑल राइट. तो बायोटेररिज्म एजेंट ही पूछा हुआ है, एफएमजी जैन में पूछ रखा है. नाउ नेक्स्ट इंपॉर्टेंट पैथोजन हमारा क्या है इसमें, बैसिलस में? बैसिलस सीरियस. यस डॉक्टर केवल, रैक्सीबैक्यूमैब इज अ मोनोक्लोनल एंटीबॉडी. ठीक है, नॉट यूज दैट कॉमनली, बट यस इट इज मेंशन्ड in the books. Serious comes from the word cereals. Actually, this fellow Bacillus cereus was found to cause food poisoning in conjunction with cereals. तो उसका नाम ऐसे पड़ गया. It has to be seriously studied. So it causes food poisoning or gastroenteritis, which is of two types: emetic type and diarrheal type. In emetic type, what is the main clinical feature? Vomiting. In diarrheal type, it is diarrhea. What is the incubation period in emetic type? 1 to 6 hours. And what is the median incubation period? 1 hour. And the incubation period in case of diarrheal type is 8 to 16 hours. Yes. Relax. Chinese fried rice. Okay? Everybody is obsessed with Chinese fried rice. What are the foods implicated? Chinese fried rice. Nilanjan, that is what we are doing. All of these are PYTs of FMGE only. Chinese fried rice and foods which are reheated. For example, buffets. Okay? That is the other food. Who asked this? Uh, Khalil. Okay. Then what are the serotypes implicated? 1, 3, and 5. 1, 3, and 5. नाउ डायरियल टाइप में कौन से फूड्स implicated है? अ वैरायटी ऑफ फूड्स, जैसे कि सीरियल्स तो हैं, मीट, वेजिटेबल्स, एंड व्हाट आर द सीरोटाइप्स? 2, 6, 8, 9, 10, 13। 2, 6, 8, 9, 10, 13। ठीक है। व्हाट इज द सेलेक्टिव मीडियम फॉर बैसिलस सीरियस? MYP अगार, मैनेटॉल, एग योग, polymyxin B agar. Okay, very good. And this, diarrhea is self-limiting. Vomiting also is self-limiting. Is this clear? चलो आगे बढ़ते हैं. द नेक्स्ट इंपॉर्टेंट ग्राम पॉजिटिव स्पोर बेरिंग रॉड इज व्हाट? क्लॉस्ट्रीडियम. सो क्लॉस्ट्रीडियम, क्लॉस्ट से सोचो, क्लॉस्ट से मतलब क्लोज्ड, कुछ बंद है. सो सोचो these these bacteria, they are anaerobes, unlike the other spore bearing genus bacillus, which is aerobes and facultative anaerobes. ठीक है, ये एक फंडामेंटल डिफरेंस है बिटवीन द टू स्पोर बेरर्स, बैसिलस एंड क्लॉस्ट्रीडियम में. बैसिलस आर इधर aerobic or facultative anaerobes, but clostridium are anaerobes. ठीक है. ग्राम स्टेन पे GPBs, that is gram positive bacilli, or GPRs. All right. Now spores over here are what? The spores are bulging spores. And make a note of the fact that in case of bacillus, the bacillus organisms, they have got non-bulging spores. Non-bulging spores. If I ask you, tell me an important, a very eminent figure in boxing, क्या mind में आता है? The legend of all times, क्या mind में आता है? Oxygen needs anaerobic मैंने बता दी. Motile by Peritricus flagella. Yes, exactly, मोहम्मद अली. ठीक है. तो बेसिकली मैंने आपसे क्यों पूछा, मैंने यहाँ क्या लिखा? बल्जिंग. अब मेरी बात बड़े ध्यान से सुनो, लिटिल मेमोराइजिंग टिप फॉर ऑल ऑफ यू. ठीक है. सो बॉक्सिंग के दौरान व्हेन यू क्लोज द फिस्ट, ठीक है, क्लोजिंग द फिस्ट विल लीड टू व्हाट? बल्जिंग ऑफ द बाइसेप्स. तो क्लोजिंग द फिस्ट से सोचोगे आप लोग क्लॉस्ट्रीडियम, उससे क्या आएगा? बल्जिंग स्पोर्स. बल्जिंग स्पोर्स का ये मतलब है, लेट्स से इफ दिस इज अ स्पीशीज ऑफ क्लॉस्ट्रीडियम, ठीक है, द स्पोर्स विल बी वाइडर देन द बैसिलरी बॉडी. ठीक है, वाइडर देन द बैसिलरी बॉडी. So it's a gram positive fellow, so I'm using blue color, but remember the spores are not stained by gram stain. So bulging spores means the width of the spore is wider than the bacillary body. ये मतलब है. I hope everyone gets this. So motile by Peritricus flagella except Clostridium perfringens. Okay, Clostridium perfringens and Clostridium tetani type 6. And what are the capsulated species? Clostridium perfringens and Clostridium butyricum. Clostridium butyricum. Now, perfringens is also known as Welchii. Perfringens नाम यूज होता है यूएस में, एंड वेल्चाई कहाँ होता है? यूके में. एंड यूके यूएस कितने फ्रेंड्स हैं आपस में, आप सबको पता ही है. ठीक है, तो वो नाम पे भी लड़ते हैं. तो परफ्रिंजेंस यूएस में ज्यादा कॉमनली यूज करते हैं, वेल्चाई किसमें? यूके में. ओके. व्हाट आर द स्पेशल फीचर्स? टू स्पेशल फीचर्स. यस, एब्सोल्युटली. स्टॉर्मी फर्मेंटेशन. Stormy fermentation on litmus milk medium, on litmus milk medium, and target hemolysis, target or double zoned hemolysis, double zoned hemolysis on blood agar. Mama, by the way, what do you exactly mean by membrane splitting? Now, Clostridium की जितने भी species है ना, all of them are notorious. They are badnaam for producing toxins, and these toxins themselves cause disease. So, Clostridium perfringens produces a variety of toxins, major toxins and minor toxins. You don't have to remember all of them. So, you just have to remember that based on the pattern of toxin production, there are 5 types of Clostridium perfringens: A, B, C, D, and E. And a very important toxin is this one, alpha toxin, also known as lecithinase, that is also known as phospholipase. This is the most important valence factor. It is produced by the all 5 types of C perfringens. And what is its important action? It decreases myocardial contractility, and it increases aggregation of neutrophils and platelets in the blood vessels. And that is why, commonly जो clinical minutes आते हैं उसमें से कुछ mention होगा कि gram stain showed only gram positive bacilli, absence of neutrophils. Why? Because neutrophils तो busy हो गए in forming aggregates in the blood vessels. And this toxin can be detected by Nagler's reaction. It can be detected by Nagler's reaction. ठीक है. Now, diseases caused by Clostridium perfringens are as follows. Gas gangrene, also known as Clostridial myonecrosis. Thankfully, it is very, very rare now. ठीक है. तो इसमें क्या होता है, अगर जब ये कॉमन था भी सही, imagine there's a very horrific road traffic accident happening where the blood vessels are severely damaged, and it's a very dirty wound. Dirty in the sense that the wound is soiled, and soil में क्या present है, the spores of this bacterium. So when the blood vessels are damaged, and the spores are there in the wound, what will happen? The spores are now getting the spores are now present in an anaerobic environment, and this fellow Clostridium perfringens is anaerobic. The spores will germinate. Germination of spores means the spores will transform into Clostridium perfringens, and then it will produce toxins, and then it will cause the disease, gas gangrene. So a very important valence factor over here is the alpha toxin. Thankfully, this is a very, very rare entity. And what is the mainstay of treatment of gas gangrene? It is not, antitoxin has a questionable role. Antibiotics still are started, but what is the mainstay? Surgical debridement. Surgical debridement is the mainstay. Okay. Other treatment measures, you can start antibiotics so that bacteria are killed and no more toxin production is there. Antitoxin, as I said, has got a questionable value. Its exact role is not clear, so I'm writing question mark. And hyperbaric oxygen, who was facilitating germination of spores? It was the presence of anaerobic environment. तो अगर आप उसको ही खत्म कर दोगे तो spores germinate ही नहीं होंगी, है ना? Then the second important disease apart from gas gangrene is gastroenteritis. The most common cause is type A of Clostridium perfringens. Incubation period is 8 to 16 hours. See, the questions on food poisoning are very, very, easy. And उसमें अगर आपको incubation period याद है और foods याद है, symptoms पता है, तो you will crack these questions. And it occurs due to what? It occurs due to an enterotoxin which creates holes in the cell membrane of the gut cells. The foods associated are meat and its products. Then we have got what? Pigmel or Enteritis necroticans. It is caused by type C strains of Clostridium perfringens. ठीक है. अभी recently ना एक event हुआ था. There's a movie by एस राजामौली सर, तुम लोगों ने देखा हो वाराणसी, अगर टीवी देखते होगे तो. ठीक है. तो उसका बड़ा ग्रैंड इवेंट हुआ था, एंड द लीडिंग लेडी ऑफ दैट मूवी वाज देयर ऑन द इवेंट, पीसी, प्रियंका चोपड़ा. तो वैसे याद कर सकते हो आप, पीसी से पर्सनल कंप्यूटर भी होता है, वो भी याद कर सकते हो. तो पीसी, पी से पिगबेल, कॉज्ड कॉमनली बाय टाइप सी स्ट्रेन्स. इट इज कैरेक्टराइज्ड बाय नेक्रोटिक lesions in the jejunum. Necrotic lesions in the jejunum. Now, the next important species is Clostridium tetani. It causes the disease what? Tetanus ठीक है, look at this. This is a gram stain picture showing the characteristic drumstick appearance of Clostridium tetani. ठीक है, so a spherical terminal spore. Spherical terminal spore. Okay, drumstick appearance, by the way, is also seen in, it is also present in or seen in Clostridium tetanomorphum. Clostridium tetanomorphum. Tetanomorphum is a non-pathogenic species which has been named so because morphologically it mimics what? Morphologically it mimics Clostridium tetani. ठीक है, now mechanism of action of toxins पे questions आते हैं. So this is a comparative evaluation of the two important toxins. One is produced by Clostridium tetani, that is tetanus toxin, which acts on the central nervous system, and the second important toxin is produced by another species of Clostridium, that is Clostridium botulinum, which acts on the peripheral nervous system. ठीक है, तो tetanus toxin acts on the central nervous system. It inhibits the release of the inhibitory neurotransmitters GABA and glycine, so it will lead to a descending spastic paralysis. Botulinum toxin, it acts on the peripheral nervous system. It will inhibit the release of the excitatory neurotransmitter, that is stimulatory neurotransmitter acetylcholine at the peripheral nerve ending, so it will lead to a flaccid paralysis. ठीक है, one of the earliest symptoms क्या है भाई? One of the earliest symptoms is trismus or lockjaw. ठीक है, trismus or lockjaw. And other features, there can be arching of the back, opisthotonus. ठीक है, कैरेक्टरिस्टिक ग्रिन ऑन द फेस, मतलब कि इमेजिन करो कि किसी ने ना कोई बहुत गंदा जोक मारा और सामने वाले को जबरदस्ती हंसना पड़ रहा है, तो मुंह बना के हंस रहा है, तो समथिंग लाइक दैट, राइजस सार्डोनिकस. ठीक है, then respiratory failure if the respiratory muscles are involved. What is the mainstay of treatment? The mainstay of treatment is, who is the villain over here? The toxin. Toxin को hero से मिलवा दो. Who is the hero? Anti-toxin. ठीक है, specifically monovalent anti-serum. What is meant by monovalent? There is only one type of tetanus toxin known. So monovalent anti-serum means one type of antibody is there in this anti-serum, which is going to target, which is going to, deal with the villain, that is the tetanus toxin, which is there in the patient. And what are the other measures? You can start antibiotics, okay, wound debridement, and supportive treatment like respiratory support if needed. Then we have got Clostridium botulinum, which causes the disease botulism. Disease. It causes the disease botulism. Now it produces toxins A, B, C1, C2, D, E, F, G. कितने हो गए? 1, 2, 3, 4, 5, 6, 7, 8. So 8 types of toxins are there. Now C2 is a cytotoxin, and the rest are neurotoxins. Remaining 7 are neurotoxins which are responsible for botulism. Okay? And amongst these neurotoxins also, most commonly A is implicated, followed by B, followed by E. हिंदी में अबे अबे बोलते हैं ना कई बार, अबे बहुत ही कैंडिड लैंग्वेज में अपने दोस्तों को, ठीक है, नॉट सबको ऑब्वियसली, इट इज डिसरिस्पेक्टफुल, बट हम जो क्लोज फ्रेंड्स हैं जिनसे हम फ्रैंक होते हैं उसमें कई बार ना आपस में बैंटर में अबे अबे बोलते हैं, तो अबे याद कर लो, एंड रेली एफ, सो दिस इज द सीक्वेंस. सो सोचो इफ दीस, obviously we don't know कि भाई कौन सा toxin is implicated, and to, patient needs urgent treatment. So that is why for treatment, please remember, polyvalent antiserum is given. A polyvalent antiserum means it will have antibodies against toxin A, antibodies against toxin B, toxin E and F. तो 4 तरह की antibodies कम से कम रहेंगी, so that whatever toxin is responsible, that is neutralized by its, specific antibody. Is this clear, everyone? ओके, नाउ व्हाट इज द मैकेनिज्म ऑफ एक्शन ऑफ एंट्रोटॉक्सिन्स? ये हमने ऊपर डिस्कस कर लिया ऊपर वाली स्लाइड में. ठीक है, तो अब ये मुझे बताओ फटाफट से. प्रिवेंट्स रिलीज ऑफ एसिटाइलकोलिन, इनहिबिट्स एसिटाइलकोलिन एस्टरेज, इनहिबिट्स रीअपटेक ऑफ एसिटाइलकोलिन, और ब्लॉक्स पोस्ट सिनेप्टिक रिसेप्टर्स. ध्यान से क्वेश्चन पढ़ो. सौरभ, बी वेरी केयरफुल इन रीडिंग द क्वेश्चन. दोबारा से पढ़ो और फिर मुझे बताओ. प्रिवेंट्स रिलीज ऑफ एसिटाइलकोलिन, दोबारा देखो, ये देखो क्या लिखा है यहाँ पे, botulinum toxin acts on PNS, inhibits release of stimulatory, that is excitatory neurotransmitter जिसका नाम होता है acetylcholine at the peripheral nerve ending. अब ऑप्शन देखो भाई, ध्यान से पढ़ना है क्वेश्चन, हमें गलती नहीं करनी है. प्रिवेंट्स रिलीज ऑफ एसिटाइलकोलिन, भाई ये हो गया ना. सेकंड ऑप्शन क्या है, इनहिबिट्स एसिटाइलकोलिन एस्टरेज, तो गलत है ना भाई, है ना. So please be very careful in reading the question. Now, types of botulism, botulism, it is due to what? It is due to canned food. It is due to canned food. अब मेरी बात बड़े ध्यान से सुनो. Canning is what? Canning is a process of preservation of food. Clostridium botulinum जो organism है ना, इसकी spores are there in the atmosphere. भगवान की तरह omni present है, हर जगह present है. So when we are doing canning, मतलब when we are preserving the food, we take a can, we add the food, and there's a process of removing the air. And at that point of time, let's say if Clostridium botulinum की spores अंदर आ गई, तो spores को हम देख तो सकते नहीं है, और आपने ढक्कन बंद कर दिया. Now, spores have got a very good environment, anaerobic environment, because it's an anaerobic organism. The spores will germinate. They will, produce what? They will produce the bacteria, and bacteria will produce the toxin. Okay, so it is due to intake of the canned food, and specifically which foods are usually implicated? Alkaline vegetables, for example, green beans, peppers, etc. Okay, the incubation period, as I said, it is due to what? It is due to intake of what? Intake of the toxin. So we expect the incubation period to be short, but surprisingly it is longer. It is more than 24 hours. You know why? Because the toxin is a little lazy to act. It's a little lazy to act. तो यहाँ पे क्या फीचर्स रहेंगे? The patient will have diplopia, dysphagia, constipation, respiratory failure can also occur. Respiratory failure can also occur. Okay. Is this clear, everyone? Now, second kind of by the way, food botulism is the most common type. It is the most common type of botulism. Most common type of botulism. Now, the second kind of botulism is infant botulism. It is because of ingestion of the spores by what? By infants. Okay. Infants होते हैं when they are let's say crawling outside, जहाँ पे पार्क में बच्चे को घुमाने के लिए लेके गए हैं और वो ऐसे जमीन पे खेल रहा है. So injection of spores कैसे? Injection of spores either because of pica or contaminated honey. So when this entity was discovered, then the it was asked कि बच्चे को क्या दिया था, what was given to the child. And in some communities it is a practice of giving the infants honey. So the initial cases were reported from such communities only, and when that honey was examined, it was found to contain the spores. Okay, so after ingestion kya hota hai? The spores germinate in the alkaline environment of the gut, and then bacteria are produced and they will lead to floppy baby syndrome. Basically the child will stop sucking milk, the child will keep falling over, because it's a spastic paralysis, है ना, या peds में भी पढ़ते हो. So please remember, a fundamental difference between food botulism and infant botulism is that food botulism is because of intake of the toxin, and infant botulism is because of intake of the spores. ठीक है. Then rarest kind of botulism क्या है भाई? Wound botulism. ठीक है. Wound botulism, it is associated with deep space infections, and remember, the gastrointestinal features are absent over here. They are absent in wound botulism. And what is the mainstay of treatment of any kind of botulism? It is polyvalent antiserum. And other supportive measures you can do, for example, antibiotics if wound botulism is the cause, then wound debridement. If the patient is going for respiratory failure, then supportive treatment, supporting respiration. Okay. Now, Clostridium difficile. By the way, its new name is Clostridioides. Clostridioides difficile. पता है इसको difficile क्यों बोलते हैं? Because it is tough or difficult to grow or isolate, and it is the most common cause of nosocomial diarrhea. D for diarrhea. All right. Then diseases can be categorized as mild and severe. Mild में there will be watery diarrhea. Remember, diarrhea is watery, not bloody. ठीक है. Severe case में क्या होगा? Pseudomembranous pseudomembranous colitis. ठीक है. तो ये होता क्यों है? देखो, आपने देखा होगा कई बार कोई एंटीबायोटिक्स अगर किसी को प्रेस्क्राइब की जाती हैं तो साथ में क्या बोला जाता है कि प्रोबायोटिक्स ले लो। वो रीजन क्या होता है? एंटीबायोटिक्स जो दी जा रही है पेशेंट को ना, दोज एंटीबायोटिक्स दे किल द कमेंसल्स। इसीलिए उनको रिप्लेस करने के लिए उनको मतलब उनकी भरपाई करने के लिए प्रोबायोटिक्स दी जाती है। So imagine if probiotics are not given and somebody is taking antibiotics so that the gut flora is destroyed and there is just no bharpai in the form of probiotics. So what will happen? The gut commensals are going to be destroyed, but if C. difficile is present, which by the way is present as a gut commensal in certain people, that will flourish. So when the gut commensals ka number tones down, C. difficile ka number will flourish. All right. So some antibiotics, when they're taken orally, they will destroy the gut commensals, but they will spare what? They will spare C. difficile, and this C. difficile, whether it will cause disease or not, it will also depend on a very important factor. Remember, मैंने थोड़ी देर पहले आपको क्या बोला? Whatever species of Clostridium are there, they produce disease by toxins. In C. perfringens, we learned about alpha toxin, है ना? Then tetanus or botulism वाले toxins मैंने बता दिए. अब C. difficile में भी toxigenic strain. Toxigenic मतलब toxin producing या toxin generating. Toxigenic strains cause disease. Cause disease. जो ऊपर मैंने disease लिखवाया है, pseudomembranous colitis and diarrhea. And what are the most commonly responsible antibiotics? These are the antibiotics which are used most commonly these days, blatantly these days. That is third generation cephalosporins. Third generation cephalosporins. Clindamycin, remember, was the first antibiotic with which this disease entity, that is a C. difficile infection, was associated or linked with. And what are the other antibiotics? Macrolides, quinolones. And you would be surprised sometimes, even metronidazole and vancomycin, which by the way, both of them are given for treatment of this condition, of this infection, sometimes they also can cause C. difficile infection. No, not Bhuvna, not at all. I'm just saying a generic statement, not with all antibiotics. Wow, thank you so much, KP Chaudhary. So delighted to know this. Congratulations. Of course, you will learn about all these details regarding the antimicrobials in pharmacology. I just made a very general statement to give you an idea of the pathogenesis. Of course not. Of course, आपको हर एंटीबायोटिक के साथ थोड़ी देनी होती है वो, ठीक है? एजिथ्रोमाइसिन कोई खा रहा है तो कोई क्यों देगा? समझे? What are the earliest lesions on histopathological examination? Yes, Saurav, absolutely right. Summit lesions. Summit lesions. तो summit से याद करो, we are just summing up what our Clostridium वाला bacterium. ठीक है? अब diagnosis के लिए हम क्या लेंगे? We will take obviously feces. लेकिन कल्चर इज नॉट कन्फर्मेटरी बिकॉज कल्चर पे इवन नॉन टॉक्सिजेनिक स्ट्रेन्स भी ग्रो कर जाएंगे. ठीक है. ठीक है. Yes, yes, Nilanjan, we will take a break in just a bit. Just let me please finish this part. ठीक है. ठीक है. Cefoxitin, cycloserine. fructose agar, that is CCFA, is one selective medium, and second one is cefoxitin cycloserine egg yolk agar, that is CCYA. Both of these are what? Both of these are selective media. But please remember, culture पे growth आती भी है, तो we will not know whether the toxigenic type of C. difficile has grown or the non-toxigenic type. ठीक है? So ideally, we should prove that toxin is there in the sample, for which we have got ELISA to our rescue. So ELISA to detect what? To detect the toxin. Toxins are proteins only. ELISA can detect it. PCR to detect the genes of the toxin. Genes of the toxin. Then what is the gold standard? Gold standard हम करते ही नहीं है, because it's very very time consuming, technically difficult, and high-fi facilities are needed. And what, by the way, is the gold standard? Gold standard is cell or tissue culture cytotoxicity assay. Okay. Now what about treatment? देखो, अब guidelines have been changed and you would read this in detail in pharmacology also. कुछ साल पहले तक बच्चों में क्या बोलते थे? The drug of choice is metronidazole, and adults में भी ये बोलते थे. But now please note, the drug of choice, vancomycin, metronidazole, and there's one more drug, fidaxomicin. Okay, fidaxomicin, and vancomycin is given orally in this condition. Otherwise, vancomycin is given parenterally. This is the only oral indication of vancomycin. This is also famously asked. And experimental therapy क्या है? Fecal bacteriotherapy. मतलब कि you are basically doing fecal transplantation. Doing fecal transplantation, you are transplanting the gut flora from a normal healthy person into the patient by, let's say enema. And your funda is what? The normal flora which you are introducing to the patient, that normal flora की presence में the C. difficile will tone down, उनका number कम हो जाएगा. So this is based on the principle of bacterial interference. बैक्टीरियल इंटरफेरेंस. तो देखो, कई बार ना इंसान एक दूसरे के काम में टांग अड़ाते हैं, बट और यहाँ पे बैक्टीरिया भी एक दूसरे को इंटरफेयर कर रहे हैं, बट दिस इज अ गुड इंटरफेरेंस. इज दिस क्लियर? इज दिस क्लियर एवरीवन? Okay guys, it is 9:47. Is it okay if we take a like 15 minutes break? Okay, so we will start at 10:05 p.m. Okay, see you after the break. what? It causes the bimari diphtheria, which is called as go to in Hindi. First, look at the gram stain appearance. You can see gram positive rods. And the word corini, it means what? Club shaped. Club shaped basically means that the ends of these rods are swollen like this, club shaped. Okay? So Corynebacterium diphtheriae. And the arrow points to the typical club shape. Look at this. Typical club shaped bacterium. And the arrowhead points to typical V or L shaped. So what is this arrangement known as? What is this arrangement known as? Chinese letter or cuneiform arrangement. Okay. So this is known as Chinese letter or cuneiform arrangement of these organisms. And famous question, the colonies produced on the selective medium are black colonies of club shaped gram positive rods on selective medium, that is potassium telluride blood agar. Potassium telluride blood agar. Okay. Now you must be knowing that Corynebacterium diphtheriae is notorious for producing for having certain granules in its cytoplasm, and these are produced very nicely on a medium called as Loeffler's serum slope. Now Loeffler's serum slope is an example of an enriched medium, and on this medium growth appears in 6 to 8 hours, and this medium is good for induction of granules. Okay, now what are these granules basically? These granules are nothing but they are they are reservoirs of energy and phosphate. In other words, they are polymetaphosphates. Basically, they are phosphate groups only, and they are more concentrated at the ends of the bacilli. So they are also known as polar bodies. They are also known as volutin granules. They are also known as Babes Ernst granules. Okay, so polymetaphosphates, because they are reservoirs of energy and phosphate, they are known as polar bodies or Walden granules or Babes Ernst granules. These are the different names, and these granules are produced when the nutrients are deficient, are decreased or deficient. मतलब not much nutrients are available. You know, इसको भी आपको रट्टा नहीं मारना है, इसमें भी एक concept है important. ठीक है, so what is that concept? That concept goes like this: when nutrients are in abundance, what will happen? The bacteria will tend to divide by what? Binary fission. And during binary fission, what will happen? The chromosome also will replicate. So when chromosome multiplies, when it replicates, phosphate groups will be consumed. All right. But when the nutrients are not enough, not adequate, the bacteria in that case will not divide. So these phosphate groups will be accumulated in the cytoplasm of these bacteria, and these phosphate groups are nothing but the granules of Corynebacterium diphtheriae. So as I said, they are polymetaphosphates. Now, does it make sense why these granules should be produced when nutrients are decreased? Okay, I hope this is clear, everyone. Let me know. And they are stained most commonly by what? Albert stain. Okay, Albert stain. Look at the next page first. Okay, Albert stain. We look at this. These long sticks, the green sticks which you are seeing, they are nothing but the bacteria. And the at the ends of these bacteria, you can see dark bluish colors. The some spherical bodies. They are nothing but the granules. So as I said, the granules are concentrated more at the ends or the poles of the bacilli. So just little memorizing tip regarding Albert stain. BGGB. BGGB लिखो, बीच में डंडा मारो, और अब हम क्या लिखेंगे? पहले बी से बैसिलाई, जी से ग्रीन, फिर दूसरे जी से ग्रैन्यूल्स, एंड फिर बी से ब्लू. तो bacilli will appear green and the granules will appear blue. Okay, now what are the other stains which may be used for the granules? Pond's stain. Pond's stain or Neisser stain. Please remember, Neisser stain is not for Neisseria. Not for Neisseria. Wahi Neisseria which we read, before the break, Neisseria meningitis, Neisseria gonorrhoeae. Okay? Acha, an important virulence factor, VF, virulence factor of Corynebacterium diphtheriae is what? Exotoxin. Exotoxin. So toxin producing strains, only they will cause diphtheria. Not all Corynebacterium diphtheria are going to have the toxin. This exotoxin, it's a protein, and protein, you all know, comes from what? Mrna comes from gene. So there are certain phages which have got the genes, uh, which have got the genes for this toxin. So when those phages का क्या मतलब होता है? The viruses which attack bacteria. Okay, so when those phages containing the genes for this exotoxin, when they attack or infect Corynebacterium diphtheriae, the Corynebacterium diphtheriae strains, they get the gene and they express it. When the gene is expressed, the toxin will be produced. Okay, so the toxin producing strains, they have got genes for the toxin, and they have got it from beta phage or coronaphage. Beta beta phage or coronaphage, and this process is known as lysogeny. What is lysogeny or lysogenic conversion? One and the same thing. When a phage carrying the gene of this toxin, it attacks Corynebacterium diphtheriae, then Corynebacterium diphtheriae express that gene, matlab they will produce the diphtheria toxin and they can cause diphtheria. If they are not infected by this phage, they will not get the gene to produce the toxin. No toxin means no diphtheria. So diphtheria, remember, is a toxin mediated disease. Is this clear, everyone? So it is lysogeny or lysogenic conversion. Is this clear? So what are the reservoirs of this organism? Cases and carriers. Transmission, respiratory, droplets. Please remember, the organism is not invasive. Okay? It colonizes the epithelium of the throat. The most common type of diphtheria क्या हुआ? The most common type of diphtheria is faucial diphtheria. What are fauces? They are the regions where the palatine tonsils are located. And FMGE July 2024 का क्या question है? The diphtheria toxin, what is its mechanism of action? This again is a two subunit toxin made up of two subunits, A and B. It inhibits protein synthesis by adding what? By adding ADP ribose to what? Elongation factor 2. Elongation factor 2, when it gets combined with ADP ribose, it cannot focus on its job. What is the job of EF2 in our cells? Elongation of protein chain, matlab protein synthesis. So it will be distracted from its job of protein elongation when ADP ribose आके इससे चिपक जाता है. ठीक है, तो remember A and B components है ना. A is the active subunit, B is the binding subunit. B subunit will bind to the cellular receptor, after which A will enter inside, and it is the A subunit which will bring about the addition of ADP ribose to EF2, matlab ADP ribosylation of EF2. Okay? Now, in the throat, a dirty gray pseudomembrane will be formed. It may extend into larynx or trachea, causing obstruction, and the toxin circulates in the blood, but the bacteria, remember, they are localized. And what is the effect of toxin on the systemic circulation? It causes myocardial and nerve damage. Remember, there is no bacteremia. So we say diphtheria is not a systemic infection, it's a systemic disease. Okay? And how to make a diagnosis? Demonstration of toxin by actually two types of tests: in vivo tests, which are not done nowadays, and in vitro tests. In vitro mein, there are two tests: LX gel precipitation test, which shows arrowhead precipitates in case of a toxigenic strain, and cell culture cytotoxicity test. Cell culture cytotoxicity test. Okay? Then what is the mainstay of treatment? Who is the villain here? The toxin. So mainstay of treatment is antitoxin. Remember, You can start toxoid, but that is for future protection. Toxoid is nothing but the vaccine. Okay, so toxoid is not going to do anything for the current situation, which is because of the toxin. So आपको क्या करना चाहिए, आपको एंटी टॉक्सिन देना चाहिए पेशेंट को. You may start toxoid for future protection of the patient, मतलब कि toxoid से और antibodies बन जाएंगी. ठीक है. As far as antibiotics are concerned, remember PERC, गोला बनाओ, डंडा बनाओ. Now P stands for what? Penicillin. E से erythromycin. Now both of these are drug of choice. And R stands for what? R stands for rifampicin. C stands for what? Clindamycin. These are alternates. So R and C, these are alternates. Okay. So that's about Corynebacterium diphtheria. I hope everything is clear. Clear, everyone? Shall we proceed? Rickettsia. Now, Rickettsia are atypical bacteria. You know why? Because they are obligate intracellular, just like viruses are. So, obviously, therefore, they won't grow. They are not grown in cell-free culture media. Cell-free culture media, matlab ki, culture media which do not have, living cells like blood agar, chocolate agar, etc., etc. Okay. Now, Rickettsia infect what? They infect vascular endothelial cells. ठीक है, so they infect the blood vessels, vascular endothelial cells, leading to rash. But rash is absent in which rickettsial fever? Can anyone tell me rash is absent in which rickettsial fever? Nirisha, Bartonella has been separated from rickettsia because it is not obligate intracellular. ठीक है. The rash is absent in Q fever, and most of the species are transmitted by what? Some arthropod vector. Some arthropod vector, ठीक है, such as louse, mite, tick. And animal reservoirs are present for all the species of rickettsia except rickettsia provazeki. ठीक है. And therefore all rickettsial infections are zoonotic diseases except which one? Except epidemic typhus, which is caused by Rickettsia provazeki. Now, guys, please remember, animal reservoir simply means that, there are certain animals who are the residents, that is the reservoir of certain microorganisms. And we get infected with these microorganisms when we deal with these animals in some way or the other. So, for us, such infections are said to be zoonotic diseases. But there is one species of genus, one species in under Rickettsia, that is Rickettsia provazeki, which has got no animal reservoir. We, that is humans, are its reservoirs. So it is not a zoonotic disease. Please remember this. Okay. Now, different disease groups are there under rickettsia. For instance, सबसे पहले we will talk about the typhus fever group. So it's of two types, epidemic typhus and endemic typhus. Now, epidemic typhus is caused by, देखो, epidemic, remember, it's a very dangerous event. सुनने में काफी negative लगता है, epidemic. So epidemic is usually provoked by something. So from that, remember, the causative organism rickettsia provazeki. ठीक है. And what is the vector? The vector is human body louse. Now, when I say human body louse, automatically it gives you the information that this louse is going to infest only human beings and not any animal. Okay, so animal reservoirs are none. Okay, we, that is humans, are the reservoir of this bacterium. And in the olden days, this entity, epidemic typhus, was very common amongst jail inmates. Why? Because in the olden days, there was not much focus on hygiene, and when hygiene is not maintained, what do we expect? Louse infestation. All right. And the vector over here also is human body louse. So, because it was common amongst jail inmates in olden days, it is also known as jail fever. Now, let's say somebody has recovered from epidemic typhus, few years later that person gets symptoms again. That is known as reactivation typhus, also known as recrudescent typhus, also known as Brill-Zinsser's disease. Okay, it occurs because of what? It occurs because of decrease in the number of antibodies. If I ask you what is the vector of reactivation typhus, answer is none. There is no vector. There is no vector because it is a reactivation infection. ठीक है? So the body के bacteria, they themselves are from the latent state, they are now नींद से जाग रहे हैं. Why? Because of decrease in, because of jeopardized immunity, because of decrease in antibodies. So there is no vector over here. Okay. Now coming to endemic typhus, which by the way is less severe than epidemic typhus. It is caused by Rickettsia typhi, also known as Rickettsia museri. Okay, these are two different names of the same bacterium. Just like we sometimes have got multiple names. एक घर का नाम होता है, nickname, एक official नाम होता है. कई बार हमारे friends हमें affectionately किसी नाम से बुलाते हैं. तो similarly, there are certain microorganisms which have got more than one name, synonymous names. What is the vector over here? The vector is rat flea, and understandably, therefore, the animal reservoirs are what? Rodents. Animal reservoir are rodents. Okay. Now, in general, the clinical features of rickettsial disease are as follows: extreme degree of exhaustion, thakan, that is prostration, myalgia, rash. Why? Because blood vessels are involved. Fever and HSM, matlab hepatosplenomegaly. Then the second kind of disease is spotted fever group. अब देखो, इसमें I have made one table. इसमें 1, 2, 3, 4, 5. The disease which is number one, Rocky Mountain spotted fever. I have written it as number one disease. So remember, it is number one in severity also. ठीक है? तो क्या है ये? RMSF stands for Rocky Mountain spotted fever. So it is extremely or most severe rickettsial disease. The vector is hard tick and the causative organism is Rickettsia rickettsii. Okay. So why have I written extremely oblique most severe? The reason is, please remember, this is an example of a rickettsial disease where the rash first appears on the extremities and then on the trunk. This is unlike other rickettsial diseases. Unlike other rickettsial diseases, where the rash first comes on the trunk and then involves the extremities. Okay, so just this is just a little memorizing tip for all of you. तो extremely severe से extremities याद रखो, extremities पे rash सबसे पहले आएगा. ठीक है. Then next three diseases, remember them as IAS. IAS, Indian Administrative Services. I say Indian tick typhus, A say Australian tick typhus, S say Siberian tick typhus. तो यहाँ पे भी the vector is the same, hard tick, hard tick, hard tick, and IAS respectively are caused by, remember, CAS, Rickettsia connori, Rickettsia australis, Rickettsia siberica. Then rickettsial pox. Twist in the tail. This is not caused by tick, and this itself is an MCQ. Key, which kind of a spotted fever is not tick transmitted? Answer is rickettsial pox. It is transmitted by mite. It is caused by Rickettsia acari. ठीक है. अब देखो, माइल्डेस्ट रिकेट्सियल डिजीज ओवरऑल कौन सा है? ये देखो, माइट के नाम में क्या आ रहा है? एम आई, और माइल्डेस्ट की स्पेलिंग में भी एम आई आ रहा है. तो माइट वाला लीस्ट सीवियर है. कौन सा फिर लीस्ट सीवियर है? रिकेट्सियल पॉक्स. तो मैंने इस टेबल में रिकेट्सियल पॉक्स डेलिबरेटली आई हैव केप्ट इट एट द लोएस्ट एट द बॉटम. सो रिमेंबर, इट इज द लोएस्ट इन सीवियरिटी. I hope this is clear. This, by the way, I'm talking about the overall most severe and overall least severe या mildest rickettsial disease. Is this clear, everyone? वेरी स्कोरिंग क्वेश्चन आर आस्क्ड. ठीक है, तो ये टॉपिक आपको अच्छे से पता होना चाहिए. नाउ नेक्स्ट वी हैव गॉट स्क्रब टाइफस. नाउ स्क्रब टाइफस you should suspect in a patient if a patient has got fever, rash and ellenpathy. that is lymphadenopathy. It is caused by what? It is caused by Orientia susu gamushi. It is not tasu gamushi. It is pronounced as susu gamushi. T is silent. Then I don't know why T is used. Anyways, what is the vector? Very, very important. The vector are the larvae of trombiquilid mite known as chiggers. The name of the larvae are chiggers, and therefore the disease is also known as chiggerosis. The disease is also known as chiggerosis. ये वाला पिक्चर बेस्ड क्वेश्चन आया हुआ है एफएमजी जुलाई 2024 में ट्रॉम्बिकुलेट माइट लार्वा. ठीक है, दिस इज हाउ इट लुक्स लाइक. Okay, this is how it looks like. And look at this. Sometimes, S-char also is present in case of scrub typhus, and this again has been asked as a image based question. Now, this disease is something which is associated with what is known as zoonotic tetrad. Okay, so V से हो गया हमारा vector, which is nothing but the trombiculid mite का larva. Okay, then R से rodents, which are the reservoir. एस से क्या है? स्क्रबी और बुशी वेजिटेशन. स्क्रबी और बुशी वेजिटेशन का क्या मतलब होता है? वेजिटेशन मतलब लो लाइन ट्रीज. these low lying trees में ना rodents आसानी से रह सकते हैं. तो these scrubby और bushy vegetations are a very conducive home or environment for the thrival of rodents. And W stands for wet or rainy season because the larvae are produced in rainy season. ठीक है, तो just remember the tetrad as VRSW. देखो tetrad किसका पूछ रहे हैं? zoonotic tetrad of scrub typhus है ना. तो one of the components तो scrubby vegetation हो ही जाएगा, फिर vector लगा लो, फिर rodents and बारिश. Okay. Then Q fever, इस पे काफी क्वेश्चंस आते हैं. One thing I already mentioned about Q fever at the outset of rickettsial infection, that there is no rash in this case, and also there is no arthropod vector. So some students ask, is it a zoonotic disease? Of course, yes, it's a zoonotic disease. You don't have to always have a vector for zoonotic disease. Is anthrax not a zoonotic disease without any vector? It is. Okay, so anthrax में हमने 3 I's पढ़े थे ना, inhalation, inoculation, ठीक है, इंजेशन. तो यहाँ पे भी सेम 3 आईज आर इन्वॉल्व: इनहेलेशन, इनोकुलेशन, इंजेशन. एनिमल रिजर्वॉयर्स आर प्रेजेंट. इट इज वेरी मच, क्यू फीवर इज वेरी मच अ जूनोटिक डिजीज, एंड इट इज कॉज्ड बाय व्हाट? It is caused by Coxiella burnetii. It is caused by Coxiella burnetii. Now, clinical features: it may cause fever only, or it may cause pneumonia. It may even cause hepatitis, or it may cause infective endocarditis. Now, even though it has been clubbed under rickettsia, but recently it has been possible to grow this organism in cell-free medium. This is a recent update. Please remember this. Okay. Now, coming to the diagnosis of rickettsial infections, serology, that is antibody detection in the serum of the patient. You can go for detection of specific antibodies by ELISA and indirect immunofluorescence. Specific antibody का क्या मतलब होता है? An antibody which has been produced against an antigen which is there only in rickettsia and not in any other microorganisms. And non-specific antibody, this has been asked in FMGE Jan 2023. Wheel Felix reaction. Wheel Felix reaction, it's an example of a heterophile. It's an example of a heterophile agglutination reaction, and it is a tube agglutination test. It is a tube agglutination test, but it is not done commonly. Okay. Now, as far as treatment of rickettsial infections is concerned, remember tetracyclines or chloramphenicol can be given. Now we come to the cousin brother or sister of rickettsia, that is chlamydia, because just like rickettsia, these also are atypical bacteria. That is, they are also obligate intracellular. They are also obligate intracellular. And another feature which is similar to viruses. See, this feature of being obligate intracellular is also common to viruses. And another feature which is similar to viruses is that chlamydia can pass through. They can pass through bacterial filters. They can pass through bacterial filters. And these are very special bacteria because in them the cell wall is made up of not peptidoglycan, but it is made up of proteins. It is made up of protein. And these are the only bacteria in the entire, medical bacteriology which exist in two morphological forms, the elementary body and the reticulate body. So each elementary body, it is the extracellular form, it is present in the environment. And infection kaise hoga? Environment se hi hoga. So it is the infective form. It is the infective form. So in the environment, elementary body is present and we acquire it from the environment and get infected. So this is the infective form. But once elementary body enters the human body, it will undergo, imagine plastic surgery, just like Ekta Kapoor ke serials mein nahi hota, some actor leaves the show or some problem happens, then she ropes in another actor. Okay. And what is the plot then? In which direction does the plot of the serial goes? कि रोड ट्रैफिक एक्सीडेंट हो गया, फिर प्लास्टिक सर्जरी करानी पड़ेगी. तो सोचो, when elementary body enters the human body, it undergoes plastic surgery. मतलब it undergoes what? A morphological transformation, and it becomes क्या? Reticulate body. So reticulate body is the intracellular form. It is the replicative form. अब भाई जब ये सेल में आ गई तो काम धाम करेगी ना, ऐसे खाली थोड़ी बैठी रहेगी. So it undergoes replication. And remember, it is bigger in size than the elementary body. All right. Then now we have got three important species of genus Chlamydia. Pehla hai Chlamydia trachomatis, which is divided into 15 serovars or serotypes, which are clubbed into three groups. Okay, go to the next page. These are the three groups: A, B, BAC, D2K, L1, L2, L3. A, B, BAC. What do these cause? Trachoma. कुछ टाइम पहले तक दिस वाज द मोस्ट कॉमन इन्फेक्शियस कॉज ऑफ ब्लाइंडनेस इन इंडिया, बट अभी एज पर WHO अपडेट, इंडिया हैज बीन डिक्लेयर्ड फ्री ऑफ ट्रैकोमा, which is a very good news. I had posted this update on my Instagram handle also. So guys, as I said, tonight, if I've got some energy left, I will share some posts in my Insta story. Otherwise, tomorrow morning for sure. So please follow me on Instagram. I've got this microbiology handle. You will get MCQs and a lot of updates and doodles and cartoons in microbiology, which will be helpful to you. Okay? So D2K, D2K pe aate hain. It causes what? It causes non-gonococcal, an STI, non-gonococcal urethritis. Non-gonococcal urethritis, that is NGU. NGU का ये क्या है? Most common cause. D2K is the MCC, most common cause of NGU. Again here also, males and females, just like I told you in gonorrhea, in males urethritis, and in females endocervicitis. Endocervicitis. Okay, endocervicitis. Now overall NGU जो है ना, देखो note, NGU is less severe than GU. देखो G से GU मतलब gonococcal urethritis, G for grave, ज़्यादा severe. Okay, and also thankfully NGU is more common. It is more common than gonococcal urethritis. अच्छी बात है क्योंकि NGU ऐसे भी less severe है. ठीक है. Coming to L1, L2 and L3. Now these cause an STI, namely lymphogranuloma venereum, that is LGV, characterized by what? A painless, mostly it is painless, sometimes can be painful, a painless genital ulcer plus painful inguinal LNpathy or lymphadenopathy. And amongst these three serovars, L2 is the most common cause, MCC. So most commonly LGV is caused by L2. Again देखो दो नंबर का काम L2. Okay. Now, a diagnosis of Chlamydia trachomatis infections can be done by microscopy, which by the way is very less sensitive. Agar karni ho to what will we see in the microscope? We will see inclusion bodies. Inclusion bodies. These most commonly have got the RB, reticulate body. ठीक है, but may have elementary body too. If suppose you have taken the sample at a time where elementary body has just entered the host cell, in that case you will see elementary body under the microscope. But elementary body almost immediately transforms into reticulate body. So most commonly in these inclusion bodies, what is seen, it is the reticulate body. Now culture previously was considered the gold standard, but not anymore. Culture, because these are obligate intracellular organisms, the culture can be done in HeLa cells, Hep2 cells, and an important cell line, namely McCoy cell line. But culture is no longer the gold standard. Nucleic acid amplification test, that is PCR, now is considered to be the gold standard test. Now it is considered to be the gold standard test. Okay, so that's about Chlamydia trachomatis. And as I said, there are two more species of Chlamydia. There has been a taxonomical change in the in these organisms, and the genus name has been now changed to Chlamydophila. ठीक है, तो कुछ डिफरेंस नहीं है, पहले क्लैमाइडिया बोलते थे, क्लैमाइडोफिला बोल रहे हैं. क्लैमाइडोफिला सेटासी causes psittacosis, which refers to what? Infection in the cetacean family of birds. Cetacean family of birds, इसमें क्या आता है? Parrots. So later on, it was found that apart from parrots, this bacterium can infect other birds also. And what is the general name for infection of birds? Ornithosis. So ornithosis means infection of birds in general. So basically, it causes infection of birds. Okay. Now in humans, it's a zoonotic infection. So whoever is in vicinity with the infected bird, that human can be infected. And how humans exactly are infected, what is the mode of infection? Infection is acquired by inhalation of the inhalation of the dried up excreta of the birds, which become what? Which become aerosolized. Is this clear? Then how a diagnosis can be made? Remember, complement fixation test and microimmunofluorescence. ठीक है, microimmunofluorescence, and CFT is nothing but complement fixation test. ये याद रखो. Then the third one is Chlamydophila pneumoniae. ठीक है, नाम से क्या पता चल रहा है? Pneumoniae. अब देखो, let me highlight MON and A in pneumoniae, which means 1. So number of serotypes क्या हो गया? 1. It causes the disease pneumonia. It is the toughest of all the three species to culture. So amongst Trachomatis, Cetaceae, and Pneumonia, this one is the toughest one to culture. And it is also thought to play a role in, remember, AAS, atherosclerosis, asthma, and sarcoidosis. No, Abdullah, the bacteria have been found to, survive for a certain period of time. And when these excreta become aerosolized, they may be having viable bacteria, which then can be inhaled. Okay? Yes, fantastic, Nirisha. The name of that the strain is T-War strain. Okay? And what is the treatment of chlamydial infections? Remember, M, छोटा C and बड़ा Q. तो M से क्या है? Macrolides. C से nothing. Q से quinolones. ठीक है? All right. Now, next very important organism, Leptospira interrogans, which you all know causes what? Leptospirosis, which is the most common zoonotic disease on Dharti Mata, that is Mother Earth. पहले इसकी ना शक्ल देख लेते हैं कैसा दिखता है ये. This is a scanning electron microscope of Leptospira interrogans. You can see it has got hooks in opposite direction. Hooks in opposite direction. This is very important, opposite direction, because there's another species, Leptospira biflexa, which has got hooks in the same direction. So pathogenic जो species है interrogans, that is why it is called as interrogans, as if it is interrogating somebody. So hooks in opposite direction, remember, looking like a question mark. Now, leptospirosis is the most common zoonotic disease, most common zoonotic disease on earth, that is Dharti Mata. It is also known as what? It is also known as Sewer workers disease, also known as rat fever. Please don't confuse it with rat bite fever. ये rat bite से नहीं होता है. Rat bite fever दूसरे बैक्टीरिया कॉज करते हैं. ठीक है. It is also known as paddy fever. It is also known as 100-day fever because sometimes fever may be very prolonged. What by the way is known as 100-day cough? 100-day cough क्या होता है भाई? 100-day cough is nothing but pertussis. याद रखो इस चीज को. ठीक है. Then the severe form of leptospirosis is known as Weil's disease. The severe form is known as Weil's disease, also known as iterohemorrhagic fever. Iterohemorrhagic fever. And have you guys, any of you has seen that web series, very well made web series Kala Pani on Netflix? Anyone? देखा है किसी ने काला पानी? अच्छा, जब टाइम हो ज़रूर देखना, ठीक है? एग्ज़ाम्स के बाद देखना दैट दैट हैज़ गॉट अ कनेक्शन विद दिस बीमारी, ठीक है? बहुत इंटरेस्टिंग है, तो बहुत अच्छा है वो। अनफॉर्च्युनेटली उतना ना मतलब फ़ेमस नहीं हुआ। आई डोंट नो व्हाई, मे बी वाज़ नॉट प्रमोटेड वेल। तो एक चीज़ याद रखना ज़रूरी नहीं कि जो चीज़ फ़ेमस ना हो उतनी, वो अच्छी भी ना हो, ठीक है? फेम डज नॉट ऑलवेज गारंटी यू क्वालिटी, ठीक है? दैट वाज अ वेरी वेल मेड सीरीज। एग्जाम के बाद देखना। नाउ आर पे आते हैं। आप ब्रेक से पहले मैंने आपको सर एस एस राजा मौली वाली मूवी के बारे में बताया था ना कि ट्रेलर आया। पी सी प्रियंका चोपड़ा मैंने बताया था। पिग, पी से पिगबेल और सी से टाइप सी स्ट्रेन्स ऑफ क्लॉस्ट्रिडियम परफेंजेंस। तो हम वापस में उन्हीं की एक क्लासिक मूवी पे आते हैं, आर। तो मेडिकोस को पिक्चर वाली आर के अलावा एक और आर पता होना चाहिए। And what is that RRR in the context of leptospirosis? Rodents, which are an important animal reservoir. Even cats and dogs also are reservoirs, but rodents are most important ones. ठीक है. Then R से दूसरे R से क्या है? Rainfall. आप लोगों ने देखा होगा ना कि leptospirosis के cases जो होते हैं, they are way more common in floods or very heavy rainfall. And T से R से rice, that is paddy fields. Paddy fields. ठीक है. तो प्लीज रिमेंबर आर. All right. So, what is the link amongst these three R's? देखो, during heavy rains, torrential rains or floods, what happens? The चूहे जिनके बिल है ना, rodents के जो बिल हैं, उनमें पानी भर जाएगा. The rodents will be forced to come out. And rodents, I said, are an important reservoir. मतलब they are residents of these bacteria. So these bacteria, Leptospira interrogans, they are shed in the urine of the rodents asymptomatically. Rodents को कोई disease नहीं होगा. So when rodents are forced to come out of their burrows, they will urinate here and there, and they will contaminate surface water. That is the link between rodents and rainfall. And rodents are notorious for infesting what? Rice field, that is paddy fields. So this is RRR for us. Okay. So this has been asked in FMG December 2024 exam. ठीक है. अब how humans get infected? Remember, humans get infected by contact of the broken skin. ठीक है. Not intact skin, contact of the broken, that is breached skin. मतलब स्किन पे चोट होनी चाहिए. Either broken skin का contact या mucous membrane का contact with the contaminated water, urine, blood or the tissues of the infected rodent, and not by rodent bite. ठीक है. So let's write this. Humans get infected by contact of breached skin or that is mucous membrane with the contaminated surface water. Dihydrogen monoxide matlab water, urine, tissues or blood of the infected animal, not on rodent bite. And if we talk about the clinical features, Some people may be totally asymptomatic. Some people may have fever only. Acha, by the way, this has been asked as a question. This was a NEET SS exam ka question. Ki what is the outcome of infection in most infected people with leptospira interrogans? So most people don't mount any symptoms only. क्योंकि immunity भगवान की कृपा से बहुत बढ़िया है कि symptoms ही नहीं होंगे, अच्छी बात है ना? So most infected people are asymptomatic and those which do develop symptoms उनमें क्या क्या हो सकता है? या तो only fever होगा, ठीक है, या तो ओनली फीवर, और सम पीपल में डेवलप हेपेटोरेनल सिंड्रोम, हेपेटोरेनल syndrome, that is liver and kidneys shut down. This is seen in Weil's disease, ठीक है. And what is the pathology of leptospirosis? Now guys, this is a question mark. कुछ साल पहले तक क्या बोलते थे? वास्कुलाइटिस. बट अभी जो लेटेस्ट एडिशन है ऑफ हैरिसन्स इन्फेक्शियस डिजीजेस, उसमें एक लंबा सा पैराग्राफ दे रखा है कि वास्कुलाइटिस हो सकता है, ये आर्टरीज इन्वॉल्व हो सकती हैं. फिर उस पैराग्राफ के एंड में ना आप हंसोगे सुनगे कि क्या लिख रखा है कि दिस इज स्टिल नॉट दिस इज स्टिल अ क्वेश्चन मार्क, ऐसा कुछ लिख रखा है, मतलब कि दिस इज स्टिल नॉट प्रूवन येट. ओके, बट प्रीवियसली ना ये एक ओल्ड क्वेश्चन है जो पूछा जाता था, ठीक है. तो अगर ये ओल्ड क्वेश्चन मान लो रिपीट हो जाए तो हम वास्कुलाइटिस ही मार्क करेंगे. तो अभी भी जो थ्योरी है वो वास्कुलाइटिस ही है, वो अलग बात है कि इट इज स्टिल टू बी proven with confidence. Okay. Now, what are the important pathological features in the kidney? Interstitial, interstitial nephritis and tubular necrosis. An important pathological feature in the liver, centrilobular necrosis. ठीक है. Then diagnosis के लिए we can do microscopy of blood and urine, but it has got a very low sensitivity. Low sensitivity, तो ये usually कोई नहीं करता है. Now culture, इस पे question आता है. Culture can be done on 3 media. Remember them as fake, but culture also is not done routinely. Not done because it is cumbersome, मतलब difficult to do. ठीक है. तो FE की fake, हम 3 media के नाम याद करेंगे. F से Fletcher's medium, E say EMJH medium, Ellinghausen, Macular, Johnson, Harris medium. You don't have to remember the full form. These are names of the people who devised this medium. And K say क्या है? Korthoff's medium. Korthoff's medium. So remember FEK fake. Now serology. Now I come to a very interesting part of this infection. Serology, remember, is the gold standard. And what is the method used? Microscopic agglutination test, that is MAT, microscopic agglutination test, or MAT, to detect what? To detect antibodies. And this test, ये भी recent question आया हुआ है. This uses live bacteria, live leptospira interrogans. ठीक है. This was a question asked कि कौन से बैक्टीरिया यूज होते हैं live. ठीक है. Now treatment as far as is concerned, penicillins and tetracyclines can be used. Now we come to Treponema pallidum. See, Leptospira interrogans also is a spirochete, मतलब spiral, very slender organism. Treponema pallidum also is a spirochete. Okay. Now, what disease it causes? It causes syphilis. Most common mode of infection is sexual intercourse, in which case it is referred to as venereal syphilis. सिफिलिस. ठीक है. देखो वेनेरियल का पता है एक्चुअल मतलब क्या होता है? इट पर्टेन्स टू वीनस, द गॉडेस ऑफ लव. सो दिस adjective venereal has been used for sexually transmitted infections, but there are other modes also. For example, blood transfusion. That is why syphilis, Treponema pallidum is one of the organisms which are for which blood is screened, okay, before transfusion. Blood transfusion, transplacental organ transplantation. So venereal syphilis, as I said, it's an STI, sexually transmitted infection it is. And incubation period क्या है? 9 से 90 दिन. ठीक है. This has been asked in INICT exam. ठीक है. अब INICT में आया तो आपको भी आ सकता है. INICT में in fact incubation period के ना match the following question आया था, and precisely This question, 9 to 90 days wala na, it was asked in INICT May 23. Okay. On my Instagram handle, therefore, I had put up 3 separate posts for incubation periods of important diseases. वो भी मैं share कर दूंगी. So please stay connected with me on Instagram. Okay. I'll share it in my story. Okay, late tonight, if possible. Otherwise, tomorrow morning. And I'll do one thing, guys. I'll make a separate highlight folder for all of you. तो highlights में क्या होगा, आप highlights में click करोगे ना तो वो stories आप देख सकते हो. Usually stories तो 24 hours में delete हो जाती है, but वो highlights मैं इस RRR Remarkable Rapid Revision नाम से I'll make a highlight folder, उसको देख सकते हो आप. ठीक है, तो चलो RRR, एक RRR तो में हम पढ़ ही रहे हैं अभी, और एक RRR हमने बैक्टीरिया के कंटेक्स्ट में कर लिया. ठीक है, इंस्टा आईडी मेरा है mj.micro. ठीक है, mj इनिशियल्स ऑफ माय नेम एंड माइक्रो, यू नो, माइक्रोबायोलॉजी. ठीक है, इन मेरे सोशल मीडिया हैंडल्स इस बुकलेट के फर्स्ट पेज पे बाय द वे दे भी रखे हैं. ठीक है. Now, coming to the stages of syphilis, primary syphilis. When we were learning today about donovanosis or granuloma inguinale, I told you कि उसमें क्या होता है, painless genital ulcer, and यहाँ पे भी क्या है, painless genital ulcer known as hard or Hunterian chancre plus painless inguinal ellenpathy, that is lymphadenopathy. So this is how you distinguish donovanosis from syphilis. In donovanosis, there is no painless inguinal lymphadenopathy. All right, and these lesions are highly infectious. They resolve on their own, but that does not mean cure. Patient enters secondary syphilis, which is a systemic infection. Matlab the organisms now are in the bloodstream. So it is characterized by what? It is characterized by a generalized skin rash, and therefore syphilis is also known as great pox. It is also characterized by generalized ellenpathy, that is lymphadenopathy. Okay, the skin rash, by the way, it involves palms and soles also. It involves palms and soles too. Then oral mucus patches, and also genital lesions, multiple genital lesions known as condyloma lata, which are also painless. So in primary syphilis, remember, there's a single genital lesion which is painless, and in secondary syphilis there are multiple genital lesions which are painless, known as condyloma lata. Okay. Now, if not treated, these lesions resolve, but it does not mean cure, and patient enters a latent stage. Just a second, guys. Now, latent stage means what? Latent matlab छुपा हुआ. तो in latent syphilis, what happens? In latent syphilis, there are no clinical features, but positive serological tests for syphilis. Positive serological tests for syphilis. ठीक है. Now, if untreated, some patients enter the stage of tertiary syphilis, which is of 3 types: benign tertiary, cardiovascular, and neurosyphilis. Benign tertiary syphilis is also known as gummatous syphilis. Now, G से gamma, G से granuloma. So, gummatous syphilis is characterized by lesions known as gammas, which are granulomatous lesions. They can occur anywhere in the body, but most commonly they are found in bones and skin. Okay. Cardiovascular syphilis, this you read in pathology as well. So यहाँ पे क्या हो रहा है? The pathology is obliterative and arteritis of the vasa vasorum supplying ascending aorta and arch of aorta. Okay, and weakening of these vessel walls therefore will lead to what? Aneurysm, syphilitic aneurysm. Okay. Now coming to neurosyphilis. Neurosyphilis may manifest as what? Either asymptomatic, meaning CSF will be positive for serological test, but patient will have no symptoms, or it may manifest as aseptic meningitis, that is lymphocyte predominant meningitis, or it could be meningovascular syphilis with involvement of the blood vessels and stroke like features, or if spinal cord is involved, then tabes dorsalis. Okay, tabes dorsalis, or neuropsychiatric manifestations may occur, which are described as general paresis. Previously, it was called as general paresis of the insane, but it's a derogatory term, so it is not used anymore. It's just referred to as general paresis. Condyloma acuminata is seen in case of HPV, human papilloma virus. Condyloma acuminata are nothing but the anogenital warts caused by the DNA virus HPV. Okay? Most commonly caused by HPV genotypes 6 and 11. Okay. Now coming to the diagnosis, next page. Microscopy, it's more fruitful to do microscopy in the primary and secondary syphilis, because ideally in primary syphilis, because antibodies tab tak nahi bani hongi, the older an infection becomes, there are more chances that antibodies would have been produced by the immune system. ठीक है, तो प्राइमरी एंड सेकेंडरी सिफिलिस में ज्यादा सेंस बनती है माइक्रोस्कोपी करने की, बट यूजुअली रिमेंबर सिफिलिस के लिए माइक्रोस्कोपी कोई भी नहीं करता, बिकॉज देर आर वंडरफुल सीरोलॉजिकल टेस्ट अवेलेबल. ठीक है, बट यस, प्राइमरी सिफिलिस में माइक्रोस्कोपी मेक सेंस. आई मीन टू से सेकेंडरी सिफिलिस में अगर ऑप्शन हो ना, तो गो फॉर सीरोलॉजी. ठीक है, इवन दो माइक्रोस्कोपी मे बी पॉजिटिव, बिकॉज सीरोलॉजी इज वे इजियर टू डू. ठीक है. Now, dark ground microscopy can be used to see, Treponema pallidum. But what are other disadvantages? The smear has to be seen immediately. The smear has to be seen immediately, otherwise the organisms will die. Okay, so you have to see the motile organisms. Then a better stain is DFA stain, that is direct fluorescent antibody stain, which is highly specific, so look at this picture. So dark ground microscopy, pehla wala, direct fluorescent antibody staining looks like this, and this is the silver impregnation method. So silver impregnation method bhi kar sakte hain. Okay. Then serology. It makes more sense to do serology in secondary and tertiary syphilis. Okay? Because by that time antibodies are expected to have been produced. So we have got the non-specific or non-treponemal test and the specific or treponemal test. As the name implies, in non-specific or non-treponemal test, the antigen used is a non-specific antigen, namely cardiolipin. Okay? Cardiolipin. So cardiolipin is not an antigen specific to Treponema pallidum. Specific yet treponemal tests are the ones where the antigen is derived from the pathogenic strain. Pathogenic strain of Treponema pallidum, namely Nichols strain. Nichols strain. So the antigen which is derived from Nichols strain is a specific antigen. Matlab it is found only in the pathogenic Treponema pallidum, not the non-pathogenic strains. The non-specific or non-treponemal test कौन से हैं? Wassermann test, which is a complement fixation test, CFT. Then CANS test. CANS test is a tube precipitation or a tube flocculation test, whereas VDRL test is a slide precipitation or a slide flocculation test. VDRL is what? Venereal Disease Research Lab, यूनाइटेड स्टेट्स पब्लिक हेल्थ सर्विसेज, जहां पे ये टेस्ट डिवाइस किया गया था. देन वी हैव गॉट आरपीआर. आरपीआर क्या है? इट्स नथिंग बट रैपिड प्लाज्मा रिएजेंट टेस्ट. रैपिड प्लाज्मा रिएजेंट टेस्ट, इट्स अ मॉडिफिकेशन ऑफ वीडीआरएल टेस्ट. क्या मॉडिफिकेशन होती है? द एंटीजन दैट इज कार्डियोलिपिन इज कोटेड विद carbon particles to make the reaction visible to the naked eye. लेकिन VDRL में you need the help of a microscope to comment on the reaction, to assess the reaction. ठीक है. तो आरपीआर में नेकेड आई से हम देख सकते हैं रिएक्शन को। देन व्हाट आर द एडवांटेजेस ऑफ नॉन स्पेसिफिक टेस्ट? भाई जरूरत क्या है नॉन स्पेसिफिक टेस्ट करने की व्हेन वी हैव गॉट स्पेसिफिक टेस्ट टू मेक अ कंफर्मेटरी डायग्नोसिस? एडवांटेजेस ये हैं कि नंबर वन, दीज आर टेस्ट फॉर प्रोग्नोसिस, सो दे आर प्रोग्नोस्टिक टेस्ट, मतलब कि दे विल बिकम नेगेटिव विद सक्सेसफुल ट्रीटमेंट। नंबर टू, दे आर चीप। आप लोगों ने देखा होगा जो एंटीनेटल केयर के दौरान ना एक इम्पॉर्टेंट इन्वेस्टिगेशन की जाती है प्रेग्नेंट लेडीज की, एंड दैट इज फॉर सिफिलिस। तो उनमें इन प्रेग्नेंट लेडीज में आरपीआर टेस्ट इज डन। व्हाई इज इट इम्पोर्टेंट? बिकॉज ट्रांसप्लेसेंटल ट्रांसमिशन ऑफ ट्रिकोनियम पैलिडम कैन अकर, है ना? तो अगर आरपीआर टेस्ट विच इज अ सस्ता टेस्ट, इफ इट कम्स आउट टू बी पॉजिटिव, देन दैट फीमेल प्रेग्नेंट फीमेल विल बी सब्जेक्टेड टू अ स्पेसिफिक टेस्ट so as to confirm whether it is syphilis or it is false positive, because congenital syphilis is a known entity. ठीक है, तो अगर the mother pregnant lady is infected with Treponema pallidum, simply start treatment so as to protect the baby. ठीक है? Now, what are the disadvantages? Disadvantages of non-specific tests are they are not confirmatory and biologically false positive reactions can occur. Now, examples of such false positive reactions are, देखो इसको ऐसे लिखो, वायरल वैक्सीनेशन, वायरल निमोनिया, वायरल हेपेटाइटिस। ठीक है, नंबर वन। देन इन्फेक्शियस मोनोन्यूक्लियोसिस, देन नंबर थ्री ऑब्वियसली प्रेगनेंसी, एचआईवी इन्फेक्शन, रिलैप्सिंग फीवर, ठीक है, ओल्ड एज, ट्रॉपिकल पल्मोनरी, इयोसिनोफिलिया, इंट्रावीनस ड्रग अब्यूज, autoimmune disorders, for example RASLE, and chronic tropical diseases, for example मलेरिया, लेप्रेसी। डोंट बी पेट्रिफाइड ऑफ दिस लिस्ट। इस पे ना मैंने काफी टाइम पहले इंस्टाग्राम पे एक रील बनाई थी एक निमोनिक के साथ। तो वो निमोनिक ये था कि अ वीआईपी ईट्स हॉट रोटी इन एसी। अ वीआईपी, वेरी इंपॉर्टेंट पर्सन, ईट्स हॉट रोटी, ROTI, हॉट रोटी इन एसी, एयर कंडीशनर। तो वो भी मैं शेयर कर दूंगी, तो वो विजुअल एक इंप्रेशन माइंड में कर लेना, वो आपको याद हो जाएगा, सारे एग्जांपल्स याद हो जाएंगे, डोंट वरी। ठीक है, नाउ द स्पेसिफिक ट्रिपोनिमल टेस्ट जो है ना, दे आर ऑब्वियसली कंफर्मेटरी। फॉर एग्जांपल वी हैव गॉट ट्रिपोनिमा पैलिडम, ट्रिपोनिमा पैलिडम particle agglutination test, that is TPPA, which is overall the most S and S, मतलब कि most sensitive and specific test. Number two, Treponema pallidum heme agglutination test, that is TPHA, TPHA। ठीक है, देन तीसरा क्या है हमारा, ये ना क्या कहते हैं, एक है FTA absorption test, FTA absorption test। दिस इज बेस्ड ऑन द प्रिंसिपल ऑफ इम्यूनोफ्लोरेसेंस, इनडायरेक्ट इम्यूनोफ्लोरेसेंस। एंड चौथा क्या है हमारा, ये है ट्रिपोनिमा पैलिडम इमोबिलाइजेशन टेस्ट, जो कि आजकल नहीं करते हैं। ठीक है निरीशा, ओके, गुड क्वेश्चन। अच्छा मुझे एक बात बताओ, कोई गाना या पिक्चर हम 10 बार सुने, है ना, हमें याद नहीं हो जाता, होता है ना? तो एक ही की है याद करने का कि आप बार बार रिवाइज करो। ठीक है, मुझे तो पढ़ाते हुए 10 साल हो गए हैं। ठीक है, आई स्टिल रीड टुडे। तो ऐसा नहीं है कि बस 10 साल हो गए हैं एंड बस हो गया, ऐसे नहीं है। तो we also have to update ourselves and enhance our knowledge, so that we as educators give you guys our best. When we give our best and you do your best, then बल्ले बल्ले हो जाएगी, आप लोग सक्सेसफुल होंगे। और अगर आप सक्सेसफुल हुए तो दुनिया में दो लोग हैं जो आपसे भी ज्यादा खुश होंगे, एक आपके मम्मी पापा और एक आपके टीचर्स। ठीक है, तो एक ही की है कि बार बार रिवाइज करो। और मेरी कोशिश इस सेशन में देखो यही है, मैंने शुरू में भी कहा था कि अगर आपको सिर्फ मेरी बातें सुन के ही 50-60% आपको आज ही याद हो गया, दैट मीन्स आई हैव सक्सीडेड। ऑब्वियसली आज के सेशन के बाद आपको टाइम निकाल के इसको रिवाइज भी करना है, तभी सेशन का फायदा होगा, है ना? Now, what are the advantages of ट्रिपोनिमल टेस्ट? एक तो भाई ये कंफर्मेटरी, सबसे बड़ी एडवांटेज यही है, और क्या चाहिए? What are the disadvantages? The advantages of non-specific test become disadvantages here. तो नॉन स्पेसिफिक की डिसएडवांटेज क्या थी? वो सॉरी, नॉन स्पेसिफिक की एडवांटेज क्या थी? वो सस्ते थे, ये क्या है? महंगे हैं, दे आर कॉस्टली. दूसरी एडवांटेज क्या थी नॉन स्पेसिफिक की? दे आर यूज्ड एज प्रोग्नोस्टिक टेस्ट, मतलब कि दे बिकम नेगेटिव विद सक्सेसफुल ट्रीटमेंट, बट these remain positive. These remain positive even after treatment, even after treatment. So they can't be used for prognosis. ठीक है, चलो. अब ट्रीटमेंट के लिए भाई द ड्रग ऑफ चॉइस इज पेनिसिलिन. लकीली कोई रेजिस्टेंस टू पेनिसिलिन हैज नॉट बीन नोटेड. ठीक है, दैट्स अ गुड थिंग. अब बैक्टीरिया को हम बाय बाय बोलते हैं, प्रणाम करते हैं, आगे जाते हैं माइकोलॉजी पे. ठीक है, माइकोलॉजी स्टडी ऑफ फंगाई, मेडिकल माइकोलॉजी स्टडी ऑफ मेडिकली सिग्निफिकेंट फंगाई. अब देखो बहुत इजी है ये. डर्मेटोफाइट्स जो होते हैं ना, दे कॉज व्हाट? सुपरफिशियल माइकोसिस. माइकोसिस टर्म का क्या मतलब है? फंगल इन्फेक्शन. सुपरफिशियल मतलब कि दे इन्वॉल्व सुपरफिशियल स्ट्रक्चर्स लाइक स्किन, हेयर एंड नेल्स. तो डर्मेटोफाइट्स में देर आर थ्री जेनेरा: ट्राइकोफाइटॉन, माइक्रोस्पोरम, एपिडर्मोफाइटॉन. ट्राइको, ट्राई मतलब तीनों. सो दीज इन्वॉल्व ऑल थ्री: स्किन, हेयर एंड नेल्स. माइक्रोस्पोरम, इट इन्वॉल्व्स व्हाट? स्किन एंड हेयर. ठीक है. एंड एपिडर्मोफाइटॉन इन्वॉल्व्स क्या? देखो, एक डिग्री होती है ना, सुपर स्पेशलिटी डिग्री, MCH. आप सी की जगह एस कर दो, MSH याद कर लो. ठीक है. और क्या याद करो? ENS याद कर लो. देखो, ENS का कोई फुल फॉर्म नहीं है, ENS. तो N से हमारा क्या हो गया? नेल्स, और S से स्किन. अब देखो, स्किन जो है ना, वो तीनों ही इन्वॉल्व कर रहे हैं. ठीक है. तो ऑब्वियसली जब हम ये याद करेंगे, स्किन तो तीनों में ही बेचारी, स्किन तीनों में ही इन्वॉल्व हो रही है. तो अगर आपको MSH याद हो गया, तो फिर यहाँ पे ई, मतलब ई से एपिडर्मोफाइटन, तो यहाँ पे हेयर तो आ नहीं सकता, तो यहाँ पे नेल्स डाल दो, है ना. चलो, अब इस पे क्या क्वेश्चन आता है भाई? पिक्चर बेस्ड क्वेश्चंस आते हैं ऑन द एसेक्सुअल स्पोर्स, दैट इज माइक्रो एंड मैक्रोकोनिडिया पे. तो माइक्रोकोनिडिया, मैक्रोकोनिडिया, दे आर व्हाट? एसेक्सुअल स्पोर्स, एज द नेम implies. Microconidia are small in size, they are unicellular, and macroconidia are bigger in size, they are multicellular. अब पहले ट्राइकोफाइटॉन की बात करते हैं. ये देखिए, ट्राइको में माइक्रो अबुंडेंट है. देखो, जो लोग कितने लोग बोल बोल के पढ़ते हो, बोल बोल के, ट्राइको में माइक्रो, ट्राइको में माइक्रो, ट्राइको में माइक्रो. तो बस इसको ऐसे में सुन के याद कर लो भाई. ट्राइको में माइक्रो अबंडेंट होते हैं. तो मैक्रोकोनिडिया वेरी फ्यू, और वेरी फ्यू एंड रेयर. When present, they are pencil shaped or cylindrical. Okay, now guys, have a look at this. Microscopic appearance of various dermatophytes. LPCB mount. तो LPCB mount बेसिकली ना हम कॉलोनीज के साथ करते हैं, लैक्टोफिनॉल कॉटन ब्लू माउंट टू सी द स्पोर्स. तो यहाँ देखो, लेट मी जस्ट जूम दिस पिक्चर. सबसे पहले वाला कौन सा है? ट्राइकोफाइटॉन मेंटाग्रोफाइट, स्पीशीज का नाम है. लुक एट दिस. यू कैन सी इतने छोटे 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 डॉट्स. सो ऑल ऑफ दीज आर माइक्रोकोनिडिया. तो ट्राइको में माइक्रो अबंडेंट है. एंड लुक एट दिस ब्यूटीफुल बिग साइज पेंसिल शेप्ड और सिलिंड्रिकल मैक्रोकोनिडिया. ठीक है. अब हम आते हैं माइक्रोस्पोरम पे. अब देखो, माइक्रोस्पोरम में ना क्रॉस कर दो. माइक्रोस्पोरम में क्रॉस ऐसे कर दो, ऐसे. तो जो चीज ऊपर अबंडेंट थी, मतलब ऊपर क्या था? ट्राइकोफाइटोन में माइक्रोकोनिडिया वर अबंडेंट, बट इन केस ऑफ माइक्रोस्पोरम, मैक्रोकोनिडिया बिकम अबंडेंट. ठीक है. और इसको ऐसे क्रॉस कर दो. तो माइक्रोस्पोरम में माइक्रो रेयर है, माइक्रो रेयर है, मैक्रो अबंडेंट है. ठीक है. इज दिस क्लियर? अब दीज आर बोट और बोट शेप्ड और फ्यूजिफॉर्म. ऑब्वियसली गाइस, जो मैक्रोकोनिडिया जो मैंने बोला बड़े साइज के हैं, तो उन्हीं की हम शेप पे कमेंट कर पाएंगे ना. अब ये देखो, ये पिक्चर देखो कितनी अच्छी है. माइक्रोस्पोरम कैनिस जो है ना, लुक एट दिस, इट इज लाइक दिस, बोट शेप्ड और फ्यूजिफॉर्म ऐसे. ठीक है. नाउ एपिडर्मोफाइटॉन. अब दिस इज एन इंटरेस्टिंग फेलो. देखो एपी का क्या मतलब होता है, अबव या ऊपर। ये बड़ा नार्सिसिस्टिक डर्मेटोफाइट है, अपने आप को बस एक कहावत है ना कि हम ही हम, बाकी सब पानी कम। इसको अपने सामने कोई पसंद नहीं है, ठीक है? तो ये बहुत ही ईगोइस्टिक, घमंडी टाइप का है। सो इसको छोटे-छोटे माइक्रोपोनिडिया से कोई मतलब नहीं है, तो दे आर टोटली एब्सेंट, टोटली एब्सेंट। It has got abundant club shaped or clavid shaped macroponidia which are present in bunches. ये बहुत इंपॉर्टेंट है भाई, इमेज बेस्ड क्वेश्चन इन सब पे आ रखे हैं, तो मैंने इमेज भी डाली हुई है। लुक एट दिस, एपिडर्मोफाइटोन, ये तीसरे वाला। लुक एट दिस, these are club shaped and you can see these are present in bunches like this, ठीक है? खलील, ओके, यू कैन प्लीज रेस्ट, और आप रिकॉर्डेड वीडियो फिर बाद में देख लेना, अभी तो देखो चलेगा। थैंक यू सो मच निरंजन। चलो गुड, आई एम सो ग्रेट कि याद हो रहा है साथ साथ। चलो, वेरी गुड, कि दिस इज व्हाट गिव्स मी एनर्जी एक्चुअली, ठीक है? तो नेक्स्ट इंपॉर्टेंट एमसीक्यू, हेयर परफोरेशन टेस्ट। तो यहाँ पे ना वी लुक फॉर अ वेज शेप्ड wedge shaped perforation in hair. We basically treat the sample of the we basically treat take the colonies and we take a petri dish, उसमें hair डालते हैं और प्रोसीजर होता है, उसको डिटेल में नहीं जानना। बस याद रखो, if a wedge shaped perforation is found, it is said to be a positive hair perforation test, and hair perforation test is positive in case of trichophyton mentagrophytes and microsporum canis. This is a PYQ आया हुआ है ये, ठीक है? It is negative in trichophyton rubrum. तो रिमेंबर, रूब्रम रोता रहेगा, रूब्रम रोता रहेगा। क्यों रोएगा? क्योंकि उसने परफोरेट नहीं किया हेयर को, हेयर परफोरेशन टेस्ट उसका नेगेटिव आ गया, ठीक है? ऑलराइट, आगे बढ़ते हैं। क्रोमोब्लास्टोमाइकोसिस, देखो वेरी वेरी इंटरेस्टिंग। इसकी भी ना मैंने इंस्टाग्राम पे क्या कहते हैं, मैंने एक स्लाइड डाली थी। तो एज आई सेड, मैं वो सब शेयर कर दूंगी, वो प्लीज आप लोग देखना। आपकी हाईलाइट का नाम ना मैं आर 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 कर दूंगी, रिमार्केबल रैपिड रिविजन करके। ओके, तो अब इसको पहले हम सुनते हैं, इसको हम ऐसे याद कर लेंगे। क्रोमो मीन्स व्हाट? कलर। याद है आज क्लास के बिगनिंग में क्रोमा की वजह से वो ग्रीन ग्रीन टेक्स्ट नहीं दिख रहा था, ठीक है? तो क्रोमा का क्या मतलब होता है? कलर, कलर, ठीक है? तो एक क्रोमा अब हम पढ़ रहे हैं। ब्लास्टो से आप सोचो कि समथिंग इज कमिंग आउट ऑफ द स्किन, समथिंग इज ब्लास्टिंग आउट ऑफ द स्किन, स्किन से कुछ परफोरेट होके बाहर आ रहा है, बाहर निकल रहा है। माइकोसिस मतलब फंगल इन्फेक्शन। सो क्रोमोब्लास्टोमाइकोसिस इज अ टाइप ऑफ mycosis. Which category of mycosis is it? It is a subcutaneous mycosis, matlab it involves the skin and subcutaneous tissue, and it is seen in males more commonly as compared to females, affecting lower limbs more commonly than the upper limbs, and the mode of infection is traumatic inoculation. Now let me connect the dots for all of you. When I say it occurs because of traumatic inoculation, please remember the causative fungi of this fungal infection, they are not commensals, they are there in the environment, saprophytes. They are found on the dead and decaying organic matter, vegetation. तो मान लो कोई घर से बाहर गया, अब गाइस दिस इज जस्ट अ मेमोराइजिंग टिप, ठीक है? डोंट टेक इट पर्सनली एट ऑल, दिस इज जस्ट अ टिप टू हेल्प यू रिमेंबर। घर से बाहर कौन ज्यादा रहता है? मेल्स रहते हैं। तो जब मेल्स घर के बाहर जा रहे हैं, दे आर मोर प्रोन टू ट्रॉमा और एक्सीडेंट। एंड मान लो कोई गिर गया, गलती से पैर फिसल गया, तो ज्यादा पॉसिबिलिटी चोट आने की कहाँ होती है? पैर पे, लोअर लिम्ब पे। तो इस तरह से आप याद करो, मोड ऑफ इन्फेक्शन ट्रोमेटिक इनोकुलेशन। घर से कौन बाहर जाएगा यूजुअली? और बाहर है फंगाई, एनवायरमेंट में फंगाई, तो घर से बाहर कौन जा रहा है मोर कॉमनली? मेल्स देन फीमेल्स। और जब वो गिर विर जाएगा तो लोअर लिम्स मोर कॉमनली अफेक्टेड विल बी अपर लिम्ब, ऐसे याद कर लो। इट्स अ क्रॉनिक स्लो ग्रोइंग इन्फेक्शन। द लीजन्स आर व्हाट? पॉलीमॉर्फिक। पॉली मीन्स मेनी and morpho means morphology. So different variety, different types of lesions, buffet of lesions can be seen, but the most common type is kya? Verrucous lesions, that is cauliflower, cauliflower like lesions। मतलब कि गोभी का फूल देखा है ना, उसका सरफेस इज सो इरेगुलर, उबड़ खाबड़ सा। सो दैट इज द मीनिंग ऑफ verrucous or cauliflower like lesion with an irregular surface. Other types of morphologies of the lesions can also be encountered, like for example ulcerative lesions, ठीक है? Ulcerative lesions, or nodular, that is tumor like lesions can also be encountered. And chromoblastomycosis, remember chromo matlab color, अब हम आते हैं उस color वाले चीज पर. तो chromo matlab color. This fungal infection is caused by dementatious or pheoid fungi. These are colorful fungi. When I say colorful fungi, I don't obviously mean vibgyor, I just mean black or brown hyphae. Okay, please remember there is nothing called as black fungus. That was a media generated term for mucor, when mucormycosis cases escalated during the covid pandemic. पर उसको black fungus नहीं बोलते हैं. That black fungus जो मीडिया ने बोला था, actually that was in reference to the necrosis, necrotic lesions caused by mucor, क्योंकि mucor is an angio-invasive fungus. ठीक है, तो remember यहाँ पे भी ना, these fungi are not called as black or brown fungi, they are known as dementaceous or pheoid fungi, जिसका क्या मतलब होता है, the fungi have got black or brown hyphae. Now these are the names of the fungi. ठीक है, इससे घबराना नहीं है इनके नाम से. अपने आप को ये दिलासा दो कि एग्जाम में ना मेरे सामने 4 ऑप्शंस हैं, ना कोई मेरा वाइवा ले रहा है, ना मुझे लिखना है. ठीक है, स्पेलिंग की भी टेंशन नहीं है. तो बस आपको ये मैं सजेस्ट करूंगी, जस्ट ऑन अ स्मॉल स्टिक ऑन राइट दीज 5 नेम्स एंड स्टिक इन योर रूम. रोज इसको दिन में 3-4 बार देखोगे, याद हो जाएगा. तो वी हैव गॉट फॉन्सेशिया पेड्रोसॉय, फॉन्सेशिया कॉम्पैक्टा, इसको मैं ऐसे लिख देती हूँ, ऐसे याद कर लो. देखो एफ से फॉन्सेशिया है, तो फॉन्सेशिया की 2 स्पीशीज हो गए, पीसी अगेन प्रियंका चोपड़ा, पेड्रोसॉय कॉम्पैक्टा, और पीसीआर. पीसीआर याद कर लो, पी से फियालोफोरा वेरुकोसा. देखो यहाँ पे वेरुकोसा आ गया, मैंने वेरिकोस लीजन बताई है. फिर क्लैडोस्पोडियम कैरियोनाe, ठीक है, करीना कपूर कैरियोना याद कर लो, राइनोक्लैडियला एक्वास्पर्सा. इतने फैंसी फैंसी अजीब से नाम, ये क्रोमोब्लास्टोमाइकोसिस में है, जिसका नाम इतना लंबा है. ठीक है, ऑल राइट. डायग्नोसिस, वेरी वेरी फेमसली आस्क्ड क्वेश्चन, एफएमजी जून 22 में जैसे फॉर एग्जांपल पूछा गया था. बायोप्सी, आप टिश्यू बायोप्सी लोगे, यू विल सी दी स्फेरिकल सेल्स विद इंटरनल सेप्टेशंस. लुक एट दिस. सो दीज सेल्स आर ब्राउन सेल्स. ठीक है, लेट मी ड्रॉ schematic diagram over here. These are brown cells like this with these internal septations. And what are these cells known as? They look like or they resemble copper pennies, so they are known as copper penny cells. Is absolutely right, Nirisha. Copper penny cells, also known as medullary bodies, also known as muriform cells, also known as sclerotic bodies. ठीक है, हम इसे कैसे चालाकी से याद करेंगे? रिमेंबर एमएससी मास्टर ऑफ साइंस, पर यहाँ पे एम 2 बार आएगा. ठीक है, M2SC मास्टर ऑफ साइंस. एम से मेडलर बॉडीज म्यूरिफॉर्म सेल्स, सी से कॉपर पेनी सेल्स. तो शक्ल से ही पता चल रहा है इन बॉडीज की, इन सेल्स की. एंड हमारा उससे क्या है, एस से स्क्लेरोटिक बॉडीज. इज दिस क्लियर एवरीवन? क्लियर एवरीवन? Now coming to sporotrichosis. Sporotrichosis again is a subcutaneous, infection. And interestingly, this is caused by a thermally dimorphic fungus. Thermally dimorphic fungi generally cause systemic infection, but this is an exception. So it is caused by the fungus, namely Sporothrix. Sporothrix shankai. This is an example of a thermally. This is an example of a thermally dimorphic fungus. मतलब कि di मतलब दो, morphic मतलब morphology. So at 2 different temperatures, these fungi exist in 2 different morphologies. The thermally dimorphic fungi. ठीक है, तो 2 डिफरेंट टेम्परेचर्स कौन से हमारे लिए इंपॉर्टेंट है? तो ह्यूमन बॉडी टेम्परेचर, बिकॉज वी आर गोइंग टू कलेक्ट सैंपल फ्रॉम द पेशेंट. तो 37 डिग्री सेल्सियस पे ये फंगस कैसे दिखते हैं, इनकी शक्ल कैसी होती है, वो पता होना चाहिए. प्लस दीज फंगी आर देयर इन द एनवायरमेंट, सो वी शुड नो व्हाट इज देयर मॉर्फोलॉजी एट द एनवायरमेंटल टेम्परेचर. इन एंटायर मेडिकल माइकोलॉजी यू हैव टू रिमेंबर 6 थर्मली डायमॉर्फिक फंगाई, जिसमें से एक तो हम अभी पढ़ रहे हैं, बाकी 5 अभी आने वाले हैं. सो इट कॉजेज व्हाट? इट कॉजेज subcutaneous, it causes subcutaneous infection, and this fungus is present on wood and plants, and it is an exogenous infection. And the mode of infection again is traumatic inoculation. And the most common clinical form is what? The most common clinical form is lymphocutaneous. lymphocutaneous sporotrichosis, and this has been asked as a question. So देखो क्या होता है ना, this fungus has got a predilection to involve the lymphatic vessels. So trauma हुआ, मान लो कोई कारपेंटर है, he is working with the wood, and the wood has got the fungi obviously are not visible to the naked eye, or you are plucking a rose for somebody, ठीक है, the and you unfortunately get pricked by the rose thorn. So and the rose also is harboring this fungus. So in either case, the fungus enters the enters the through the skin, it enters then the subcutaneous tissue, it eventually enters what, lymphatic vessels, because it loves to invade lymphatic vessels. Now lymphatic vessels are not going to keep quiet, they will respond by showing what, lymphangitis. And what will the fungus do then? Fungus dar jayega, and it will come out of the lymphatic vessels, and it will produce ulcers in series or rows. Initially there will be nodular lesions which will break down to form ulcers. So the typical description of lymphocutaneous sporotrichosis is what? It is characterized by ulcers in series or rows. And this entity, remember also, मैंने लोअर लिम्ब की फोटो डाली है, रिमेंबर अगेन ट्रोमेटिक इनोकुलेशन, घर के बाहर गया, मेल गया, गिर गया, तो लोअर लिम्स मोर कॉमन देन अपर लिम्ब, इससे याद रखो। ये एग्जैक्ट सेम पिक्चर एग्जाम में आ रखी है, आज इन एफएमजी दिसंबर 2024, ठीक है। चलो, दिस डिजीज बाय द वे is also known as rose pickers disease. So think twice before plucking rose for anyone. Rose pickers disease, also known as carpenter's disease, also known as rose gardener's disease. And in India, in our country, it is prevalent in the sub-Himalayan region. Okay? Himalayan region. Now we come to the morphology. से दिखते कैसे हैं? So look at this. This is the morphology at 37 degrees Celsius. The thermally dimorphic fungi exist as yeast at 37 degrees Celsius. So look at this. This is the yeast, which is cigar shaped, central cigar shaped yeast, surrounded by the peripheral host reaction, which is nothing but deposition of antigen antibody complexes, and this together is looking like a star. So this whole thing, the central cigar shaped yeast cell surrounded by this star like host cell reaction, together this is known as asteroid body, also known as splendor Hopley phenomenon. Splendor Hopley phenomenon. Now look at this. This is the morphology encountered at 25 degree Celsius, matlab environmental temperature. So, these small, round things are what? These are all the microconidia of this fungus. And what is their arrangement? How is their arrangement described as? ये फूल की तरह लग रही है ना, flower की तरह. So this is described as what? All of these are referred to as rosette arrangement. So remember in sporotrichosis, we learned about rose thorn prick, rose thorn prick, rose garter disease, and rosette arrangement. Okay, rosette arrangement. Then treatment, the drug of choice क्या है? Potassium iodide. Okay, the drug of choice for all forms of sporotrichosis is potassium iodide. देन ऑपर्चुनिस्टिक माइकोसिस, ठीक है। कई बार ना हम लाइफ में ऐसे लोगों से डील करते हैं जो एक्चुअली में अपॉर्चुनिस्ट होते हैं, दे आर नॉट आर वेल विशर्स, ठीक है, तो वो हमें बेवकूफ बनाते हैं, मैनिपुलेट करते हैं इन सर्टेन सिचुएशंस। तो in microbiology also we have got infections known as opportunistic infections. For example, opportunistic mycosis, meaning these are the fungal infections caused by the fungi which will not disturb anyone if there is no risk factor, there is no opportunity. But when these fungi get some opportunity, for instance jeopardized immunity, then they will disturb that individual. So opportunistic mycosis का एक बहुत important कई सारे risk factors होते हैं, like malnutrition, alcoholism, HIV infection. इन सब में common क्या है? Jeopardized immunity. And आने वाले टाइम में ना देखो, माइकोलॉजी इज गोइंग टू बी, आई मीन माइकोलॉजी नाउ आल्सो इज इंपॉर्टेंट, बट आने वाले टाइम में और ज्यादा इंपॉर्टेंट होगी। पता है क्यों? क्योंकि वी हैव गॉट एन एवर इंक्रीजिंग पॉपुलेशन ऑफ transplant recipients and cancer patients वगैरह, which by the grace of God they are getting now much better treatments. But in treatments का एक side effect क्या है? Compromised immunity. So that, is a little debilitating, because then these patients, even though their, life is prolonged, these therapies add certain years to the life of these patients, but they have to pay a price, and that is opportunistic infections by organisms which will not disturb otherwise normal people. I mean, जिनकी immunity अच्छी है. ठीक है. So opportunistic mycosis, सबसे पहले हम बात करेंगे candidiasis और moniliasis. It is caused by genus Candida, which is yeast like fungus. ठीक है. And Candida albicans is the most common cause of infection in humans. It's a commensal on the skin, oral cavity, gastrointestinal tract, and genito urinary tract. And what are the risk factors? HIV infection, ठीक है, alcoholism, steroids and other immunosuppressive drugs, ठीक है, zinc deficiency. And what are the clinical features? Yes, of course, diabetes, also diabetes mellitus. Oral thrush or oral candidiasis characterized by white painless patches in the oral cavity. Then esophageal candidiasis is described as what? An AIDS defining illness. AIDS defining illness. Meaning, if it also is characterized by whitish patches along the esophageal mucosa. And if it is diagnosed and the status of HIV is not known in that individual, then that patient should be investigated for HIV infection, because this is an AIDS defining illness. Okay. Then vaginal candidiasis is seen in diabetic females asked in FMGE Jan 24 exam. So, what are the clinical features? Initially a watery vaginal discharge, and this is going to be followed by curdy white. This is going to be followed by curdy white vaginal discharge, and the patient is going to have intense pruritus, okay, intense pruritus, and vaginal pH vaginal pH is not altered. It is going to remain less than 4.5. Any other condition you can think of where vaginal pH is not altered? Or sorry, vaginal pH is altered. Normal pH of vagina is acidic only, that is less than 4.5. Here it is not altered. But also make a note key in bacterial vaginosis and trichomoniasis, the vaginal pH becomes more than 4.5, matlab it is altered. Okay. Then intertrigo, infection of the folds of skin, those areas of the body where skin forms folds, like axillary region, submammary region. Then it can cause infective endocarditis, candidemia, and UTI also. So when you do a wet mount, you will see what? You will see these budding yeast cells. Okay. Gram stain will show you gram positive budding yeast like cells, budding yeast like cells. तो वेट माउंट में भी ना आपको ऐसे बडिंग yeast like cells दिखेंगे, बट वो ब्लू कलर के नहीं होंगे, बिकॉज इट्स सिंपली अ वेट माउंट. ग्राम स्टेन पे आपको कलर दिख जाएगा. रिमेंबर, ऑल फंगाई आर ग्राम पॉजिटिव ओनली, बट ग्राम स्टेन सबसे अच्छे से किस फंगस को स्टेन करता है? कैनेडा और क्रिप्टो, मतलब कि यीस्ट एंड यीस्ट लाइक फंगाई. ठीक है. Then culture on SDA will show you white or creamy pasty colonies. Colgate toothpaste जैसी consistency of the colonies, so it is called as pasty colonies. ठीक है. Then germ tube or Reynolds Brodé phenomenon. तो इसमें क्या करते हो? कॉलोनीज ली, उसको सीरम के साथ मिक्स किया, and then you incubate it for 4 hours, and then you see the projection of a tube-like structure from the parent yeast, something like this. ठीक है. Germ tube formation, and germ tube formation is positive in Candida albicans and also Candida doubliniensis. Then we have got chrome agar medium. Again, देखो chrome मतलब color. तो इस पे different species produce different color की colonies, and based on the color of the colonies we can comment on the species. Canada albicans for example produces green colored colonies. ठीक है, green colored colonies, वही green color, ठीक है, जिससे आज थोड़ा सा हम अटके रहे शुरू में. Then treatment. Cutaneous candidiasis and oral thrush के लिए topical azoles are the drug of choice. Esophageal and vaginal के लिए oral fluconazole या फिर caspofungin. Disseminated के लिए liposomal amphotericin B and caspofungin. Please do these drugs very well from your pharmacology. ठीक है. Now coming to next opportunistic mycosis, cryptococcus. It is also known as European blastomycosis. It was actually discovered in the late 19th century in Germany. तो इसको यूरोपियन बोल दिया था, ठीक है. और ब्लास्टोमाइकोसिस एक नाम है जो उस टाइम पे ना फंगल इन्फेक्शंस के लिए यूज किया जाता था, ठीक है. एंड इट इज कॉज्ड बाय क्रिप्टोकोकस नियोफॉर्मन्स. इट इज अ ट्रू ईस्ट. ट्रू ईस्ट मतलब no pseudo hyphae. Whereas remember Canada जो होता है ना, Canada is called Canada is not a true yeast. Canada, why? Because Canada shows pseudo hyphae formation. So Cryptococcus neoformans has got a polysaccharide capsule. In immunocompetent people there are no clinical features, but in immunocompromised people it will lead to pulmonary infection followed by CNS infection, that is a meningitis. Meningitis. And इस meningitis का important clinical feature क्या है? The patient will complain of headache. Not the typical Koenig's and Brudzinski's signs will be positive in such people, but patient will come to you and tell you कि डॉक्टर साहब बहुत दिन से सर में दर्द हो रहा है. और अगर आपको HIV स्टेटस पता है उस पेशेंट का कि वो HIV पॉजिटिव है, then you should definitely rule out Cryptococcus neoformans. ठीक है. Then virulence factors. Of course जहाँ पे कैप्सूल आ गया वो तो प्रिंसिपल virulence factor होगा ही. तो polysaccharide capsule is what? It is antiphagocytic. It is antiphagocytic and ability to make melanin using phenyl oxidase. Then this is not V, this is U. U stands for urease and P stands for phospholipase. Phospholipase. Okay. Then what are the risk factors? HIV infection with CD4 count less than 200 per microliter, very important very important risk factor. Then hematologic malignancies, immunosuppressive agents, and transplant recipients. Remember them as HIT. These people will be hit by this fungus. 2H H2IT. पहला H HIV infection, दूसरा H hematologic malignancies, I say immunosuppressive agents, and T say transplant recipients. ठीक है. So, cryptococcal meningitis, remember, this is the MCC, most common cause of meningitis in immunocompromised people, mainly with HIV, less than 200 microliters, frequently asked question. Now, what specimens can you collect for diagnosis? Blood, CSF, urine, या फिर skin scrapings. Okay. Make a note of the fact that agar to skin is involved, if skin is involved by cryptococcosis, it implies the infection has disseminated. So it is disseminated cryptococcosis. So skin involvement is a proof of dissemination by this fungus. Then it's a capsulated fungus, and very commonly negative stain is done by India ink or nigrosin. This also has been asked, FMGE December 2024. So look at this. Capsule appears as a refractile delineated space around budding yeast cell. Look at this. So this is Cryptococcus neoformans, India in preparation. Arrow points to a budding yeast of Cryptococcus neoformans. You can note the thick, translucent polysaccharide capsule. Sometimes, the width of the capsule can be more than the width of the fungus itself. Okay. Then gram stain. Gram stain will show you gram positive budding yeast, something like this. Okay. Mucicarmine stain. Remember, mucicarmine में CAR आ रहा है ना, CAR. So remember, it stains the carminophilic. It stains the carminophilic cell wall of this fungus. तो ऐसे याद कर लो. Car से carminophilic. Then antigen detection can be done. किसका antigen? The capsular polysaccharide antigen, because it is shed off by the fungus in the body. So it can be detected in CSF, urine, or serum by latex agglutination test. This is, by the way, a highly S and S sensitive and specific test. Then culture. On STA, white mucoid, white shiny colonies, white colonies which are mucoid, that is shiny. Why they are mucoid? They are shiny or mucoid because of the PS capsule, polysaccharide capsule. Remember, Candida colonies are also white, but they are pasty. They don't shine. They are not mucoid. Okay, they are pasty, look like toothpaste, सफेद रंग का Colgate toothpaste, but these are also white, but they're shiny. Bird seed and Niger seed agar. B for bird, remember B for black colored colonies. Okay, then Niger itself means black only, so Niger seed agar may be black colonies. Urease test is positive. So remember, this is an important example of a fungus which is urease positive. Then another question, Maltese cross appearance. It refers to what? It refers to the arrangement of capsular material. ये arrow डाल के please लिख लो. Capsular material of Cryptococcus neoformans as seen under which microscope? Polarizing microscope. ठीक है? Polarizing microscope. ठीक है? Maltese cross एक किसी उसमें दिखी जाती है, किसी पैरासाइट में. यस, बबेसिया में. ठीक है, बबेसिया विच इज द भाई ऑफ प्लास्मोडियम. ठीक है, बिकॉज बोथ प्लास्मोडियम एंड बबेसिया आर इंट्राइराइथोसिटिक प्रोटोजोआ. नाउ कमिंग टू द ट्रीटमेंट पार्ट. Cryptococcus without CNS involvement, the drug of choice is fluconazole. ठीक है. In HIV infected people with CNS involvement, an induction phase for 2 weeks is needed with the help of amphotericin B plus flucytosine, and this should be followed by oral fluconazole therapy till CD4 count rises to more than 200 per microliter for 6 months. Please remember this. ठीक है. Then aspergillosis पे आते हैं. देखो, aspergillosis is caused by the fungi Aspergillus, and just like God, these fungi are also, omnipresent. They are there in the environment. They are saprophytes. If we are infected with this fungus, हमें तो symptoms ही नहीं होंगे because भगवान की कृपा से हमारी immunity बहुत बढ़िया है. But unfortunately, in people with jeopardized immunity, they will cause infection. And I'll tell you in a bit कि what exactly should be the reason of jeopardized immunity. ऐसा नहीं है कि हर cause of jeopardized immunity में aspergillosis हो जाएगा. नहीं, ऐसा नहीं है. Like for example, aspergillosis is very uncommon in HIV patients. ठीक है, अभी उसके बारे में बात करेंगे. So these are saprophytes. The important species are as follows. Aspergillus fumigatus. अब देखो, ध्यान से सुनना इसको. इसके नाम में क्या है? फ्यूमी. फ्यूमी मतलब फ्यूरियस, गुस्से वाला. ठीक है. So that is why this is the species which is the most disturbing for us. This hates humans. So this is the most common species causing human infections. ठीक है, पहली बात. And also it is responsible for most of the cases of pulmonary and allergic aspergillosis. Pulmonary and allergic aspergillosis. Then aspergillus flavors, it loves hospital environment. Okay? It is notorious for causing skin, sinus, and ocular infections. Then Niger colonizes the respiratory tract. It colonizes the respiratory tract and commonly causes what? Otitis externa, infection of the external auditory canal. And what is the mode of infection in case of aspergillus? Inhalation of the airborne conidia. Okay. Now, what are the risk factors? The risk factors include, remember, neutropenia, and that is why this infection, aspergillosis, is uncommon in HIV patients because in them, neutrophil count is preserved till the very last stages of AIDS. What is the problem in HIV, by the way? The problem is with T helper cells, okay, not the neutrophils. So that is why, aspergillosis, unlike candidiasis and cryptococcosis, is uncommon in HIV patients. Okay. Now, first entity is allergic bronchopulmonary aspergillosis, that is ABPA, which is seen in 1% of asthma patients and 15% of cystic fibrosis patients. It's basically a hypersensitivity reaction to Aspergillus fumigatus, but sometimes other species may also be involved. It is characterized by increased IgE antibody. Okay, and Aspergillus species specific antibodies will be there. Like for example, if Aspergillus flavus or any other species is involved, so against those species specific IgE and A will be present. Bronchial obstruction with mucus plugs is going to be there, causing coughing, pneumonia, breathlessness, and also make a note of the fact that bronchiectasis also is present in ABPA, very important. Then aspergilloma or fungal ball. Let's say, and this is the cavity in the lungs. So this cavity, two important causes of cavity, in the lungs, especially in India. Kya hai? TB and cancer. So sometimes what can happen is these aspergillus fungi, which are saprophytes, they can colonize this cavity and they won't invade the cavity. So aspergilloma or fungal ball is nothing but colonization of this cavity by the fungus, and this is non-invasive. This is non-invasive. All right. Again, the most common cause over here is Aspergillus fumigatus. Okay. Now, invasive aspergillosis. This entity, this fungus, I mean, can cause invasion of the blood vessels. It is an angio-invasive fungus, and the important risk factors are neutropenia and diabetes mellitus. Neutropenia and diabetes mellitus, remember. Then it can cause infection of cornea, that is keratomycosis, seen in farmers during the harvesting season, in the setting of splinter injury. So, during the harvesting season, when the farmers are using splinters, let's say if the splinter is contaminated with aspergillus, तो farmer को तो आंख से दिखेगा नहीं ना कि फंगस है, हमें भी नहीं दिखेगा. गलती से आंख में चला गया वो स्प्लिंटर, तो फिर क्या होगा? इन्फेक्शन हो जाएगा, इन्फेक्शन ऑफ द कॉर्निया, केरेटोमाइकोसिस. ठीक है. तो अगेन हियर आल्सो मोस्ट कॉमन कॉज इज aspergillus fumigatus. Then we have got otomycosis. This, by the way, is the most common disease in humans caused by aspergillus, and इसका भी MCC most common cause क्या है? इसका है Niger. इसका है Niger. ठीक है. Now look at this. Let me zoom this picture. So aspergillus fumigatus, septate hyphae. The long arrow points to the septate hyphae of aspergillus, and you can notice the straight parallel walls. And the typical short arrow points to the typical low angle Y-shaped branching. Look at this. Can you see this long arrow is pointing towards the septa? ठीक है, ये वाला, ये this is the septa. ठीक है, and look at this छोटे वाला arrow. Let me just delineate it, then I will remove the marking. Look at this. This is the acute angled branching shown by this छोटे वाला arrow. ठीक है. Now guys, a memorizing tip. इसको देखो जरा. Aspergillus. ए से हम क्या याद करेंगे? Acute angled, that is dichotomous ब्रांचिंग. ठीक है. और एस से हम क्या याद करेंगे? सेप्टेट हाइफे. ठीक है, सेप्टेट हाइफे. तो ए एस याद करो, इसे एक्यूट एंगल्ड या डाइकोटोमस ब्रांचिंग, एंड एस से क्या है? सेप्टेट हाइफे. नाउ इनवेसिव डिजीज का ड्रग ऑफ चॉइस क्या है भाई? देखो, इनवेसिव डिजीज मतलब कि खतरनाक डिजीज, एंड अ वेरी फैंसी वर्ड फॉर खतरनाक इन इंग्लिश इज वोरेशियस. ठीक है. तो वोरेशियस हम याद करेंगे वोरिकोनाजोल. वोरिकोनाजोल. तो इनवेसिव डिजीज का ड्रग ऑफ चॉइस क्या है? वोरिकोनाजोल. ABPA. A is a vowel and I is also a vowel. तो ABPA का ट्रीटमेंट ड्रग ऑफ चॉइस इट्राकोनाजोल. ठीक है. देन सिंगल एस्परगिलोमा के लिए सर्जरी इज इंडिकेटेड, एंड क्रॉनिक पल्मोनरी एस्परगिलोसिस के लिए ना ऊपर वाले दो ड्रग्स को नीचे ले आओ. इट्राकोनाजोल लिख दो, वोरिकोनाजोल, दोनों में से कुछ भी यूज कर सकते हो. बोथ कैन बी ड्रग ऑफ चॉइस. प्रोफाइलॉक्सिस के लिए पी से प्रोफाइलैक्सिस, पी से पोसाकोनाजोल. पोसाकोनाजोल. ये सब एमसीक्यूज है. ठीक है. देन वी कम टू म्यूकरमाइकोसिस. Again, mucormycosis, it is belonged it is caused by the class of the fungi, namely Phycomycetes, also known as Zygomycetes. ठीक है. तो इस क्लास में ना ये तीन जेनेरा आते हैं: राइजोपस, एब्सीडिया and mucor. So when mucor is responsible for infection, it is known as mucormycosis. All of these fungi are saprophytic in nature and they show right angle or obtuse angle branching, something like this. Okay, and they have got no septae, so the hyphae are said to be xenocytic. Okay, so this is how they branch. See, they are irregularly shaped hyphae, and this is the right angle or acute angled branching. The appearance of the hyphae is wide. They are काफी चौड़े होते हैं, thick walled ribbon like aseptate, that is cenocytic. So look at this, mucor species and non-separate hypha, arrow points towards an irregular shape, non-separate hypha of mucor. Now what are the risk factors? Remember DD, demand draft, desferrioxamine therapy and diabetic ketoacidosis. Now desferrioxamine kya hai? It is an iron chelator. People who are receiving, repeated blood transfusions like thalassemia patients and all, they are at risk of iron overload. And iron जब body में excess में होती है, it can attack our tissues. So simultaneously, these patients are also started on desferrioxamine. Desferrioxamine is a chelator of iron, meaning it will form a complex with iron so that iron is not available to attack the tissues. That tissue damage does not occur. Okay. These people who are receiving desferrioxamine, they are prone to infection with these fungi, phycomycetes like Rhizopus absidia mucor. Why? Because these fungi are iron loving fungi. आयन लविंग फंगाई, तो ऑब्वियसली किसी ह्यूमन बीइंग की बॉडी में ही आयन अतिरिक्त मात्रा में है, तो भाई उसको तो देखो ये सैप्रोफाइट है, आपको और मुझे भी इन्फेक्शन हो सकता है, बट वी आर नॉट ऑन डेस्फेरियोक्सामाइन थेरेपी, सो इवन वी आर, इफ वी आर इन्फेक्टेड वी वोंट हैव एनी सिम्पटम्स, आवर इम्यून सिस्टम इज इनफ फॉर taking care of us. But unfortunately, in such patients who are getting desferrioxamine, in them symptoms will occur, symptomatic disease will occur. Then another important risk factor is diabetic ketoacidosis. And remember, this fungus again is angioinvasive, just like Aspergillus, and it leads to thrombosis, hemorrhage, infarction, and necrosis. So, because of this necrosis, the media generated wrong term was in vogue 3-4 years back, black fungus. Okay? And what are the most important members of the immune system against these fungi? Neutrophils. Neutrophils. Okay. Then we come to rhino-orbitocerebral mucormycosis. This is the most common and fulminating type of mucormycosis. What is the mainstay of treatment of this? Surgical debridement. Okay. Surgical debridement. Antifungals like amphotericin B, posaconazole, isavuconazole can be used. This has been asked as a question. Isavuconazole किसके लिए use करते हैं? Cytokines like interferon gamma and granulocyte monocyte colony stimulating factor can also be given. But देखो, mainstay तो surgical debridement ही है, even before you start any of the antifungals or cytokines. Now coming to systemic mycosis, these are caused by again thermally dimorphic fungi, जिनके बारे में मैंने थोड़ी देर में आपको बताया था. एक थर्मली डायमॉर्फिक फंगस पोरोथेरिक शेंका तो हमने कर ही लिया, बाकी 5 अब करेंगे. तो सबसे पहले हिस्टोप्लाज्मा कैप्सुलेटम, इट इज नॉट कैप्सुलेटेड, दिस इज एन ओल्ड क्वेश्चन. द डिजीज कॉज्ड इज हिस्टोप्लाज्मोसिस, also known as Darling's disease, named after Sir Samuel Darling, who described this fungus. Morphological forms. At 37, budding yeast form is seen. That is the parent yeast cell with a single tiny bud, 2 to 4 microns. Look at this. So yeasts are there within the macrophages. The purple things are the yeast and the brown things are the macrophages. So it looks something like this. देखो, ये रहा parent yeast cell, इससे एक tiny छोटा सा bud arise करेगा. This is 2 to 4 microns in size. And at 25 degrees Celsius, the mold or filamentous form, that is septate hyphae with asexual spores or conidia. Now look at this. The asexual spores are of 2 types. Macroconidia. Macroconidia को क्या बोलते हो? Tuberculate. Tuberculate macroconidia, and microconidia simply called as microconidia. If the question mentions this word tuberculate, okay, you have to mark histoplasmosis. This is, pathognomonic of histoplasmosis, the presence of tuberculate macroconidia. So these are, multicellular macroconidia which have got these, spine-like projections. These are spikes on their surface. And microconidia बहुत छोटे-छोटे होते हैं, unicellular microconidia. Okay. Then second one is, by the way, guys, please remember, in case of thermally dimorphic fungi, questions are very frequently asked on morphology. तो ये सारे morphologies आपको याद होनी चाहिए. And very simple scoring questions these are. Blastomyces dermatitis. देखो, blasto मतलब कहीं blast होना. Let's say कहीं CNG blast हो गया. ठीक है, तो ब्लास्टोमाइसिस डर्मेटाइटिस जो करता है बीमारी ब्लास्टोमाइकोसिस, द डिजीज ब्लास्टोमाइकोसिस इज आल्सो नोन एज, सी से शिकागो डिजीज, एन से नॉर्थ अमेरिकन ब्लास्टोमाइकोसिस, एंड जी से गिल क्राइस्ट डिजीज. Morphology: at 37 degrees Celsius, they are in the form of, remember, broad based budding yeast cells, something like this. This is the parent yeast cell. It is giving rise to a daughter bud. And look at this. This is a broad base. So it is also known as what? Figure of eight appearance. Okay. In histoplasmosis, and even in Candida and Cryptococcus, where also budding is seen, there the daughter bud has got a narrow base. Okay. So remember, B say blastomycosis, B say broad-based budding yeast cell. Okay. Then at 25 degrees Celsius, mold form is present with septate hyphae and pear-shaped microconidia. Pear-shaped microconidia. There are no macroconidia. Just a little memorizing tip, guys. Blasto. मैंने क्या बताया शुरू में? CNG ब्लास्ट हो रहा है, तो कहीं भी कुछ ऐसे ब्लास्ट हो गया तो टुकड़े 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 छोटे छोटे पीसेस बच जाएंगे, है ना? तो यहाँ पे भी ना छोटे छोटे माइक्रोकोनिडिया होते हैं, ठीक है, माइक्रोकोनिडिया. रिमेंबर, देर आर देयरफॉर नो मैक्रोकोनिडिया. ब्लास्ट के बाद कुछ भी मैक्रो नहीं बचने वाला, ठीक है. मोड ऑफ इन्फेक्शन, इनहेलेशन ऑफ कोनिडिया. So these are schematic representations, yeast with broad-based budding cells at 37 degrees Celsius and mold with microconidia at 20 degrees Celsius. Environmental temperature. Look at this actual picture. Look at this. Large, broadly based budding yeast cells, characteristic of blastomyces dermatitis in a GMS stain biopsy section from a human leg. Okay. Then Coccidioides immitis. This is the third dimorphic fungus amongst the systemic mycosis. Mold form is there in the soil. All right. And infection has been associated with certain ground disturbing activities, such as landscaping, construction, farming, archaeological excavation, because all of these activities will facilitate what? It will facilitate the arthrospores, which are the infective form, to be present in the air. These arthrospores, these are barrel shaped. They are very, very tiny and they can be inhaled to set up the infection. Okay. So look at this. These are the arthropods associated with the mold form. And remember, this form at 37 degrees Celsius, this kind of a spherical form is seen, that is a spherule with endospores. So that, यहाँ पे the yeast form is not seen at 37 degrees Celsius, unlike the other dimorphic fungi. यहाँ पे there's a spherical structure filled with endospores. Okay. Look at this. Actual picture of arthrospores. Look at this. This is how they look like, beautiful barrel-shaped arthroconedia. Then we have got, look at this actual picture of a spherule. So long arrow points towards a spherule and the short arrow points to an endospore. So these spherules are thick wall structures containing many, many endospores. Okay. And it is coccidioidomycosis is found in the arid regions of what? Arid regions of California, Arizona, and the disease is also known as valley fever or California fever. ठीक है. रघुल पायलट मराइनर कैप्टन व्हील इसमें नहीं दिखता, वो पैराकॉक्सीडियोइड्स में दिखता है. पैराकॉक्सीडियोइड्स ब्रेजिलियंस तो भी आगे आएगा. आई थिंक आप उसके बारे में कह रहे हो शायद. So Paracoccidioides brasiliensis, again a dimorphic fungus. It causes what? It causes the disease South American blastomycosis, also known as Lutz mycosis. Okay? And it is found where? It is found in Brazil and अदर कंट्रीज ऑफ साउथ अमेरिका। देखो मैं तुम्हें एक चीज बताती हूँ। देखो पैरा के ना यहाँ पे दो चीजें हम दो मतलब याद करेंगे। देखो पैरा का एक मतलब हिंदी में क्या होता है, नीचे पड़ा हुआ, लाइंग एट द बॉटम, नीचे पड़ा हुआ। तो नीचे पड़े होने से क्या करेंगे, साउथ अमेरिका, नॉट नॉर्थ अमेरिका, साउथ अमेरिका। ठीक है, दैट इज वन मीनिंग। पैरा का एक और मतलब क्या होता है, ऑल अराउंड, ऑल अराउंड। तो यहाँ पे हम क्या याद करेंगे, यहाँ पे ऑल अराउंड द यीस्ट सेल बड्स आर प्रेजेंट। तो मल्टीपल बड्स, इससे पहले वाले जो हमने फंगाई पढ़े, हिस्टोप्लाज्मा, ब्लास्टोमाइकोसिस, वहां पे पेरेंट यीस्ट सेल से वन सिंगल बड वाज अराइजिंग। यहाँ पे पेरेंट यीस्ट सेल से मल्टीपल बड्स आर अराइजिंग, रिजेंबलिंग कैप्टन व्हील्स अपीयरेंस, आल्सो नोन एज mariner's wheel appearance or pilot's wheel appearance. I hope this is clear. ठीक है, then we have got the last dimorphic fungus, Penicillium marnifi, also known as Talaromyces marnifi. So at 37 degrees Celsius, it exists like this: yeast cell, yeast cell, which with the transverse fissure. Okay, so it doesn't show typical budding. It undergoes multiplication by transverse fission. So this is the transverse fissure found in this yeast cell. And at 25 degrees Celsius, it exists as a mold form. Mold form, which has got a red wine colored pigment, and the microconidia show brush border or broom like arrangement. Brush border or broom like arrangement of microconidia. And what are the reservoir of this fungi? Chinese bamboo rat. मैंने चाइना इन माइक्रोबायोलॉजी पे भी एक पोस्ट करी थी, दैट आल्सो आई विल शेयर इन माय स्टोरी, ठीक है, टुमारो। तो सोचो घर में चाइनीज बैम्बू रैट आ गया, तो उसको भगाने के लिए आपने ब्रूम, झाड़ू पकड़ लिया। ठीक है, यस यस, एब्सोल्युटली ट्रैंक्विल। यहाँ पे देखो, अच्छा वन मोर थिंग, यहाँ पे मान लो इफ ओनली टू बर्ड्स आर देयर, देन इट इज नोन एज व्हाट, दैट देन इट इज नोन एज मिकी माउस अपीयरेंस। आई वंडर व्हाई डोंट दे कॉल इट मिनी माउस अपीयरेंस। एनीवेज, so it is known as mickey mouse appearance if only two birds are there. Okay, so with this we complete our systemic mycosis, and now we will do some important topics of what, parasitology. The first topic is trichomoniasis, very famously asked question. So trichomoniasis is a sexually transmitted infection, and it is caused by trichomonas vaginalis. अब देखो, टी से ट्राइकोमोनास, टी से हम क्या लिखेंगे, ट्रोफोजोइड। सो दिस इज बाय द वे अ प्रोटोजोन, दिस इज अ प्रोटोजोन। टी से ट्राइकोमोनास, टी से ट्रोफोजोइड। सो ओनली ट्रोफोजोइड स्टेज इज सीन, नो सिस्ट स्टेज, नो सिस्ट इज सीन। दिस वाज आस्क्ड इन एफएमजी जून 2022। ठीक है, मेल्स आर यूजुअली यहाँ पे एसिम्टोमेटिक, अनलाइक गोनोकोकल एंड नॉन गोनोकोकल यूरोथ्राइटिस वेयर मेल्स आर यूजुअली सिम्टोमेटिक, यहाँ पे मेल्स आर एसिम्टोमेटिक। फीमेल्स ऑन द अदर हैंड दे आर symptomatic. The vaginal pH is altered, it becomes more than 4.5 and whiff amine test is positive. मतलब vaginal discharge को आप mix करोगे with 10% KOH, you will appreciate a fishy odor, and this odor is because of the production of amines. And colposcopy will show you colpitis macularis or strawberry vagina, which means what? It just means punctate hemorrhages on ectocervix and vagina. ठीक है, तो स्ट्रॉबेरी टंग आज हमने कर लिया, स्ट्रॉबेरी वजाइना हो गया। मोटिलिटी, टी फॉर ट्राइकोमोनास, टी फॉर ट्विचिंग मोटिलिटी। आज मैंने आपको बताया कि ट्विचिंग मोटिलिटी इज सीन इन सर्टेन स्ट्रेन्स ऑफ गोनोकोकस, उसको प्लीज याद रखना, अदरवाइज मोस्ट ऑफ द स्ट्रेन्स ऑफ गोनोकोकस आर नॉन मोटाइल। तो यहाँ पे द मोटिलिटी इज ट्विचिंग मोटिलिटी, ऑल्सो नोन एज वॉबलिंग मोटिलिटी, ऑब्लिक ऑल्सो नोन एज जर्की मोशन, जर्की मोशन। ठीक है, एंड फॉर कल्चर Feinberg's medium. is used. And as far as treatment is concerned, metronidazole is given. And look at this, the two trophozoites of Trichomonas vaginalis. Look at this. So let me draw a schematic diagram. So, it has got a single nucleus. देखो trichomonas में mono आ रहा है ना, so this is a single nucleus, and it has got these, you know, two pairs of flagella coming from the anterior end and one sidewards flagella. This is known as undulating membrane. Undulating membrane. इसकी भी मैंने ना एक डूडल बनाया था, that also I will share. Whatever we have covered today, उसके relevant जो भी post मेरी already insta पे हैं, that I will share in the in my stories, Instagram story tomorrow. इन द हाईलाइट RRR, जब आप मेरी स्टोरी देखोगे ना तो उस पर टॉप लेफ्ट में RRR लिखा हुआ आएगा, तो उससे समझ जाना कि वो आपके लिए है, उसको जरूर देख लेना। ठीक है। नाउ nematodes और roundworms, these are examples of what? Nematodes or roundworms, they are examples of helminths. Helminths also are a kind of parasites. तो parasites broadly दो तरह के होते हैं, protozoa and helminths. Nematodes which cause infection by ingestion of eggs. EAT, ingestion से याद करेंगे हम EAT, where E stands for Enterobius vermicularis. A से क्या है? Ascaris lumbricoids. T से क्या है? Trichuris trichiura. It is trichiura, not trichiuria. ठीक है। Nematodes which cause infection via skin penetration by the larvae. ठीक है। तो remember them as HS, मतलब highly sticky worms. High HS मतलब highly sticky, where H stands for hookworms and S stands for strongyloids. Strongyloids stercoralis. ठीक है, strongyloids stercoralis. Now have a look at this. Nematode, Ascaris, hookworm, strongyloid cercoralis. These are examples of small intestinal nematodes. मतलब कि the adult worms in these three nematodes, they reside in the small intestine, whereas Enterobius and Trichuris trichura, they are examples of large intestinal nematode. Just a little memorizing tip, guys. Remember let. L से क्या है? Large intestine, E से Enterobius और T से क्या है? Trichuris trichura. Now infective form in case of Ascaris lumbricoids, egg हमने ऊपर लिखी दिया है, hookworm में larva, strongyloids में भी larva, Enterobius में egg और Trichuris में भी egg, ये हमने ऊपर भी लिख लिया है। ठीक है। Diagnosis, detection of egg in the feces, hookworm में भी egg in the feces, strongyloids में larvae in the feces, Enterobius में egg in the perianal region, not in the feces, egg in the perianal region. ठीक है। एंड ट्रिचरस ट्रिचरा के केस में एग इन द फीसेस, एग इन द फीसेस। तो एग्स पे काफी क्वेश्चंस आते हैं, आगे हम अभी उसके बारे में डिस्कशन करेंगे। Ascaris lumbricoids is also known as what? It is also known as giant roundworm. ठीक है। It is roughly 25 centimeters in size. Now adults of this worm, by the way, adults are macroscopic, they are big enough to be seen with naked eye. ठीक है। Adults cause intestinal clinical features जैसे कि abdominal pain, diarrhea. Because these worms are so huge in size, they can cause intestinal obstruction, intestinal perforation, और अगर common bile duct में घुस गए तो they can cause what? Obstructive jaundice. Obstructive jaundice. And the larvae migrate in the lungs. L से larvae, L से lungs, L से हम क्या लिखेंगे? Low flowers pneumonia. Low flowers pneumonia basically is a hypersensitivity reaction to larval antigens. तो lungs में क्या हुआ है? eosinophilic exudate होता है in response to the larval antigens. ठीक है। Remember, there is no malabsorption. Ascaris malabsorption नहीं कराता। malabsorption कोई आपसे पूछे कौन सा पैरासाइट कराता है तो आप क्या बोलोगे? Giardia lamblia. ठीक है। Not Ascaris, because Ascaris जो है वो ल्यूमन में रहता है, गट के ल्यूमन में रहता है, तो वहीं से द इंजेस्टेड फूड जो हम लोग खाना खाते हैं उन्हीं से वो अपनी सस्टेनेंस डिराइव करता है। No malab, it doesn't attach to the gut cells. Then hookworms. देखो, remember hookworms hook onto the skin. तो mode of infection क्या है? Skin penetration. And what is the infective form? Larvae. ये हमने ऊपर लिख दिया है। Now, old world hookworm is ankylostoma duodenale, and the new world hookworm is Necator americanus. ओल्ड वर्ल्ड न्यू वर्ल्ड का क्या मतलब हुआ? ये कोई चीजें भी पता होनी चाहिए. ओल्ड वर्ल्ड का मतलब है कि बिफोर कोलंबस क्रिस्टोफर कोलंबस डिस्कवर्ड अमेरिका, मतलब ओल्ड वर्ल्ड में क्या क्या आता था? यूरोप, एशिया, अफ्रीका. तो आफ्टर अमेरिका वॉज डिस्कवर्ड, तो अमेरिकन कॉन्टिनेंट्स वर रेफर टू एज द न्यू वर्ल्ड. तो देखो नाम से पता चल रहा है भाई, न्यू वर्ल्ड वाले के नाम में अमेरिकैनस आ रहा है. ठीक है, तो ओल्ड वर्ल्ड हुकवर्म, न्यू वर्ल्ड हुकवर्म. टिपिकल फीचर हुकवर्म्स का क्या होता है? आईडीए, आयरन डेफिशिएंसी अनीमिया. ठीक है, आईडीए, आयरन डेफिशिएंसी अनीमिया. एडल्ट्स कॉज जीआईटी सिम्टम्स. ठीक है, abdominal पेन, लाइक फॉर एग्जांपल abdominal pain, diarrhea, and adults only cause IDA, iron deficiency anemia, and larvae in the lungs, they again cause Loeffler's pneumonia, Loeffler's pneumonia, and the larvae penetrating the skin, they cause a pruritic, maculopapular lesion called as ground itch. Ground itch. Remember, the larvae are the infective form. ठीक है, the larvae penetrating the skin जो infective form है, that is the filariform larva. This is the filariform larva, and strongyloids also. This is the filariform, that is L3 larva. This is L3 both. ठीक है. नाउ कमिंग टू स्ट्रोंगाइलॉइड सर्कोरैलिस. अब इसके नाम में क्या है? स्ट्रॉन्ग, बहुत स्ट्रॉन्ग है ये, और बहुत यूनिक भी है, इसलिए काफी स्ट्रॉन्ग कंटेंडर है ऑफ बीइंग आस्क्ड इन एग्जाम. तो इट इज स्ट्रॉन्ग इनफ टू पेनिट्रेट द स्किन. ठीक है, तो अगेन यहाँ पे भी इन्फेक्शन कैसे हो रहा है? स्किन पेनिट्रेशन से. इन्फेक्टिव फॉर्म क्या है? लार्वा. दिस इज द ओनली वर्म इन द एंटायर हेलमिंथोलॉजी विच इज व्हाट? विच इज ovoviviparous, मतलब कि the female lays the eggs which immediately hatch into larvae. So eggs are not going to be there in the feces. Eggs will be there for a very transient period of time. Eggs को बड़ी जल्दी मची रहती है कि वो larvae में convert हो जाए, परिवर्तित हो जाए. So the feces of the patient have only larvae, and the female is also parthenogenetic. Parthenogenesis का क्या मतलब होता है? The female gets the female can lay eggs without fertilization by the males. So eggs laid without fertilization by males. And interestingly, strongyloid cercoralis is both a पैरासिटिक वर्म एंड अ फ्री लिविंग वर्म. मतलब कि अगर किसी ह्यूमन बीइंग को ये इन्फेक्ट ना भी करे, तब भी ये अपनी लाइफ साइकिल आराम से एनवायरनमेंट में भी पूरी कर सकता है. एंड वेरी इंपॉर्टेंट, इट इज नोटोरियस या बदनाम फॉर कॉजिंग ऑटो इन्फेक्शन. ऑटो इन्फेक्शन इन इम्यूनो कॉम्प्रोमाइज्ड पीपल, हाइपर इन्फेक्टिव साइकिल्स करता है. एंड डिसेमिनेटेड स्ट्रोंगालोइडियासिस इज सीन इन HIV पॉजिटिव पीपल. ठीक है. नाउ डायग्नोसिस के लिए, जैसे मैंने बोला, कल्चर लार्वे विल बी देयर इन द फीसेस. कल्चर ऑफ द लार्वे कैन बी डन बाय हरडा मोरी कल्चर. एंड कॉन्सेंट्रेशन ऑफ द फीसेस के लिए एक टेक्निक होती है, बियरमैन्स टेक्निक. बियरमैन्स टेक्निक. द ड्रग ऑफ चॉइस ओवर हेयर इज ivermectin. Ivermectin, by the way, it is also the drug of choice for onchocerciasis. Onchocerciasis क्या होता है? It is nothing but river blindness, okay, caused by a filarial worm. ठीक है, तो एस्कैरिस, हुकवर्म और स्ट्रोंगलोइड्स क्या थे? स्मॉल इंटेस्टाइनल नेमेटोड्स. मतलब कि द एडल्ट वर्म्स आर फाउंड इन द स्मॉल इंटेस्टाइन. लार्ज इंटेस्टाइनल नेमेटोड्स में है एंटेरोबियस एंड ट्रिचुरिस, मीनिंग देयर एडल्ट वर्म्स आर फाउंड इन द लार्ज इंटेस्टाइन, वो उनका रेजिडेंस है. तो एंटेरोबियस वर्मिकुलरिस इज आल्सो नोन एज पिनवर्म और सीटवर्म और थ्रेडवर्म. ये बहुत छोटे छोटे होते हैं ये. ठीक है, एंड फीमेल्स के ना पोस्टीरियर पार्ट ऑफ द बॉडी पे एक पिन लाइक प्रोजेक्शन होती है, तभी इसको पिनवर्म भी बोलते हैं. सीट मतलब टॉयलेट सीट, और बहुत दे आर मिलीमीटर्स इन साइज, दे आर नोन एज थ्रेडवर्म, बहुत छोटे होते हैं. एंड क्लासिकली इट कॉजेज व्हाट? ऑटो इन्फेक्शन. इसके बारे में कहावत मशहूर है एंटेरोबियस के बारे में you had the infection as a child, you have the infection now, and you will have infection, and your kids also will have infection. When you have kids, your kids will also have infection. कहने का मतलब है the auto infective cycles are very very common, and they go on for years and years, almost refractory to treatment also. So most of the infections, but remember, are asymptomatic. अगर symptoms होते हैं तो classical clinical feature क्या होगा? Perianal pruritus. Perianal pruritus. The eggs are found in the perianal region and not in the feces. Not in the feces. The diagnosis is made by NIH swab, National Institute of Health swab, and स्कॉच टेप टेक्निक. ठीक है, तो स्कॉच टेप में you take a cellophane tape and stick it onto the perianal region in the morning before child goes for a bath. Then you remove that tape, and you see under the microscope, you examine it for the presence of eggs. NIH swab maybe na, there is a swab, and at one end of it is a cellophane tape, and it is rubbed over the perineal region, and then again it is transferred onto a slide and then seen under the microscope for the eggs, because eggs will not be present in the feces. Trichuris trichura, it is also known as what? It is also known as whipworm. And sigmoidoscopy shows what? Coconut cake rectum, which means adult worms. Adult worms hanging from sigmoid colon and rectum. ठीक है. Yes, there is some echo issue. I don't know why it is. ठीक है. Yes, but make sure you just keep a checklist of the PY Qs of the previous 3 years. Most of the things I've tried to incorporate in this. Okay, just a second, guys. Now we come to the morphologies of the eggs. What a coincidence! Even I'm tired. But ये हमें ढंग से तो करना ही पड़ेगा ना. थोड़ा सा ये और बचा है. We are almost on the verge of completion only. Now look at these eggs from left to right. Trichuris trichura, the first one. Look at this. It is barrel shaped, okay, and it is bile stained, okay. So remember yellow, yellow, dirty fellow. So yellowish, yellowish tinge matlab bile stained, okay. Then Enterobacteriaceae vermicularis. Look at this. It is whitish, whitish. So it is, and it is plano-convex, okay, plano-convex. So plano se yaad karo, it's very neat and clean. So it is non-bile stained. अब आते हैं एस्कैरिस. एस्कैरिस एंड हुकवर्म्स, हुकवर्म एग्स, बाय द वे, they are diametrically opposite to each other. ठीक है, क्या मतलब? Ascaris has got this rough surface. ठीक है, rough surface with these mammulations, these irregular, irregular surface like this, mammulations, and it is bile stained. Hookworm, on the other hand, has got a smooth surface and it is non bile stained. And inside all of these, this is these segmented ovum known as segmented ovum is there, and these segments are known as what? The segments are known as blastomeres. blastomeres. ठीक है. Now, ant amoeba histolytica, what type of parasite is it? It is a protozoan, and it is motile by what? It is motile by pseudopodia. What is the infected? What is the infective form? Infective form is this tetra or quadrinucleate cyst. Tetra or quadrinucleate cyst. And the disease causing form is the trophozoite form. Okay. The trophozoite form, by the way, it has got RBCs. It has ingested RBCs, so it is also known as hematophagus or erythrophagous trophozoites. Now, what disease does it cause? The diseases, 95% cases of Enterobacteriaceae histolytica are of intestinal amoebiasis. Okay? Intestinal amoebiasis. The most common site is ileocecal region, and the second most common site is sigmoid colon. Clinical features, the patient will have abdominal pain, blood in the feces, that is bloody diarrhea or dysentery, tenesmus. Okay. The large intestine has flask shaped or water bottle ulcers, that is ulcers with a narrow mouth and a broad base. Okay. Then 5% of the cases are of extraintestinal extraintestinal amoebiasis. So what happens? The trophozoites which are there in the intestine, they may invade the. They obviously invade the gut wall. That is why ulcers occur. But sometimes this invasion may be so florid that they can enter the portal vein and go here and there, and that is known as extraintestinal amoebiasis, also known as invasive amoebiasis. Invasive amoebiasis. Now, the most common site is liver. This is the most common site, and the typical feature is amoebic amoebic liver abscess. इसमें कौन सी पस प्रेजेंट होती है? पस का डिस्क्रिप्शन क्या है? एंकोवी sauce pus. Anchovy sauce pus. And clinical features, right upper quadrant pain, ठीक है, fever, hepatomegaly. Now look at this interesting picture. The cyst of Entamoeba histolytica or dispar with trichrome. You can see three nuclei are visible in the focal plane, and the cyst contains a chromatoid body with typical blunt ends. These chromatoid bodies are nothing but aggregations of ribosomes. In the mature cyst, matlab ki tetranucleate या quadrinucleate cyst, the chromatoid bodies will not be there. Okay. And look at this. I told you ना trophozoites have ingested RBCs, तो हमारा खून पीते हैं trophozoites literally. So trophozoites with ingested RBCs, these RBCs appear as dark inclusions. Okay. The nuclei, remember, have got small centrally located karyosom and a thin peripheral chromatin. All right. All right. Now, next few important points about Plasmodium, which is the malarial parasite. Now, in case of Plasmodium, remember the definitive host क्या है? Female anopheles mosquito, not the males, because females need a blood meal for egg maturation. And the intermediate host, the intermediate host are us, that is humans. Definitive host वो होता है जिसमें parasite की क्या होती है, sexual cycle होती है, and the intermediate hosts are the ones in which parasite की कौन सी होती है, asexual cycle होती है. ठीक है. So there are four species over here: वाइवैक्स, फैल्सिपारम, मलेरिया एंड ओवेल. इनको लेफ्ट से राइट कैसे याद करोगे? We listen to FM. We listen to FM, that is fast music. and O looks like O से ovale, it looks like zero, so इसको सबसे side में कर दो, and ovale, you know, it's very very rare in India, very very rare in India, इसको बिल्कुल right में कर दो, ठीक है, very very rare in India. All right, so look at this. So the gold standard method of diagnosis of malaria is what? Peripheral blood smear. Okay, peripheral blood smear. So peripheral blood smear mein you see different morphological forms. This is a schematic diagram. I have put up actual pictures also. So early trophozoid, इसको क्या बोलते हैं? Ring form. Ring form. So look at this. In case of now, I want you all to notice a few things over here. RBC size is enlarged in which case? In case of Vivax and ovale, you can clearly see. In FM, the RBC size is normal. So just a little memorizing tip. FM मैंने क्या बताया? Fast music. Sometimes, you know, some people, including me, we are like, I have a habit of, you know, reading while some light music is playing in the background. ये कुछ noise है, कुछ शोर हो रहा है. तो सोचो, it is normal to read with some music in the background. Similarly, it is so it's normal to have fast music in the background. Similarly, the RBC size is going to be normal in case of FM. I hope you got what I meant. Just a little memorizing tip. I repeat, FM, फास्ट म्यूजिक. फास्ट म्यूजिक के साथ साथ पढ़ना इज वेरी नॉर्मल. तो यहाँ पे एफ एम से क्या मतलब है हमारा? एफ से फैल्सीपैरम, एम से मलेरिया. तो फैल्सीपैरम एंड मलेरिया जब आरबीसी को इन्फेक्ट करते हैं, आरबीसी का साइज नॉर्मल रहता है. आई होप दिस इज क्लियर एवरीवन. जरा बता दो. ओवेल में देखो ओवल शेप हो गई, ठीक है. वाइवैक्स में शेप यू नो एनलार्ज्ड होगी. Is this clear? And in falciparum, the ring form, that is the parasitic form, it may be stuck onto the cell membrane known as a col form. There may be double dot ring form or multiple ring forms. If multiple ring forms are there, it means it is falciparum. And falciparum is the most, causes the most severe disease, most severe malaria amongst all. Then this early trophocyte matures a little, it becomes late trophocyte, so it will become a little irregular as you can see over here. Okay? And band form is seen in case of what? Band form is this rectangular form. It refers to the late trophozoid of Plasmodium malariae. Okay. Then schizons are multinucleated forms which are seen. Female and male gametocyte. Now this is a question. Remember, the gametocytes in case of falciparum is what? Sickle shaped or banana shaped, whereas other gametocytes are spherical. Again, this is a question. In fact, you know why falciparum has been named as falciparum? Because falci का मतलब ही होता है sickle. ठीक है. So banana shaped gametocytes are seen in, क्या मार्क करोगे? Falciparum. अब आते हैं हम असली पिक्चर्स पे. Look at this. Remember, the gold standard for diagnosis of malaria क्या है? PBS, peripheral blood smear. Please remember this. ठीक है. Thin blood smear showing different forms of Plasmodium falciparum. Look at this. फिगर ए में क्या है? मल्टीपल रिंग फॉर्म्स एंड अकोल फॉर्म्स. लुक एट दिस. मल्टीपल रिंग फॉर्म्स, अकोल फॉर्म्स. दिस अकोल फॉर्म, मल्टीपल रिंग फॉर्म्स. ठीक है. बीच में देखो डबल डॉट और हेडफोन अपीयरेंस. लुक एट दिस. डबल डॉट और हेडफोन अपीयरेंस. दिस इज बिकॉज ऑफ द फ्रैगमेंटेशन ऑफ द क्रोमैटिन. दैट इज व्हाई डबल डॉट इज विजिबल. थर्ड में देखो टिपिकल यू नो बनाना शेप गेमिटोसाइट. अच्छा, वन मोर थिंग गाइस. इन माय स्टोरीज ना डेली आई विल बी आल्सो पोस्टिंग सम एमसीक्यूज. ठीक है. तो प्लीज स्टे कनेक्टेड विद मी टिल योर एग्जाम. ठीक है. This is a very high yield session, but please remain connected with me on Instagram and on my Telegram group. And I'll also share tomorrow. I will create one WhatsApp group also. उसको भी join कर लेना. So you can stay connected with me directly on the WhatsApp group also to ask your doubts. ठीक है. तो उस पे भी I'll keep sharing my posts so that you know till the day of exam you will keep you know revising micro daily. ये तो करना ही है करना. तो रोज एक आपको अपनी तरफ से मैं एक जरा पुश जरूर दूंगी कि अच्छा ये रिवाइज कर लो, ये भी कर लो. ठीक है. तो रिविजन आपकी अच्छी हो जानी चाहिए उससे. ठीक है. Then, thin blood smear showing different forms of Plasmodium vivax. Look at this. Now, I want you all to focus on this. See, this is the ring form. Can you see the baki wale RBCs? They are you know normal in size. Vivax wala, the infected one, has become enlarged. ठीक है. तो वाइवैक्स में आरबीसी का साइज बड़ा हो गया. गेमिटोसाइट देखो स्फेरिकल है. देन शाइजोन मल्टी न्यूक्लिएटेड. Now look at this. Thin blood smear showing the ring form of Plasmodium malariae. Ring form of Plasmodium band form. Band form is nothing but the late trophozoite. ठीक है. It is not ideally the ring form, it is the late trophozoite. ठीक है. तो L हम यहाँ क्या याद करेंगे? Late trophozoite है ना, late trophozoite. लेट मतलब मर जाना मतलब बैंड बज जाना, तो फिर बैंड फॉर्म, ऐसा रेक्टेंगुलर वाला फॉर्म किसमें देखा जाता है? लेट ट्रोफोजोइट ऑफ व्हाट? प्लाज्मोडियम मलेरिया, प्लाज्मोडियम मलेरिया। ठीक है, now look at this. Thin blood smear showing the ring form of Plasmodium ovale. So RBC has become oval in shape, and look at this. Sometimes this kind of a fimbriated margin also may be appreciated. Fimbriated margin may also be appreciated. ठीक है, now coming to leishmania. Causes the disease leishmaniasis, which has got animal reservoirs like dogs, foxes, jackals and rodents. ओल्ड वर्ल्ड मतलब एशिया, न्यू वर्ल्ड मतलब अमेरिकाज, नॉर्थ एंड साउथ अमेरिका दोनों में लीशमैनियासिस पाया जाता है। इंडिया में देखो ये इंपॉर्टेंट है एपिडेमियोलॉजी पॉइंट ऑफ व्यू से, बिकॉज आपको क्लिनिकल मिनट में देख सकते हैं कि अच्छा सो एंड सो पेशेंट हेलिंग फ्रॉम झारखंड, राजस्थान, वेस्ट बंगाल, यूपी केम विद सो एंड सो सिम्टम्स। सो एपिडेमियोलॉजी पता होनी चाहिए डिजीजेस की। नाउ वैरायटी, सबसे पहले लीशमैनिया डोनोमैनी, इट कॉजेज डिजीज एक्सक्लूसिवली इन आर कंट्री इंडिया, known as Indian visceral leishmaniasis or kala-azar or black fever. The vector is sand fly, that is Phlebotomus argentifs. आर्जेंटिप्स इसलिए बोलते हैं क्योंकि द विंग्स हैव गॉट सिल्वर कलर। और फ्लेबोटोमस क्यों बोलते हैं, इसका भी मैंने इंस्टा पोस्ट पूरा डिकोड किया है, उसको एक बार जरूर देख लेना, आपको याद रहेगा। इट इज नॉट अ जूनोटिक डिजीज, ठीक है, काला आजार इज नॉट अ जूनोटिक डिजीज, ये क्वेश्चन आया हुआ है। ह्यूमंस आर दी ओनली रिजर्वोयर। अदर फॉर्म्स ऑफ लीशमैनियासिस आर जनरली जूनोटिक डिजीजेस, जैसे मैंने पहली लाइन यहीं पे मेंशन कर दी है। ठीक है, next, leishmania tropica is found in Rajasthan, Pakistan, Afghanistan. It causes what? It causes cutaneous leishmaniasis. देखो visceral leishmaniasis है, काला आजार तो viscera को involve करता है, it's a very serious disease. Cutaneous leishmaniasis involves only skin. ठीक है, it causes cutaneous leishmaniasis, also known as Delhi boil or Baghdad boil, Baghdad boil or ओरिएंटल सोर और एलेपो बॉयल। तो बेसिकली ना अलग अलग जगहों के नाम हैं जहाँ पे ये क्यूटेनियस लीशमैनियासिस वाज एनकाउंटर्ड, फिर नाम दे दिए गए। किसी को स्किन पे कोई सोर हुआ, बॉयल हुआ, तो फिर उसको बोल दिया दिल्ली बॉयल। बगदाद इज अ प्लेस इन इराक, तो वहाँ पे भी ये इन्फेक्शन पाया गया। फिर ओरिएंटल सोर, एलेपो बॉयल। ओरिएंटल सोर मतलब कि जब ब्रिटिशर्स थे इंडिया में भी, आई थिंक अर्ली 1900s की बात है, ये तब का है। तब ये नाम आया था। ठीक है, it is not dangerous because it affects only the skin and viscera is not involved. The vector is again sandfly, specifically Phlebotomus sargentii. लीशमैनियासिस में वेक्टर सैंडफ्लाई होता है। देन ब्रेसिलियंसिस causes mucocutaneous leishmaniasis known as एस्पर्डिया। इट इन्वॉल्व स्किन एंड म्यूकस मेम्ब्रेन। द वेक्टर इज सैंडफ्लाई, यहाँ पे साइंटिफिक नेम याद करने की जरूरत नहीं है। Mexicana, Leishmania mexicana causes a type of cutaneous leishmaniasis also known as Chiclero ulcer, and the vector again over here is also sandfly. ठीक है, then morphological forms of leishmania. A mastigote, A means without, A means without, masti से समझ लो flagella, which looks like a whip. ठीक है, तो whip या flagella is absent, मतलब A flagellated. In humans, it is found in reticuloendothelial system, which is rich in macrophages. Reticuloendothelial system मतलब कि liver, spleen, bone marrow. It also invades what? Neutrophils and endothelial cells. And what is LD body? LD body is nothing but a macrophage. Macrophage filled with the A mastigotes. Look at this, a very famously asked picture based question. Look at this. पहले इसको देखो, ये देखो. So this is an A mastigote. This is an A mastigote. ठीक है, and it has got a nucleus and a kinetoplast. Kinetoplast represents the mitochondrial DNA of the parasite. So kinetoplast is present perpendicular to the nucleus. And look at this gigantic structure. This whole thing is a macrophage. This is the nucleus of the macrophage, and ये जितने आपको छोटे 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 यहाँ पे सब चीजें दिख रही है ना, they are amastogotes. तो imagine करो, macrophage is like a balloon और गुब्बारा, उस big balloon में छोटे 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 small balls are present. Those balls are nothing but amastigotes. ठीक है, एलडी बॉडी को प्लीज डोनोवन बॉडी से कंफ्यूज मत करना, जो मैंने कहाँ बताई थी, डोनोवनोसिस में, विच इज कॉज बाय क्लेप्शिला ग्रैनुलोमैटिस, जिसका पुराना नाम क्या था, कैलीमैटोबैक्टीरियम ग्रैनुलोमैटिस। ठीक है। देन प्रोमास्टिगोट, प्रो प्रोमास्टिगोट, प्रो मीन्स टू हैव समथिंग। ठीक है। मैस्टी मतलब फ्लैजिला, सो दिस इज द फ्लैजिलेटेड फॉर्म विच इज देयर इन द सैंड फ्लाई, दैट इज द वेक्टर. This is the extracellular form, and this is the infective form for humans. So sandfly, when it bites human beings, it will discharge what into the human body, promastigote, and in the human body morphological transformation of promastigote will occur, and it will transform into amastigote. Okay. It has got a single anterior flagellum, and it is pear shaped, something like this, and this is a single anterior flagellum. Okay. And it is 8 to 15 microns in size. Now, what are the clinical features of visceral leishmaniasis? It has got a long and variable incubation period, 1 to 6 months. And the classical triad kya hai visceral leishmaniasis ka? Fever, most common symptom, high grade with chills and rigor. Hepatosplenomegaly. The most consistent signs is splenomegaly, and it is followed by hepatomegaly. Obviously, because मैंने आपको बताया कि this the e-mastigote tends to infect the reticuloendothelial system, that is liver, spleen, bone marrow, and all. That is why hepatosplenomegaly will occur. Then pancytopenia, ठीक है, pancytopenia. Then lab diagnosis of visceral leishmaniasis. What specimens we can collect? We can collect bone marrow aspirate, clinic aspirate, which is most sensitive, but obviously not collected because it is very very dangerous. ठीक है. Then liver biopsy, again it is liver biopsy is avoided because it is dangerous. Okay. So splenic aspirate also is dangerous only because of the risk of bleeding. Bone marrow aspirate is collected most commonly, and then blood sample also can be collected to look for the amastigotes, that is the ld bodies. Microscopy will see what will show you what microscopy will show you ld bodies, जो मैंने ऊपर ही describe किया हुआ है, ld bodies plus free amastigotes also may be seen in the blood. Now, culture. Very importantly, culture, matlab ki specimen of the patient in which amastigotes, that is LD bodies, will be there. When you put the specimen on these culture media, NNN medium, Schneider's and Toby's medium, you will not see amastigote form. Culture grows कौन सा form? Interestingly, it grows the promastigote form. पता है क्यों? क्योंकि ये जो culture media ना, remember, they are like artificial sandfly, meaning they provide an environment like sandfly only. So when you add the sample to any of these culture media, the sample which is having a mastigote, imagine it is entering artificial sandfly, and sandfly में क्या होगा, the a mastigote has to get converted into the pro mastigote form, है ना. तो रिमेंबर ह्यूमंस के लिए इन्फेक्टिव फॉर्म क्या है जो सैंडफ्लाई में है. सो फॉर ह्यूमंस इन्फेक्टिव फॉर्म इज द प्रोमास्टिगोट, एंड फॉर सैंडफ्लाई रिमेंबर, फॉर सैंडफ्लाई जो देखो जो हमारे पास होता है दूसरों को हम वही देते हैं ना. तो सैंडफ्लाई के पास क्या है, प्रोमास्टिगोट फॉर सैंडफ्लाई. तो सैंडफ्लाई के पास प्रोमास्टिगोट है तो ह्यूमंस को वो प्रोमास्टिगोट देगी, तो ह्यूमंस के लिए इन्फेक्टिव फॉर्म इज प्रोमास्टिगोट. एंड ह्यूमंस के पास क्या है देने के लिए, a mastigote. So for sandfly, the infective form is a mastigote. So let's say if there's a patient of leishmaniasis, if some innocent sandfly bites that human being, then during the blood meal, what will be picked up by the sandfly? A mastigote. Then this a mastigote will undergo plastic surgery in sandfly ki body. It will get transformed into the pro mastigote. Okay. Now, serology. Antibodies can be detected by direct agglutination, test. Okay. And there is an ICT, immunochromatographic test, which detects the antibodies against the RK139 antigen also. Okay. Then molecular method, real time PCR can be detected to detect the kinetoplast DNA of the organisms. Remember मैंने ए mastigote में बताया था ना कि न्यूक्लियस के परपेंडिकुलरली काइनेटोप्लास्ट प्रेजेंट है व्हिच रिप्रेजेंट्स द डीएनए ऑफ द पैरासाइट. तो उस डीएनए को भी डिटेक्ट कर सकते हैं विद रियल टाइम पीसीआर. एंड व्हाट इज द ड्रग ऑफ चॉइस? व्हाट इज द ड्रग ऑफ चॉइस? पेंटावेलेंट एंटीमनी कंपाउंड्स. ठीक है. Miltefosine is the only oral drug for kala-azar, can also be given in resistant cases. ठीक है, एंड रेसिस्टेंट केसेस में और क्या आप दे सकते हो, इवन एम्फोटेरिसिन बी भी दे सकते हो. Amphotericin B also can be given. Any doubts so far? Any doubts? Next, we come to cystodes or tapeworms. So these are a kind of flat worms with a segmented body. A segmented body, and the body segments are known as proglottids. Proglottids are the name given to the body segments. Okay. So these are the five important cystodes or tapeworms. ठीक है, फ्लैट वर्म्स मतलब कि दे डोंट हैव एनी बॉडी कैविटी, अनलाइक राउंड वर्म्स लाइक एस्कैरिस एंड ऑल व्हिच हैव गॉट अ सिलिंड्रिकल बॉडी, तो उनके पास ज्यादा रूम या जगह है टू फॉर द डेवलपमेंट ऑफ ऑर्गन्स एंड ऑर्गन सिस्टम्स. so roundworms obviously are evolutionary more advanced as compared to cystodes and trematodes. ठीक है, तो टीनिया साजिनाटा के केस में द इन्फेक्टिव फॉर्म, सबसे पहले हम डेफिनेटिव इंटरमीडिएट होस्ट लिख लेते हैं. द डेफिनेटिव होस्ट humans. ठीक है, intermediate host are bovines. Taenia solium में humans, the intermediate host are pigs. Now the infective form is the, in case of Taenia saginata, is the larval form Cysticircus bovis. Bovis comes from bovine, so this is the larva. And Taenia solium के केस में Cysticircus cellulose, this is the larva. So when the infective form, देखो ध्यान से सुनना मेरी बात अब, टीनिया साजिनाटा या टीनिया सोलेम, अगर हम लार्वा इनका इंजेस्ट कर रहे हैं, लार्वा विच इज देयर इन बोवाइंस, इफ इन if inadequately cooked pork is eaten or beef is eaten which has got the larva, then what disease will occur in humans? Intestinal teniasis. Both can cause intestinal teniasis, please remember this. ठीक है, अभी टीना सोलम के बारे में मैं एक और चीज बताऊंगी कि इट कॉजेस सिस्टिसर्कोसिस, बट टीना साजिनाटा डज नॉट कॉज सिस्टिसर्कोसिस. नाउ एच नाना के केस में इन्फेक्टिव फॉर्म इज द एग. तो यहाँ पे देर इज ओनली वन होस्ट, देर इज ओनली वन होस्ट, दैट इज ह्यूमंस. There is no concept of a definitive or intermediate host. Echinococcus granulosus के केस में the definitive host is dog and the intermediate host is what? Sheep. ठीक है, humans remember are the accidental intermediate host, and for us the infective form in that case will become what? The infective form is going to be the larva, which is known as hydatid cyst. Sorry, infective form, wait, the infective form हमारे लिए eggs हैं which are present in the uh, pieces of the dog. How many of you are dog lovers? How many of you are dog lovers? Okay. Please remember one thing. If a dog is infected with Echinococcus granulosus, it is going to pass out, eggs in the feces. And if you're playing with the dog and you don't wash your hands before eating food, you are actually ingesting the eggs. So for you, for us humans, the infective form is the egg. And once we ingest the eggs, we will develop larval form in our body, and that disease therefore is known as hydatid disease. Okay? And lastly, we have got Diphtheria latum. In this case, the definitive hosts are humans, and the intermediate hosts are two, first and second. Okay? First one is cyclops and the second one are fish. दोनों ही पानी में रहते हैं. ठीक है? And the infective form is again larva present in the fish. तो fish को खाने से infection होगा. Now, eggs of all the cystodes have got hexacanth embryo, also known as oncosphere. It has got these 1 2 3 4 5 6 hooklets. तो अभी से क्या बोलते हैं? Hexacanth embryo. Both Taenia saginata and Solium, they cause what? They cause intestinal teniasis. ठीक है? And Taenia solium also causes what? Cysticercosis. You know why? क्योंकि in case human beings go on to happen to ingest the eggs of Taenia solium, the eggs can hatch in the human gut, and then larvae can be formed in the in the tissues of the humans, but this doesn't occur in case of saginata. Please remember this. And overall, most common cystode infection क्या है? एचनाना. ठीक है. देन डी लैटम, डाइफाइलोबोथरियम लैटम, इतना लंबा नाम, तो ये बड़े वाला अनीमिया कॉज करेगा. कौन सा अनीमिया? मेगैलोब्लास्टिक अनीमिया. ठीक है. तो आज हमने दो पैरासाइट्स पढ़ लिए. एक तो भाई अनीमिया वाले, आयरन डेफिशिएंसी अनीमिया हुक्म करता है, मेगैलोब्लास्टिक अनीमिया, इतना लंबा नाम, मतलब इतना बड़े वाला अनीमिया कौन करता है? Diphyllobothrium atom. Okay. Echinococcus granulosus causes what? Hydatid disease. Hydatid disease. The most hydatid disease, it is characterized by formation of larvae known as hydatid cyst in the tissues. The most common site of hydatid cyst is liver, and the second most common site is are the lungs. Look at this, tinea ke eggs in the saline mound. You can see these 6 hooklets. Okay, so this is hexacanth embryo. And look at this, eggs of H nana. सारे picture based questions हैं. So you can see the polar filaments. Look at this. Let me just mark them. These are the polar filaments. Okay, polar filaments. Okay, so the center has got a oncosphere and the outer shell. This is the outer shell, and you can see the polar filaments emerging from the poles. Okay. Now look at this. This is a schematic representation of the hydatid hydatid cyst or the larva, which occurs in our which gets formed in our tissues when we develop what? Hydatid disease जो कब होगा? When we ingest the eggs of echinococcus granulosus, which are passed out in the feces of the dog. So it has got all these layers. Outermost one is pericyst. ठीक है. It is made up of, as you can see, blood vessels and fibroblast. बीच वाली होती है ectocyst, which is acellular laminated hyaline layer. अंदर वाली endocyst. एंडोसिस्ट को क्या बोलते हो? एंडोसिस्ट को आप बोल देते हो जर्मिनल लेयर. जर्मिनल मतलब किसी चीज को पैदा करना, जर्मिनेशन मतलब पैदा होना, पैदाइश. तो एंडोसिस्ट और द जर्मिनल लेयर जो होती है ना, दिस इज पैरासाइट डिराइव्ड. पैरासाइट derived, and pericyst and ectocyst जो होती है ना, they are host derived. अब endocyst को germinal layer आप बोल रहे हो, it will give rise to these brood capsules, and inside them ये देख रहे हो आप एक खोपड़ी जैसे कुछ structures. These are nothing but the these are nothing but the future heads or future scolex. Scolex का क्या मतलब होता है? head. ठीक है. They are also ideally they are referred to as प्रोटोस्कोलिसिस, मतलब भविष्य में जाके ये हेड फॉर्म करेंगे ऑफ द सिस्टोड एंड डॉटर सिस्ट. एंड समटाइम्स ये खोपड़ियां डिटैच होके बॉटम पे इकट्ठी हो जाएंगी, उसे क्या बोलोगे? हाइड्रेटेड सैंड. These layers are important. Okay, now look at this D-latum egg in an unstained wet mound. So देखो, D-latum Diphyllobothrium latum है ना. L से latum, L से क्या मतलब होता है? लिड. लिड मतलब ढक्कन. ढक्कन का एक फैंसी इंग्लिश का वर्ड क्या है? ओपर्क्यूलम. सो रिमेंबर, दिस इज एन operculum is present, so it is an operculated egg, and this is a knob at the opposite side. And डी लैटम मैंने बोल दिया है आपको कि मेगलोब्लास्टिक अनीमिया करता है. एंड ऑल्सो मेक अ नोट ऑफ द फैक्ट दैट eggs of all cystodes are bile stained except what? Except nana ji, that is H nana. साफ सुथरे neat and clean nana ji, H nana, the egg is non bile stained. Non bile stained. Just a memorizing tip. Please don't take it too hard. Okay. Now we come to arboviruses. डेंगू, चिकनगुनिया, जीका, येलो फीवर, KFT एंड जापानीज एनकेफेलाइटिस. देखो, इन सब के केस में आर्थ्रोपॉड वेक्टर क्या है? एडीज इजिप्शियाई. चिकनगुनिया ऑल्सो एडीज इजिप्शियाई, जीका ऑल्सो, येलो फीवर ऑल्सो. ठीक है. कैसनूल फॉरेस्ट डिजीज वायरस के केस में हार्ट टिक, हीमो फाइजालिस, स्पाइनी गेरा, दिस इज अ हार्ट टिक. एंड जापानीज एनकेफेलाइटिस वायरस मैंने जान के सबसे नीचे लिखा है बिकॉज इसमें ट्विस्ट इन द टेल, क्यूलेक्स फर्टिगन्स. इतना इसको और क्या बोलते हैं? क्यूलेक्स ट्राइटेनियो रिंकस। तो इतना लंबा नाम ट्राइटेनियो रिंकस कि बंदा बोलते बोलते थक जाए, फटीग हो जाए। ऑल राइट। ठीक है, अब देखो डायग्नोसिस के लिए डेंगू के केस में ना फर्स्ट 4 डेज ऑफ फीवर के लिए NS1 एंटीजन डिटेक्ट करते हैं, 4th डे ऑनवर्ड्स IgM एंटीबॉडी। इनको डिटेक्ट करने के लिए एलाइजा इज अवेलेबल, एलाइजा फॉर NS1 एंटीजन एंड एलाइजा फॉर IgM एंटीबॉडी डिटेक्शन। चिकनगुनिया के लिए वायरस आइसोलेशन कर सकते हैं बट कोई करता नहीं है इतना, इट इज वेरी वेरी लेबोरियस एंड टाइम टेकिंग। Serology again, IgM antibody can be detected by ELISA and RT-PCR, that is reverse transcriptase PCR. RT-PCR is done for what? RNA viruses. All of these are what? All of these are RNA viruses. RNA viruses. ठीक है, जीका वायरस के केस में भी इन्वेस्टिगेशन ऑफ चॉइस इज RT-PCR. येलो फीवर वायरस अगेन सेरोलॉजी IgM एंटीबॉडी बाय ELISA. जब एंटीबॉडी डिटेक्ट करनी होती है ना, ELISA इज द फॉर्मेट यूज्ड मोस्ट कॉमनली. देन RT-PCR अगेन यहाँ पे भी serology and RT-PCR both can be used. Now, chikungunya is a disease characterized by what? It's a disease characterized by fever plus arthritis. Fever plus arthritis, बहुत भयंकर arthritis होता है, ठीक है, जो it may persist for months and months together. And chikungunya virus जो है, it belongs to the family Togaviridae. Togaviridae. येलो फीवर वायरस, एज द नेम implies, it causes jaundice in severe infection. सबको jaundice नहीं होता, but in very severe infection, fulminant infection, liver can be involved causing jaundice, and renal dysfunction can also be there. और यहाँ most commonly क्या पूछते हैं? In hepatocytes, intranuclear inclusion bodies in hepatocytes are seen, which are known as torus bodies. ठीक है, then dengue virus, there are not 5 serotypes, there are not 4 serotypes, there are 5 serotypes. ठीक है, this is an update, remember it. And what is the disease? It causes dengue fever, and characteristic feature, it is also known as breakbone fever or saddleback fever. Most welcome tranquil, बस अभी तो थोड़ा सा ही बचा है। ठीक है, तो ब्रेक बोन फीवर बहुत भयंकर पेन, ठीक है, सैडल बैक फीवर और सैडल बैक फीवर, रेट्रो ऑर्बिटल पेन, कॉम्प्लिकेशंस, डेंगी हेमरेजिक फीवर एंड डेंगी शॉक सिंड्रोम. हेमरेजिक फीवर because of what? Decrease in platelets, thrombocytopenia, dengue hemorrhagic fever can complicate to dengue shock syndrome. ठीक है, so look at this. मैंने एडीज पे भी अभी 2-3 दिन पहले एक पोस्ट भी डाली थी. It is also known as tiger mosquito and they can be identified by their white stripes on black bodies. They are daytime biters. एडीज के स्पेलिंग में डी आता है ना, तो डी से याद करो daytime biters. और ये देखो, ये टाइगर मस्कीटो इसलिए बोलते हैं because of these white and black stripes on their bodies, and peak feeding activity at dawn and dusk. ठीक है, now look at this. Aedes aegypti, dramatic bone pain, तो bone pain fever. Saddleback fever का क्या मतलब होता है? Several days of fever, फिर patient feel improvement दिखेगी उसको, फिर दोबारा से resumption of symptoms. And dengue hemorrhagic fever, most commonly, I mean, most notoriously caused by serotype 2. Serotype 2 is most dangerous. This also is an MCQ. Then bleeding, thrombocytopenia, and a high mortality rate. Then Japanese encephalitis in India, UP, specifically the Gorakhpur region, is the hot belt of Japanese encephalitis. Most cases are asymptomatic. Amplifier host कौन से होते हैं? पिग्स. मतलब कि पिग्स आप समझ लो आर लाइक अ जेरोक्स मशीन, जिनमें वायरस मल्टीप्लाई करता रहता है धड़ल्ले से. ठीक है, एंड व्हाट आर द रिजर्वॉयर्स? वेडिंग और आर्डीड बर्ड्स. ये वर्ड्स कौन से होते हैं? दीज आर द बर्ड्स व्हिच हैव गॉट लॉन्ग नेक्स एंड लॉन्ग legs. For example, ducks, cattle, egrets, herons. ठीक है, And horses, interestingly, are the only animals which develop what? Which develop symptomatic disease on infection. Symptomatic disease on infection. And clinical features. Features will be those of, encephalitis. Okay, the patient will feel drowsy and may go into coma, and sometimes residual neurological deficits may also be seen in the people who have recovered. Severe disease in is seen in extremes of age, infants and elderly. Okay, so that's Japanese encephalitis. This, by the way, is the most common cause of sporadic encephalitis in Asia, including Hamara Desh, that is India. Then Casserole forest disease, Ka virus, KFD virus. It was first discovered in our country only in Shimoga district in Karnataka. It causes what? Hemorrhagic fever. Humans, we are accidental host. अगर हम ऐसे जंगल में चले गए जहाँ पे ये वायरस है, then we can be infected. ठीक है. वेक्टर मैंने आपको बता दिया है. Hemophysalis. Hemophysalis spiny which is a heart tick. Humans are accidental host, and वैसे this virus causes what? Monkey fever. Monkey fever. Now there is a flavivirus which is not an arbovirus. What is that flavivirus? Answer is HCV, hepatitis C virus. Hepatitis C virus. Now coming to Zika virus. ZIKV matlab Zika virus, accepted short form for Zika virus. ठीक है. मस्कीटो बाइट हमने शुरू में ही लिख दिया था. एडीज Egypti is the vector. Mother to child transmission, it is common in first trimester. देखो भाई 3 trimester होते हैं, फर्स्ट सेकंड थर्ड. जीका जेड से शुरू होता है, विच इज द लास्ट अल्फाबेट. ठीक है, लास्ट अल्फाबेट के बाद फर्स्ट अल्फाबेट सोचो ए, तो मतलब ए का एनालोग क्या है? वन. तो फर्स्ट ट्राइमेस्टर. अगर कभी याद ना आए ना कि सेकंड ट्राइमेस्टर है, थर्ड ट्राइमेस्टर, तो ऐसे याद कर लो फर्स्ट ट्राइमेस्टर. एंड ओकेजनली कैन अकर ड्यूरिंग डिलीवरी. Sexual transmission can also occur from asymptomatic males to females and females to males. Asymptomatic males to females का मतलब है कि भाई a longer the virus has been found to be shed in semen for a longer duration of time. Incubation period few days to 1 week. Majority of the infections are actually asymptomatic. ठीक है. Symptomatic people में क्या होगा? F plus R plus C meaning fever plus rash plus conjunctivitis. देखो फीवर रैश तो उसमें भी हो सकता है, डेंगू में, चिकनगुनिया में, बट दोनों में से किसी में, डेंगू चिकनगुनिया दोनों में कंजंक्टिवाइटिस नहीं होता. दैट्स एन इंपॉर्टेंट डिफरेंशिएटिंग पॉइंट. Congenital transmission leads to what? It leads to microcephaly, and few cases of GBS, Guillain-Barré syndrome, also have been reported. And fetal infection can be diagnosed by what? PCR of the amniotic fluid. Now coming to paramyxoviruses. Measles or rubella virus, they go all paramyxoviruses are RNA viruses. If you have, you know, if your concepts are not clear regarding, positive sense or negative sense RNA, RNA DNA को याद कैसे करना है, डोंट वरी. मैंने एक यूट्यूब वीडियो किया था अपने यूट्यूब चैनल पर, उसका भी लिंक मैं शेयर कर दूंगी. ठीक है. तो उसको प्लीज देखना, 5-6 मिनट का वीडियो, आपके सारे फंडे क्लियर हो जाएंगे उससे. ठीक है. मीजल्स या रूबियोला वायरस, सीरोटाइप ओनली वन. ओनली नेचुरल होस्ट आर अस, दैट इज ह्यूमंस. बस हमें ही तंग करेगा, हमारे पीछे पड़ेगा. इट्स अ चाइल्डहुड इन्फेक्शन. इंक्यूबेशन पीरियड इज 10 to 14 days. ठीक है. Complex spots, they appear opposite the lower second molars 1 to 2 days before the onset of rash, and they are bluish white lesions on a reddish base. ठीक है. तो लेट्स से इंक्यूबेशन पीरियड हमने 10 से 14 दिन की रेंज देखी ना. अगर मान लो इंक्यूबेशन पीरियड 10 दिन है, तो मतलब डे 10 पे मान लो फीवर आया. फीवर के 2 दिन बाद, 1 या 2 दिन बाद, कॉप्लेक्स स्पॉट्स अपीयर होंगे ऑपोजिट द लोअर सेकंड मोलर. सो कॉप्लेक्स स्पॉट्स आर bluish white spots on a red base, erythematous base, and they first appear opposite the lower second molars, and later on they may become confluent. The rash is a maculopapular red erythematous rash. Okay, it starts behind the ears on the hairline and the neck. उसके बाद it spreads to the face, trunk, and arms, and finally it involves the legs and the feet. Then Warden-Finkeldy cells are the MNGs, multinucleated giant cells, which are seen in measles. Now, as far as complications are concerned, what are the common complications? The common complications include otitis media, D से diarrhea, ठीक है, pneumonia. Now, most commonly fatal complication is pneumonia. What is the meaning of this statement? Most commonly fatal मतलब कि amongst the complications which are common, उनमें से मोस्ट कॉमनली फेटल कौन सी होती है? निमोनिया. बट ओवरऑल अगर पूछे मोस्ट फेटल कॉम्प्लिकेशन, इट इज सब एक्यूट स्क्लेरोसिंग पैनएन्केफेलाइटिस, which by the way is the rarest complication. I hope this sentence, both these sentences are clear. Any doubts? Now look at this. This was a patient who presented with complex spots on palate due to pre-eruptive measles on day 3 of the illness. ठीक है. Coming to mumps virus. What is the typical feature of mumps virus? It is bilateral, non-suppurative parotitis. Sometimes the parotitis may be unilateral also. Transmission by respiratory droplets and also fomites. And apart from bilateral non-suppurative parotitis, which is the most common presentation, it can even cause what? Epididymo or chitis in post-pubertal males, which is usually unilateral. तो इसमें भी question पूछते हैं, so it does not lead to infertility. Then it can also cause aseptic meningitis. It can cause very very rarely euphoritis and also pancreatitis. ठीक है, pancreatitis. ठीक है. These, by the way, are not complications, guys. These are clinical features only. Epididymoarchitis, aseptic meningitis. These are presentation of measles. Okay, euphoritis, pancreatitis. Okay. Respiratory syncytial virus (RSV) is the MCC. Most common cause of what? It is the most common cause of bronchiolitis in infants. Please remember this. Okay. Then para-influenza virus. It's of 4 types, and it causes what? It causes the disease croup or laryngo, tracheo, bronchitis, which is characterized by what? It is characterized by barking or brassy or seal-like cough. Barking or brassy or seal-like cough. Any doubt so far? If not, can I proceed to hepatitis viruses? Yes, yes, absolutely. Steeple sign is also seen in this condition. आगे बढ़े. Hepatitis viruses. Now, there are 5 important hepatitis viruses: HAV, B, C, D, and E. What family do they belong to? देखो, private hospitals favor rich clients. P से है पिकोरना विरिडे. ठीक है. HAV का बाय द वे पुराना नाम क्या है? एंटेरोवायरस 72. H से हेपाडना विरिडे. आई रिपीट, लेफ्ट से राइट क्या है? प्राइवेट हॉस्पिटल्स फेवर रिच क्लाइंट्स, जस्ट एन निमोनिक, नथिंग एल्स. एफ से क्या है? फ्लेवी विरिडे. फ्लेवी विरिडे. मैंने ऊपर ही लिखवाया है कि विच फ्लेवी वायरस इज नॉट एन आर्बो वायरस, आंसर इज HCV। ठीक है। आर से क्या है, देखो आर से हम याद कर लेंगे कि जीनोम इज RNA, बिकॉज एक्चुअली ना HDV बेचारा अकेला है, नो फैमिली, किसी फैमिली में नहीं आता ये, इट्स टू बी अ डिफेक्टिव वायरस। सी से क्या है कैल्सीविरिडिल। Genome. Famous MCQ. It is only HBV which has got DNA as its genome. Rest of the four hepatitis viruses have got what? Have got RNA as their genome. RNA as their genome. Cultivable. Only hepatitis A virus has been cultivated, but that too with difficulty, not very easily. None of the remaining four hepatitis viruses can be cultivated. Envelope. एब्सेंट इन ए एंड ई, प्रेजेंट इन बीच वाले। तो आप ऐसे सोच लो, बीच वाले आर लिटिल प्रोटेक्टेड, कि वो बीच में है, एंड द वंस ऑन ऑन द एंड्स ऑफ द ऑन द एक्सट्रीम साइड्स ऑफ द टेबल ए एंड ई, दे आर एक्सपोज्ड, उनके पास कोई प्रोटेक्शन नहीं है इन द फॉर्म ऑफ एनवेलप। ठीक है। वैक्सीन, yes it is there for A, for B, none for C, D के लिए भी नहीं है, लेकिन HDV for its multiplication is dependent on HBV। सो अगर किसी को HBV की वैक्सीन मिली है तो दैट पर्सन विल बी प्रोटेक्टेड अगेंस्ट HDV ऑल्सो। ठीक है। एंड द वैक्सीन इज अवेलेबल फॉर HEV, बट इट इज लाइसेंस्ड फॉर यूज ओनली इन चाइना। इंक्यूबेशन पीरियड, अब देखो इंक्यूबेशन पीरियड इन वीक्स, HAV के केस में 3 to 4 वीक्स, HBV में 10 to 12 वीक्स। अब आगे मेरी बात ध्यान से सुनना। देखो इसको ना हिंदी में देखते हैं जरा, जैसे मेमोराइजिंग टिप। व्हेन वी से हिंदी में 2-12, ठीक है, 2-12, वो मूवी थी ना जिंदगी ना मिलेगी दोबारा, ZNMD। तो ये 2 इन हिंदी इज 2, 12 इन हिंदी इज 12, तो 2-12, 10। हाउ मेनी ऑफ यू नो पंजाबी? एनी पंजाबीज इन द चैट बॉक्स? ओके। There's a word in Punjabi called as das. It means to tell. And das has got another meaning in Hindi which means 10। ठीक है, तो ये 10 है मतलब 10, एंड पंजाबी में 10 का मतलब टू टेल। Okay, then you please remember 2 to 12, 10, 2 to 13. This is just for those people who know Hindi and Punjabi. I thought I'll just share this thing, this little memorizing tip for you, but I think it is not that difficult to remember this. 2 to 12, 10, 2 to 13. ठीक है। 2 13, अगेन हिंदी स्पीकिंग जनता, तू तेरा, तेरा मतलब तुम्हारा, तो दोबारा 10 तू तेरा, क्योंकि मुझे पहली बार में समझ में नहीं आया था। ठीक है। दोज हु गॉट दिस गुड, बट दोज हु डिड नॉट अंडरस्टैंड दिस बिकॉज दे डोंट नो हिंदी और पंजाबी, इट्स ओके, यू हैव नॉट मिस्ड एनीथिंग। लेफ्ट से राइट यहाँ से याद कर लो, 3 to 4, 10 to 12, 2 to 12, 10, 2 to 13। ठीक है। व्हाट अ कोइंसिडेंस निरंजन, मुझे भी। ठीक है। मोड्स ऑफ ट्रांसमिशन, फीको ओरल रूट ए एंड ई का, एंड बीच वालों का देखो BCD राइम्स विद व्हाट? ब्लड प्लस सेक्सुअली प्लस पेरीनेटली, पेरीनेटली प्लस पेरीनेटली। ठीक है। ये ज्यादा बिल्कुल भी नहीं है निरंजन दत्ता, भाई ये बहुत बेसिक है। ठीक है। अभी जब MCQs करोगे ना तो देखोगे कि ये सब कुछ कुछ भी मैंने आपके PYT से हटके नहीं डाला है आज के सेशन में। ठीक है। वो गए, आई गेस आई एम बिकॉज एक्सप्लेनिंग ऑल ऑफ दिस ना, इसलिए आपको थोड़ा सा शायद वैसे लग रहा होगा, बट ट्रस्ट यू मी, ये तो आपको मतलब करना ही है, कि इसके करे बिना मतलब इट्स नॉट गोइंग टू बी इट्स वेरी इंपॉर्टेंट। ठीक है, इसको मत छोड़ना। Now, HAV size is 27 nanometers. Chronic or carrier state, no. Risk of hepatocellular carcinoma, no. तो बड़ा सिंपल वायरस है ए वायरस हेपेटाइटिस ए वायरस. डायग्नोसिस के लिए IgM antibody detection by ELISA, which is replaced in 1 to 3 weeks by IgG antibody, which confers lifelong protection. Or what else you can do to make a diagnosis? You can show fourfold or more increase in IgG by ELISA. और विजुअलाइजेशन ऑफ वायरल पार्टिकल्स इन द फीसेस बाय इलेक्ट्रॉन माइक्रोस्कोपी. अब ये कौन करेगा? इलेक्ट्रॉन माइक्रोस्कोपी करोड़ों का आता है. इट इज बॉट बाय इंस्टीट्यूशंस, नॉट इंडिविजुअल्स. ठीक है. और कल्चर ऑफ फीसेस ये भी कोई नहीं करता है. जब हमारे पास अच्छा सा एलाइजा मेथड अवेलेबल है, देन व्हाट इज द नीड टू गो फॉर कल्चर? ठीक है. देन हेपेटाइटिस बी वायरस. देखो बी, रिमेंबर, big importance. Please remember this virus, hepatitis B virus, is the most important cause of hepatitis. Genome, it has got DNA, okay? And the enzyme DNA polymerase is present in this virus, which has got two activities: DNA dependent DNA polymerase activity and RNA dependent reverse transcriptase activity. Reverse transcriptase activity. Okay. And in the serum of the patient, there are three types of particles present. Look at this. These are the three types of particles: spheres and filaments. So this is the sphere, this is the filament. Both of them are 22 nanometers in diameter, and they constitute the surface antigen. They together are known as the HBS antigen, which is also known as what? Australia antigen. Australia antigen. Okay. And this is known as Dane particle, the complete virus particles. Look at this. So the size of the full HBV is 42 nanometers, and the ratio of spheres and filaments is to Dane particles in the serum is 1000 is to 1. So for every single Dane particle, there are 1000 of these spheres and filaments are present. And HBV hepatitis is also known as what? Serum hepatitis. It is also known as serum hepatitis. Then mode of infection: transfusion of infected blood and blood products, sexual intercourse, percutaneous, that is needle stick injury, IV drug abusers के केस में देखी जाती है. अच्छा, जब भी मान लो इंटर्नशिप में किसी को कई बार होता है ना कि नीडल प्रिक हो गई, मतलब कई बार अनफॉर्चुनेटली इंटर्न्स को हो जाता है, तो नीडल स्टिक इंजरी के केस में ना this fellow has got a maximum, this virus has got the maximum risk of being transmitted. So the risk is 30%. For HIV, it is what? It is 0.3% risk, and for HCV it is 3%. तो सबसे कम risk HIV में है. तो 30% risk का मतलब है, if there are 1000 people who get needle stick injury, contaminated needle with HBV, uh, sorry, if there are 100 people, out of 100, 30 will get the infection, 30 will get the HBV virus. In case of HIV, it is 0.3%. So there are 1000 people who get pricked with the positive needle, 3 will get infection. And in case of HCV, it is 3%. Matlab ki if there are 100 people who get pricked, 3 will get HCV, say infection. I hope this is clear. Then mother to child transmission occurs during birth, that is perinatally, and mother to child transmission depends on HBE antigen. HBE antigen indicates what? High infectivity of the virus. High infectivity of the virus. And HBC is the core antigen. It never appears in the serum. And a person who harbors HBC antigen in the serum for more than 6 months is known as a carrier person, and this carrier person is also known as somebody who has chronic HBV infection. That's the definition of carrier. ठीक है. Now serology पे बहुत सारे questions आते हैं, मैंने आगे एक question डाला भी हुआ है. सबसे पहले बात करेंगे HBS एंटीजन की. दिस इज द फर्स्ट सीरोलॉजिकल मार्कर टू अपीयर इन द सीरम. ठीक है, तो ये फर्स्ट आ जाएगा. कितना फर्स्ट आ जाएगा? इट अपीयर्स बिफोर द ऑनसेट ऑफ क्लिनिकल फीचर्स, मतलब जॉन्डिस और इंक्रीज इन द ट्रांस अमीनेसेस, मतलब बिफोर इवन बायोकेमिकल चेंजेस हैव अकर्ड बिकॉज ऑफ हेपेटाइटिस. एंड इट यूजुअली डिसअपीयर्स 1 to 2 months आफ्टर द ऑनसेट ऑफ जॉन्डिस, rarely persists beyond 6 months. And if persists beyond 6 months, then we call that person as carrier, जो मैंने ऊपर ही लिखवा दिया है. तो 1 to 2 months में आइडियली डिसअपियर हो जाता है, 1 to 2 months आफ्टर द ऑनसेट ऑफ जॉन्डिस, मैक्सिमम 6 महीने तक रहना चाहिए, लेकिन 6 महीने के बाद भी पीछा नहीं छोड़ रहा है, तो उस बंदे को, उस इंसान को क्या बोल दोगे? कैरियर है। अनफॉर्चुनेटली वो कैरियर बन गया अब। ठीक है। देन सेकंड एंटीजन, कोर एंटीजन, दिस इज प्रेजेंट ऑन द न्यूक्लियोकैप्सिड। ठीक है। इट डज नॉट अपीयर इन द सीरम बिकॉज इट इज enclosed in the HbS coat, but in the hepatocytes it can be demonstrated by what? By immunofluorescence, but ये कौन करेगा? इसकी जरूरत ही नहीं है। ठीक है। देन HbE एंटीजन, तो HbS एंटीजन मैंने बता दिया, जो कि सीरम में सबसे पहले आता है। HbC कोर एंटीजन आता ही नहीं है। फिर HbE एंटीजन, तो इट अपीयर्स इन द सीरम ईदर कॉन्करेंटली विद और शॉर्टली आफ्टर HbS एंटीजन, तो या तो HbS एंटीजन के साथ साथ में आ जाएगा, हम साथ साथ हैं, या फिर उसके तुरंत बाद आ जाएगा। ठीक है। इट इंडिकेट्स एक्टिव, इट इंडिकेट्स एक्टिव इंट्राहेपेटिक वायरल replication, and जिसे मैंने ऊपर लिखवाया था, high infectivity, and it is a qualitative marker of viral replication. It is measured for following up a patient for HPV infection. ठीक है। देन एंटी HBC मतलब कि एंटीबॉडी टू HBC। अब देखो, कोर एंटीजन खुद सीरम में नहीं आता, बट स्टिल इट इज डिटेक्टेड बाय द इम्यून सिस्टम, तो इसके अगेंस्ट एंटीबॉडी बनती है। सो एंटी HBC जो होती है ना, इट अपीयर्स इन द सीरम 1 to 2 weeks after the appearance of HbS antigen. Initially, it will be IgM antibody against the HbC antigen, in which case we will call it acute infection. Later on, it will be replaced by IgG antibody, and it will be referred to then as chronic infection. Then, anti-HbS antibody is the protective antibody. Okay? This is the only marker present in vaccinated people. If somebody has who has not ever who was never infected with HPV gets vaccination against HPV, then this marker alone will be present. And it appears in the serum after the disappearance of HbS antigen. Then anti-HBC matlab antibody to HBC, sorry, antibody, this should be antibody to HbE. It indicates low infectivity. So remember, HbE antigen indicates high infectivity, and uska counter, uska opposite, matlab antibody to HbE will indicate low infectivity. Then we have got DNA polymerase, which is a quantitative marker of viral replication. Then we come to window period, which is the time period between the disappearance of HbS antigen and appearance of anti-HbS. Okay. Now, diagnosis of HBV during window period is done by, it is done by IgM anti-HBC. So during window period, this will be positive. This will be detectable IgM antibody against the core antigen. Now look at this graph. Very good graph. Very easy. So look at this HbS antigen. So it appears first. ठीक है, सबसे पहले ये आएगा। HbC एंटीजन नहीं आएगा। HbE एंटीजन आई सेड विल कम concurrently with or shortly after HbS एंटीजन। ठीक है, कोर एंटीजन नहीं आएगा। नाउ सी द कॉरेस्पॉन्डिंग एंटीबॉडीज। ठीक है, once HbS antigen disappears over here, then its antibody will appear. And what is this period? Window period, during which how will you make a diagnosis? Look at this. During window period, anti-HBC will be such higher concentration. During window period, even antibody against HbE also is present, but that is in low titer. So in window period, what will help you to make a diagnosis? IgM anti-HBC. ठीक है। उसके बाद antibody against HbS antigen will appear, as I said, after the disappearance of HbS antigen. तो antibody against HbC core antigen वो तो आ जाएगी, even though antigen नहीं भी आया है. Then antibody against HbE antigen will appear after the disappearance of HbE antigen. ठीक है, चलो ये क्वेश्चन बताओ फटाफट से. A patient tested positive for HbS antigen, HbE antigen, IgM. ये सारे positive markers हैं. HbS positive है, HbE है, IgM anti-HbC. What is the diagnosis? Acute hepatitis, chronic hepatitis, HBV infection in recent period, post-vaccination status. Answer is A. ठीक है. HBV infection in the recent period, this is a very vague statement, ठीक है, don't mark this even though it resemble, it conveys the meaning of acute hepatitis, but अगर हमें acute hepatitis proper medical, I mean proper term दे रखा है तो हम उसी को mark करेंगे, ठीक है. I hope this is clear. Now, hepatitis C virus, it shows what? It shows antigenic variation. तभी इसके लिए वैक्सीन अवेलेबल नहीं है, ठीक है, बिकॉज द जीन which encodes for one of the envelope proteins, it shows frequent mutation and it causes what? It causes chronic hepatitis and hepatocellular carcinoma, and alcoholism increases the risk of HCC. If somebody has got hepatitis because of HCV and that person is an alcoholic also, there are very high chances that that person will end up developing what? HCC. Most of the infections are asymptomatic, okay, and even when symptoms occur, they are similar to acute HAV or acute HBV, but they are milder. And extrahepatic manifestations are common over here. Remember them as VAPM. V for vasculitis, A for arthralgia, P for purpura, M for MPGN, membranoproliferative glomerulonephritis. Then HCV is also an important cause of what? It is also an important cause of essential mixed cryoglobulinemia. Okay. How to make a diagnosis? Detection of IgM, anti-HCV by ELISA. RT-PCR is the gold standard. Remember, it's an RNA virus. So RNA viruses, RT, that is reverse transcriptase PCR, is done. Okay. Then HCV core antigen assay can also be done. And there are, remember, 6 genotypes of HCV. Now we come to a dependent virus HDV, जो बेचारा अकेला है, कोई family नहीं है, it's a defective virus. These are, remember, defective. Why it is defective? Because it depends on HPV, its big brother, बड़ा भाई, big boss, for its multiplication, because it does not have the genes for the envelope. Remember, ये भी तो enveloped virus है ना, तो gene तो है नहीं इसके पास, envelope की तो कहाँ से लाएगा? B पे dependent है, ठीक है. अच्छा, remember, डेन पार्टिकल कौन होता है? एचबीवी. तो कंफ्यूज मत करना, ठीक है. तो ये एच डी वी है. इट इज ऑल्सो नोन एज व्हाट? एच डी वी इज ऑल्सो नोन एज डेल्टा एजेंट, ठीक है. तो डी से डेल्टा एजेंट भी है, डी से डेन भी है. डेन एचबीवी है, डेल्टा ये वाला है. डेन में वैसे भी डी एन ए आ रहा है, इसको रीअरेंज कर लो तो, सॉरी, डी एन ए आ रहा है, आगे पीछे है, तो इसको रीअरेंज करोगे तो डीएनए मिलेगा, है ना. तो डी एन और ए, मैंने पहले डी लिख दिया, फिर एन उठा लिया, फिर ए लिख दिया, तो मतलब डीएनए वाले वायरस में है ये एचबीवी, ठीक है. इसलिए बता रही हूँ ताकि तुम लोग इसमें गड़बड़ मत करो, ठीक है. तो ये देखो, इसका रिप्रेजेंटेशन है आरएनए जीनोम, ये डेल्टा एंटीजन और कोर प्रोटीन, एचबीएस एंटीजन, क्योंकि एनवेलप के लिए इसको किसकी सहायता चाहिए होती है? एचबीवी की. मोड्स ऑफ इन्फेक्शन, रिमेंबर, एसबीपी, सेक्सुअल इंटरकोर्स, ट्रांसमिशन ऑफ ब्लड एंड ब्लड प्रोडक्ट्स, इन्फेक्टेड ब्लड एंड ब्लड प्रोडक्ट्स, एंड पेरीनेटली, मम्मी से बच्चा, ठीक है. अब जैसे मैंने बोला, इट विल कॉज इन्फेक्शन ओनली व्हेन एचबीवी इज देयर. तो या तो एक व्यक्ति की किस्मत इतनी खराब है कि को इन्फेक्शन हो रहा है, मतलब कि साथ साथ एक व्यक्ति साइमल्टेनियसली इज गेटिंग इन्फेक्टेड विद बोथ एचबीवी एंड एचडीवी. दूसरी पॉसिबिलिटी क्या है? ऑलरेडी ऑलरेडी बैंड बजा हुआ है, पर्सन इज ऑलरेडी इन्फेक्टेड विद एचपीवी, अब उसको ऊपर से एचडीवी. इसकी भी किस्मत खराब ही है कि एक वायरस ऑलरेडी मतलब झेल रहा है, एक और भी आ रहा है, ठीक है. तो दिस इज नोन एज सुपर इन्फेक्शन. रिमेंबर, सुपर इज ऊपर इन सिवियरिटी. Obviously, when liver is already damaged by one kind of hepatitis virus, then if an additional hepatitis virus is causing infection, so the person will have more severe infection, so increased chances of fulminant hepatitis, chronic disease, and HCC. ठीक है. गुड मॉर्निंग. ऑल राइट. तो को इन्फेक्शन इज लेस सीवियर, ठीक है, अभी लेस सीवियर. इट हैज गॉट डिक्रीज चांसेस ऑफ ऑल ऑफ दीज, डिक्रीज चांसेस ऑफ फल्मिनेंट हेपेटाइटिस, क्रॉनिक डिजीज, एचसीजी, ठीक है. अब देखो, को इन्फेक्शन में डायग्नोसिस कैसे करोगे? कॉमनली आस्क्ड क्वेश्चन. मोस्ट वेलकम ओएदानी. यस यस, प्लीज प्लीज. देखो, मेरी मेहनत का फल तुम्हें तभी मिलेगा जब तुम लोग इसके बाद भी अपनी पूरी ईमानदारी से इसको प्लीज रिवाइज करोगे, और कोई भी डाउट आया तो उसके लिए मैं हूँ. अभी बस अब एक पेज और बचा है, उसके बाद बस प्लीज stay with me for 2 minutes. I have to tell you few important things. I will just take 2 minutes. जब इतना बैठे हो तो 2 मिनट और मेरी रिक्वेस्ट सुन लेना. 2 मिनट बैठ जाना और ठीक है. नाउ डायग्नोसिस. को इन्फेक्शन मतलब साथ साथ हो रहा है इन्फेक्शन. तो हाउ टू मेक अ डायग्नोसिस ऑफ एक्यूट एचबीवी. IgM antibody against the core antigen, तो IgM anti HBC. And how to make a diagnosis of acute HDV. IgM antibody against HDV. इसमें कोई डाउट तो नहीं है किसी को? ठीक है. अब सुपर पे आते हैं. सुपर पे सुपर इन्फेक्शन मतलब ऑलरेडी बेचारे इंसान को ना ऑलरेडी उसकी बैंड बजी हुई है, वाट लगी हुई है उसकी कि एचबीवी हो रखा है. तो क्रॉनिक है उसको इन्फेक्शन. तो क्रॉनिक एचबीवी को डायग्नोस करने के लिए आईजीजी एंटी एचबीसी. और अभी नया नया कौन आके उसके सर पे बैठा है? एचडीवी. आईजीएम एंटी एचडीवी. ठीक है. आई होप देयर इज नो डाउट इन दिस. इज वेरी मतलब दिस इज वेरी फ्रीक्वेंटली आस्क्ड. ठीक है. अब हम आते हैं HEV, hepatitis E virus. E stands for endemic or enteric. No chronic or carrier state, no HCC का risk. Clinical features are similar to HAV infection. Case fatality rate is 1 to 2%. But in pregnant ladies, इसको 1 to 2 को multiply कर दो, it becomes 10 to 20%. So remember, in pregnant, that is expectant. Expectant ladies, HEV causes a high mortality rate, 10 to 20%. इंटेंस प्रोराइटिस या खुजली बिकॉज ऑफ कोलेस्टैसिस, जिससे ना बहुत तकलीफ होती है. आई पर्सनली नो द स्टोरी ऑफ अ पर्सन जिसको हुआ था HEV और बिल्कुल खुजली खुजली मतलब बहुत डेबिलिटेटिंग अवस्था हो जाती है व्यक्ति की. डायग्नोसिस के लिए IgM एंटी HEV बाय ELISA. And it is more prevalent in developing countries. There is no vaccine except in China and there are no antivirals. Now congenital rubella is characterized by what? The triad of, इसको ऐसे याद रखो, दिल लगा के अंधा बहरा होना. दिल लगा के मतलब क्या लिखूं? पेटेंट डक्टस आर्टीरियोसिस, अंधा बहरा. आइस का फीचर कैटरैक्ट, ग्लोकोमा. बहरा ईयर्स. सेंसरी न्यूरल हियरिंग लॉस. Is this clear, everyone? If any doubts are there in today's session, please let me know. Okay, guys, I hope I am visible to you. Okay, now we have come to the end part. Acha, just a couple of things I would want to tell you all. By the way, first and foremost, my profuse thanks to each and every one of you from the core of my heart, lung, liver, kidney, pancreas, कि आप इतनी देर तक बैठे मेरे साथ. ठीक है, मेरे साथ माइक्रो पढ़ी, रिवाइज करी साथ साथ. एंड इसके बाद भी प्लीज इस PDF को आप रिवाइज करना अच्छे से. एंड इस PDF के फर्स्ट पेज पे माय सोशल मीडिया हैंडल्स आर देयर. All right, I have got my own microbiology app also. I recently launched it. उसका नाम है MJ Micro. Am I audible, everyone? Just let me know in the comment section. There's a little lag in the video. ठीक है. So अभी आपको करना क्या है? PDF रिवाइज करो. I have tried to add most of the topics, but yes, few topics like giardia and all, cholera इसमें added नहीं थे. But आप देख लो कि इन टॉपिक्स को करने में हमारा इतना टाइम गया है. सो ऑल ऑफ दीज ना आई विल कीप शेयरिंग नोट्स एंड क्वेश्चंस विद यू. तो हम इनको भी बाकी टॉपिक्स को भी रिवाइज करेंगे, ठीक है, जो आज हम नहीं भी कर पाए हैं, ठीक है, जो आज हम नहीं भी कर पाए हैं. All right. So my humble request to you is that be very mindful of the fact कि अगर आपने आज ये क्लास अटेंड करी है, आप इसको प्लीज जितना जल्दी हो सके रिवाइज कर लो. बिकॉज अगर आप इसको एक हफ्ते बाद रिवाइज करोगे ना, फिर इसका कोई पॉइंट नहीं है, ठीक है, उस चीज का ना कोई पॉइंट नहीं है. ऑल राइट. एंड just guys have a look at this as well. So as I said, see, my app MJ Micro is available on both Play Store as well as Google Store. Okay. Very soon, most likely by next week, I'll be starting a series of quizzes also on this app, and I will conduct some live sessions as well. Please download it. And those live sessions, by the way, for FMG exam, they will be free. Don't worry. And even the quizzes, they will be absolutely free. You just have to download the app and stay connected with me. And tomorrow in my इंस्टा स्टोरी आई विल शेयर द लिंक ऑफ अ व्हाट्सएप ग्रुप. ठीक है, वो आप ज्वाइन कर लो. उसको मैं टाइटल FMG एग्जाम जैन 2026 के नाम से ग्रुप बनाऊंगी. ठीक है. थैंक यू सो मच ट्रैंक्विल, दैट मीन्स अ लॉट. ठीक है. So the first thing, please download the app. Second thing, please join the WhatsApp group tomorrow. Please revise the PDF and I'm there for your doubts. ऐसा नहीं है कि आज का सेशन खत्म हो गया बस तो खत्म, टाटा बाय बाय, ऐसे नहीं है. I'm there with all of you for solving your doubts, addressing your queries till your exam. आप सबका एग्जाम बहुत अच्छा होगा. एंड मैं एक ही चीज बोलना चाहूंगी कि देखो, हमारे हाथ में सिर्फ क्या होता है ना, हमारी मेहनत ही होती है, सिर्फ हमारा हार्डवर्क होता है. तो कभी तुम्हें टेंशन हो ना एग्जाम की, जस्ट टेल योर सेल्फ कि आई एम डूइंग माई बेस्ट एंड लीव एवरीथिंग टू दी ऑलमाइटी. आपके हाथ में सिर्फ अपना कर्म करना है. भगवत गीता भी यही कहती है, कर्म करो और फल के बारे में मत सोचो. ठीक है. तो बस ये एक छोटा सा मैसेज मैं देना चाहती थी कि कभी उदास हो तो तुम मेडिटेशन वगैरह कर लो. और उदास होने की वजह से जरूरत नहीं है. यू ऑल आर डॉक्टर्स, यू आर वेरी वेरी ब्लेस्ड, बिकॉज हर एक की किस्मत में नहीं होता कि नाम के साथ डॉक्टर प्रीफिक्स लगा सके, है ना. तो आई एम ऑलरेडी वेरी वेरी प्राउड ऑफ यू, ऑल ऑफ यू. एंड आगे भी यहाँ तक आए हो तो आगे भी बहुत जाओगे, लाइफ में बहुत आगे जाओगे. बस अपना बेस्ट दो. अपने आप को तुम्हें फेस करना है कि हाँ, क्या मैंने अपना बेस्ट दिया. किसी फ्रेंड से कंपेयर मत करो. अगर आज आपने लेट्स से एक सर्टेन अमाउंट ऑफ टॉपिक्स करे हैं, तो कोशिश करो कल थोड़ा सा एक आधा टॉपिक एक्स्ट्रा करो. ठीक है. सबकी अपनी अपनी स्पीड होती है, अपनी अपनी ग्रास्पिंग पावर होती है. सो नेवर कंपेयर योर सेल्फ विद एनीबडी एल्स. Just do things at your own pace and make sure for all 19 subjects you do the previous 4 to 5 years PYTs very very well. And the mantra for success is revision, revision, revision of the relevant topics. अगर आप बहुत रेयर टॉपिक्स रिवाइज करे जा रहे हो, करे जा रहे हो और सोच रहे हो कि मैं तो बहुत रिवाइज कर रहा हूँ और कर रही हूँ, उसका भी कोई फायदा नहीं है. तो रेलेवेंट चीजें रेलेवेंट मैंने आपको बता दी है. PYTs इंपॉर्टेंट होने चाहिए. तो ये PDF अच्छे से प्लीज रिवाइज करना. मोस्ट वेलकम ओसन. PDF आई विल शेयर विद द टीम, जस्ट गिव मी 5 मिनट्स, क्योंकि ना शुरू वाला पार्ट जो ग्रीन ग्रीन था मैं उसको रीराइट करके अभी टीम से शेयर करती हूँ. आई थिंक इट विल बी अपलोडेड ऑन द टेलीग्राम ग्रुप इटसेल्फ. ठीक है. डॉक्टर भानु प्रकाश सर का जो टेलीग्राम ग्रुप है ना, आई थिंक वहीं पे आएगा. ठीक है. तो उस ग्रुप पे तो आप लोग सब कुछ हो. यू कैन ज्वाइन माय टेलीग्राम ग्रुप ऑल्सो. तो आई एम ऑन ट्विटर, इंस्टाग्राम पे आई एम वेरी एक्टिव. ट्विटर पे भी हूँ वैसे. रिसेंटली आई हैव बिकम एक्टिव. टेलीग्राम ग्रुप यू कैन ज्वाइन एंड व्हाट्सएप चैनल इज ऑल्सो देयर. तो मेरी सोशल मीडिया लिंक्स के बारे में फर्स्ट पेज पे लिख रखा है. ठीक है. ओएदानी, अच्छा, इंस्टाग्राम जिनका नहीं है, देखो फिर यू विल अनफॉर्चुनेटली लूज आउट ऑन सम इम्पोर्टेंट स्टफ. तो प्लीज मेक एन इंस्टाग्राम अकाउंट ओनली फॉर द पर्पस ऑफ योर एकेडमिक्स टिल योर एग्जाम्स एंड फॉलो योर टीचर्स. लाइक आई शेयर एक्सक्लूसिवली माइक्रोबायोलॉजी स्टफ. ठीक है. तो मेरे एकेडमिक पेज पे आपको सारी काम की बातें मिलेंगी जो आपके लिए इंपॉर्टेंट है, आपके एग्जाम के लिए. तो इंस्टाग्राम पे मेरी इतनी इंपॉर्टेंट पोस्ट हैं, अपडेट्स हैं, MCQs हैं, तो भाई उसको प्लीज मिस मत करो. अपनी पढ़ाई लिखाई के लिए एक इंस्टाग्राम हैंडल बना लो और एग्जाम के बाद उसे डिसेबल कर देना, है ना. Now let me know if you guys have any doubts from today's session or anything else you want to ask me regarding your studies or microbiology or anything else. I am here. एंड वन मोर थिंग गाइस, अभी ना रिसेंटली मैंने एक INICT का फ्री माइक्रो सेशन अपनी ऐप पे लिया था। उसका PDF इज देयर ऑन माई टेलीग्राम ग्रुप, एंड वो फ्री वीडियो है। यू जस्ट हैव टू डाउनलोड द ऐप, गो टू ऑल कोर्सेज एंड क्लिक ऑन दैट INICT। तो उसके भी टॉपिक्स आपके आपके FMG एग्जाम से ओवरलैपिंग है। आपको बैठ के नोट्स वोट्स कुछ नहीं बनाने हैं, उन टॉपिक्स की जो मैंने ऑलरेडी आज कवर कर ली है, ठीक है। मान लो उसमें आपको कुछ और टॉपिक मिल रहा है, तो वो आप प्लीज अपने इसमें नोट्स में ऐड कर लेना। तो आई थिंक यू विल बेनिफिट फ्रॉम दैट वीडियो ऑल्सो, तो उसको भी देख लेना। दैट्स एन एब्सोल्युटली फ्री वीडियो। मैं PDF रीशेयर कर दूंगी अपने टेलीग्राम ग्रुप में। यू कैन ज्वाइन माई टेलीग्राम ग्रुप। ओके। मिस्टर टेकी, अब वेटेज के पीछे मत जाओ, क्योंकि ये ना, ये एग्जाम जो भी लोग सेट करते हैं ना, आप उन पे बहुत ज्यादा, मतलब उनकी बात को बहुत सीरियसली मत लिया करो कि अच्छा माइक्रो से 15 क्वेश्चन आ रहे हैं। माइक्रो से 15 नहीं, 25 भी आ सकते हैं, 20 भी आ सकते हैं, 10 भी आ सकते हैं। तो दे डोंट डिलीजेंटली और स्ट्रिक्टली फॉलो द क्वेश्चन डिस्ट्रीब्यूशन, ठीक है। आपके हर सब्जेक्ट की पकड़ आपको होनी चाहिए। 19 सब्जेक्ट्स इंपॉर्टेंट है। ऐसे प्लीज मत करना कि मेडिसिन सर्जरी में अगर ज्यादा वेटेज उन्होंने दे रखी है तो आप बस वही पढ़ते रहो, बिकॉज काफी ना ओवरलैपिंग टॉपिक्स होते हैं, लाइक इन्फेक्शंस। जैसे मैंने आज बताया कि कुछ कुछ ड्रग्स वाले क्वेश्चन होंगे, दे आर ओवरलैपिंग बिटवीन माइक्रो फार्मा। डिटेल में किसी ड्रग के बारे में पूछा तो ऑब्वियसली वो फार्मा में कवर्ड होगा, है ना। तो जैसे फॉर एक मैंने तुम्हें बताया बल्जिंग फिशर साइन, तो वो डेफिनेटली आपको रेडियो में अच्छे से बताया जाएगा, है ना। तो कई सारे ना ओवरलैपिंग टॉपिक्स हैं। तो पिछले 3 से 4 साल के PYTs ऑफ ऑल 19 सब्जेक्ट्स आपको करने हैं। इस बात के पीछे ज्यादा नहीं पढ़ना है कि 10 क्वेश्चन बस लिख रखे हैं पीडियाट्रिक्स के, नहीं, ज्यादा भी आ सकते हैं, ठीक है। लेकिन अगर आप ये रूल माइंड में फॉलो करोगे कि मेरे को सारे के सारे PYTs करने हैं पिछले 3 से 4 साल के ऑफ ऑल 19 सब्जेक्ट, तो आप सेफ जोन में आ जाओगे। फिर आपको कोई नहीं रोक सकता, ठीक है। यस, दैट्स गुड। आई एक्सपेक्ट यू ऑल टू गिव योर बेस्ट, ठीक है। तो गिव योर बेस्ट एंड लीव द रेस्ट टू दी ऑलमाइटी। कुछ भी डरने की बात नहीं है, इट्स जस्ट अ फेस। एग्जाम्स तो आते ही रहते हैं, एग्जाम आएगा तो फिर जाएगा ना। कभी ज्यादा टेंशन हो तो ये सोच लो कि भाई एग्जाम के बाद सेलिब्रेट कैसे करोगे, कहाँ घूमने जाओगे, ठीक है। तो उससे थोड़ा मूड अच्छा रह जाता है। Yes, asteroid class is finished. I was just asking about any doubts if you guys have, or anything else you want to ask me. And I was telling about my Instagram handle and my microbiology app, जो please download कर लेना। उस पे next week से I will start conducting quizzes, and कल I will share that link of the WhatsApp group which I will create tomorrow. वो you can join please. Thank you so much, Usanna. Okay, guys, if no doubts are there, shall we call it a day? Or I should say, shall we call it a morning? Once again, my profuse thanks to all of you for staying with me till the end. Good morning. चलो फिर गाइस, अभी आप रेस्ट करो, आराम करो, ठीक है। टेक केयर एवरीवन, एंड गॉड ब्लेस यू ऑल, नॉट जस्ट फॉर योर एग्जाम्स बट एवरीथिंग, यू नो एवरीथिंग इन लाइफ, इन एवरी डोमेन, एवरी स्फेयर ऑफ लाइफ, पर्सनल लाइफ, प्रोफेशनल लाइफ, family life, everything. May God fulfill all your wishes. So on that note, I will end the streaming. Thank you and good night and bye bye.